Good morning and welcome to day 2 of the AMALathon. Welcome to AMALathon 1 2025. Welcome to day 2. Full GCSE Paper 1 revision with myself, Mr. Tanvir, Biology, Chemistry and Physics. So if anyone's currently logged in, this is a chance for you to ask any questions and I'll be giving some answers. Now yesterday, whatever questions were asked, I did answer them. So it's possible no one has any questions, but if anyone has thought of any questions since then, they can ask those questions. So if anyone has any questions, this is a good chance to ask. Otherwise, I'm going to be giving everyone some advice on how to revise, but I'm going to wait for just a few more people to log in till then. And that'll be just before we start at 10 o'clock. Today we're going to have a sharp start at 10. So we're using the full hour, we're using the full time to get through the content. As you know, yesterday we did have some issues just starting up, but that's all been smoothed out. We're starting promptly at 10 today. Thank you, Dean, for your question. So we are going to be doing a working scientifically and how to tackle questions. That's going to be as part of the pre-exam workshop. When I go through any questions, I will, if we have time, I'll go through them in more detail. But because as part of the process, what we made the initial AMALathon for is just to ensure everyone's understanding is good. So what we will be doing is as soon as AMALathon 2 kicks in, before every exam we'll be doing the pre-exam workshops, and we'll cover that in a lot more detail. But since you've requested it, whenever I get the opportunity, I will delve into it a little bit deeper than I was going to. I'm not sure exactly how many views we've gotten at the moment because YouTube's just refreshing. However, if anyone has a question, we've got 7 minutes till we start and you can ask whatever you want to ask. If it's outside of yesterday's topic, that's not a problem because we have those 7 minutes, and if I can answer the question now, I'll answer the question now. Okay, Night Shadow, so you asked, could you explain osmosis again? Osmosis is one of the topics I'll be going through today. So I'll wait till everyone's here. Dean, you asked about releasing the notes for today. I'll wait for the admin staff to message back because they'll have a better understanding of that. So Night Shadow, Dean, how did you find yesterday's session? You can be brutally honest, don't worry. Thank you, Dean. And thank you, Night Shadow. Although I'm sure your teachers at school are amazing. When you see the same person every day, then sometimes our opinion of them can change, but I'm sure they're amazing teachers. So the lesson's going to start at 10 o'clock, exactly 10 o'clock. Thank you for your question, M. Yeah, Nadia, what I can do is tomorrow in our question and answer session, and this is an answer to Dean's question as well. What I can start to do is we can actually use the question and answer session just to go through some past paper practice. So I'll pick out some 6-mark questions and we'll go through the theory behind those 6-mark questions. We'll look at how to break those questions down. So if you guys are able to get on by Q2, my initial focus will be to answer any questions people have asked that were left unanswered. Because they are, and yesterday I think I answered every single question, but just in case I do leave a question unanswered. And then whatever time is remaining, we can go through some 6-mark questions. Like I said earlier, because we're using the AMAL process, the initial thing I want everyone to leave with is a really good understanding, that you've understood every single specification point. Once we've covered that point, when you guys come back to me, hopefully for the AMALathon part 2, when we do pre-exam workshop, we'll be going through just past paper questions. It's going to be a question, going through the content that's linked to that question, showing you how to break down the question, how to answer it, and then going through the answer, and then showing you how to link, showing you how to understand the mark scheme better. Because the more past paper questions you do, the better you're going to get, the better your grades are going to get. But the first step has to be the understanding. The pre-exam workshop will be the weekend before every single exam. So the pre-exam workshop, when we're walking through the questions, these spec points are not Edexcel, these are AQA. The Edexcel session starts at 1:30. So it's AQA from 10 a.m. till 1 p.m. and Edexcel starts at 1:30. AQA 10 a.m. till 1 p.m. and Edexcel is 1:30 till 4:30. So if you're doing combined Edexcel, it's better for you to join the 1:30 session. At the moment, you won't have missed too much because the initial topic 1 is very similar in both physics, in both AQA and Edexcel. But this point onwards, everything starts to change. They're very different specifications. All right, everyone, we're going to start in 1 minute. All right, good morning, everyone. Welcome back to AMALathon 1 2025. This is day 2, where we're covering the full GCSE specification for AQA. So our sessions will be from 10 a.m. till 1 p.m. What we're aiming for today is to cover all the way from 1.2.1, which is chromosomes. We want to finish all of topic 1, and we want to start topic 2. In topic 2, we're hopefully going to get up to 2.1, which is linked to digestion. So like I said before, and like I said yesterday, if anyone has a question, put it straight into the live chat and I'll do my best to answer it. And I'll do my best to answer it live. If I can't answer it live, then there will be a question and answer session every morning from 9:45 till 10 a.m. Now, if no one asks any questions, so if there's any questions that go on, if I am able to answer every single question during our session, while doing that 9:45 to 10 o'clock session is I'll go through some past paper questions, particularly 6 markers. And I'll make sure the link to what we did the day before. So chromosomes are thread-like structures. Oh, sorry, I've not shared my screen. If you excuse me for 1 second. No, I have. Okay, chromosomes are thread-like structures found in the nucleus of a cell made up of DNA and proteins. They carry genetic information in the form of genes which determine an organism's traits and functions. Now, the easiest way to understand this is if you initially consider a cell, we've got a cell, and what's in the center of the cell? Nucleus. And if we were to zoom into that nucleus, what would we see? We would see chromosomes. In fact, you would see them as singular. You don't see them as the X shape unless they've duplicated. So that's linked more to mitosis. Now, if you were to unravel one of these chromosomes, so you have a chromosome and you unravel it, what you would get is a long thread of DNA. So within your nucleus, if you zoom in, you see chromosomes. If you unravel the chromosomes, because chromosomes are tightly wound DNA, then you'd have the DNA. If you were to take a small section of that DNA that codes for one particular characteristic, that one small section, we call that a gene. So we've got nucleus, we've got chromosomes. Actually, I'll just do that like that. And the chromosomes unravel to become the DNA. So chromosomes are made up of DNA. One small section of that DNA is called a gene, and the genes code for a specific trait or characteristic. Now, for example, we have genes for eye color, genes for what type of hair we have. We've got genes for every protein within our body. Now, if one person's got blue eyes and one person's got brown eyes or green eyes, that means they've got a gene for eye color. But we've all got different versions of the same gene. Different versions of the same gene we call an allele. Okay, now I'll go through the text. Now, just as a quick reminder on what I said yesterday, during these sessions, I am covering I'm covering all of the AQA syllabus, foundation, higher, and I'm also covering combined higher combined foundation. So I'm covering absolutely everything during these sessions. So I will let people know when I cover something which is specifically higher, if I cover something which is specifically foundation, as we go through it. So at the moment, everything that we've covered, other than 1.6 yesterday, everything we've covered has been for everyone. So only 1.6 was triple award content or separate science content. So chromosomes are found in pairs. In body cells, chromosomes usually exist in pairs, with one inherited from each parent. This pairing helps maintain genetic stability and ensures proper cell function. Now, like I just said to you, chromosomes, when you unravel them, what is it? What do we have? As in, what are chromosomes made up of? They're made up of DNA. And what do we call each small section of DNA that codes for a specific trait? We call it a gene. So we have and then we have the DNA and the nucleus. So this is slightly in a different order. We have the nucleus. If you look within the nucleus, you would find the chromosomes. Within the chromosomes are made up of DNA, and DNA is made up of genes. And different versions of genes are called alleles. If you want to call them alleles, you can, but because it doesn't make a difference as long as you can write it correctly. But the British pronunciation is alleles. Now, key terms before we start looking at mitosis and the cell cycle. So chromosomes are DNA structures in the nucleus that carry genetic information and ensure proper inheritance during cell division. And mitosis is a cell division process that creates two identical daughter cells for growth, repair, and reproduction. So mitosis and the cell cycle has got three main phases. Now I'm going to go through mitosis in a lot more detail, or slightly more detail than is needed in the AQA specification. But it doesn't make a difference. It will just help you answer questions in more detail. So when you break down mitosis, so the three main phases of the cell cycle are interphase, and then you have mitosis and cytokinesis. Now, in the AQA specification, you don't need to use the word cytokinesis. It comes within mitosis. So we have interphase. Now, this whole section here, prophase, metaphase, anaphase, telophase. So prophase, metaphase, anaphase, telophase. These are the four steps. These are the four steps that occur within mitosis. And then finally you've got cytokinesis where the two cells actually split. Now, you don't need to know in the AQA specification the names of each individual phase. But if you can memorize the description of what occurs at each phase, that will help you answer questions in a lot of detail. So first we've got interphase. Interphase is where most of the cell cycle occurs. So this is where the cell's preparing to actually go through mitosis. So we're talking about 70% of this. 70% of a life cycle of a cell is interphase, when it's just getting ready so it can start to split, so mitosis can occur. And that is where the cell's DNA replicates. All the subcellular structures increase in number. The size of the cell increases overall. So let me just recap that. Your cell size, let's say it's this initial size, it doubles in size. It becomes bigger because it's getting ready for mitosis to occur. And the whole point of mitosis is for the cell to split. So if we're going to split a cell and we want our cells at the end to be identical to the original, it means we need to duplicate all the DNA. We need to increase or duplicate the subcellular structures, and we need to increase the overall size. So we need to double the amount of cytoplasm. We need to increase the amount of cell membrane. So it's all preparation for this phase here. And what's actually happening during mitosis? And this is the part I'm saying you don't need to know the words. You don't need to know the name of each separate individual part of mitosis, but you need to know what happens there. So in interphase, as we just said, the cell grows and carries out normal functions. That's the majority of a cell's life. Prophase, the chromosomes condense and become visible. Metaphase, the chromosomes line up along the middle of the cell. Anaphase, the sister chromatids separate and move to opposite poles of the cell as the spindle fibers pull them apart. And telophase, two new nuclear membranes form around the separated chromatids. So it shows it now in a bit more detail. So you can see from interphase to prophase, we can now see the chromosomes. Now, if you look at those images really carefully, what else has happened? So if you look at interphase, can you see that there's something around the DNA, around the chromosomes? That's a nuclear membrane. From interphase to prophase, the nuclear membrane breaks down. So that's one of the first things that happens. Then the chromosomes condense, meaning we can see them now. And the next step is that the chromosomes, after they're condensed in the next phase, they line up across the middle. Like I said, you don't have to call it metaphase. And during this, and as you can see, the spindle fibers, so they lined up across the middle, or the equator of the cell, and they attach to the spindle fibers. Now in the next phase, the spindle fibers they separate the pairs of chromosomes and take them to the poles at either end of the cell. And during telophase, what starts to happen is, and if you look at interphase to telophase, what's just been made? A nuclear membrane. And now we are ready for it to split. And the split occurs during what we call cytokinesis. So the cell grows in size and increases the number of subcellular structures like mitochondria and ribosomes. That's just another recap of what happens during interphase. Chromosomes align in the center of the cell. The paired chromosomes are pulled apart by the spindle fibers towards opposite ends of the cell, and the cells divide. This detail here will be enough to answer most questions on mitosis. Now the chromosomes reach, we've covered that part. Now before we actually move on, and this is different, this is something that will come up in 2.3 and 2.4. How many splits have occurred during mitosis? So we've had one split. It split once. If we compare our initial cell, it is identical to the daughter cells. We've gone from one cell to two cells. Now it's important that you know key facts like that, because some questions will ask you to compare mitosis to meiosis. So it's important from now that you remember what actually occurs during mitosis, the outcomes of mitosis. So let's have a look at a few exam style questions. Which process makes two identical new body cells for growth and repair? And the answer is, of course, mitosis. Now, there's another question that's quite common, is that, and I'll cover this again afterwards, but if you have, for example, an egg cell and you have a sperm cell, when they combine, the initial cell that's made, because if we think back to what we learned about yesterday, half the DNA is in the egg cell, half chromosomes, half the DNA or chromosomes are within the sperm cell. When they combine, you end up with a stem cell. And initially that stem cell has no specific job. It's undifferentiated. It's not specialized, it's not differentiated. Its main job is to multiply. So one cell multiplies into two cells multiply into four cells multiply into 8. And what process allows stem cells to duplicate? Or what process makes two identical stem cells? Mitosis. Now, what I've seen in the past is where a student can answer this question, no problem. The second the word stem cell comes in, they get confused. So, let's have a look at this exam style question. We've got stage 1. Now, the actual question here is you've got stage 1, stage 2, and stage 3. What's happening during each stage? So in stage 1, the cell grows and the chromosome replicates. In stage 2, one set of chromosomes is pulled to each end of the cell. And in stage 3, the cytoplasm and the cell membrane divide to form 2 new cells. So you can see what we've just covered answers this question perfectly. Now, this question says, what percentage of the total time for the cell cycle is taken up by stage 1? Now, a lot of people, when this question first came up in a GCSE paper, left the exam really confused, really unsure of how to answer it. But actually, all you've got to do is look at how many segments you've got. So you've got 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. And how many of those segments are stage 1? 1, 2, 3, 4, 5, 6, 7. So all you've got to do is turn 7 over 10 into a percentage, because it's asked for a percentage. So 7 over 10, and if you want to turn a decimal into a percentage, you want to turn a fraction into a percentage, you just times the top number by the bottom and you multiply by 100. So it is 70%. A cell divides to form 2 new cells every 24 hours. How many days will it take for the original cell to divide into 8 cells? So after 1 day, so we've got 1 cell. After the first day, they duplicate to make 2 cells. After 2 days, we double the cells again to make 4 cells. And after 3 days, it duplicates again to become 8 cells. Now, one really important thing I want you to notice about this question is that it doesn't need you to have memorized anything. Even the previous question, it doesn't require any memorization at all. What they require is understanding. And this is why when I spoke to you about the AMAL process, the initial thing is to activate your brain, as in to understand everything. And just through that understanding, what do you automatically do? You're already answering a lot of the questions because they only require understanding. Then when you start to do past paper questions, you start to understand, number 1, what the examiner is asking for. And number 2, you start to look at things from a mark scheme perspective. Because I'm sure there's lots of people here that are getting grade 5s, grade 6, and they're thinking, how do I take you up to the next level? And the thing that you need to do to take up the next level, number 1, if you've not activated your brain, you've got to activate. Hopefully by the end of our 18 hours, you'll have activated. Then the next thing is looking at past papers. Now when you're looking through past papers, you'll start to see what content keeps coming up over and over again. If a piece of content comes up over and over again, what do? We memorize it. Then we start to understand what the examiner actually means by each question. Because I'm sure everyone's gone through this before. Even as teachers, we will see questions sometimes and really have to think about what's it asking for. And what's made it easier for me to understand is because I've seen so many past paper questions, I understand the examiner's thought process. So seeing lots and lots of exam papers and lots of practice gives you that examiner's mind. It helps you to understand the examiner's mindset. So this way, when you're doing past paper practice, you're ticking off all the questions that you can answer through understanding. You then start to understand what is memorized, and you start to understand what the examiner wants, and automatically that takes you to the next level, to the next level, to the next level. So what I would advise you all, I know I've kind of paused from the theory here, but this is really important. What I would advise everyone is once the AMALathon's finished, is get yourself into a routine of doing lots and lots of past paper questions. You have to look at your own ability level because you're not just going through those past papers. You need to spend time understanding what the question means. If there's some memorization, you need to spend some time memorizing. So my initial advice would be to do a third of a paper every single day. And by that, I mean a third of a biology paper, a third of a chemistry, and a third of a physics. So every week you're going through 2 chemistry papers, 2 physics papers, and 2 biology papers. That means by the time we get to the exam, which is 5 weeks away, you'll have gone through all 7 or 8 available papers. Now, if you come across a question that doesn't make sense, you can go back to one of the AMALathon videos. You can watch one of the AMAL animations. You can watch, if AMAL animation is not available because we are a new company and we've only covered the biology animations at the moment, you can watch, if you want to watch different, there's a few different companies that do some really good animations, like Cognito is really good, Physics, Maths and Tutor are quite good. with some of the flashcards. You do whatever's necessary. Ask a friend, ask a teacher, ask someone at home, ask the local doctor. It doesn't matter who you ask. Make sure you've gone around, understood what that question's asking. After these sessions are over, you can ask questions on one of our animations as well. So if you come to any of our posts or animations and you ask a question, say, if you don't mind, could you explain this? What we can do is we can either make a short to answer your question, or we can just answer it verbally. So you can still keep in contact with us. I'll keep answering questions. Our team will keep answering questions. So when we have mitosis, and we'll do a comparison in a bit. Initially, in the initial cell, we have 46 chromosomes. That gets when everything duplicates, when the DNA duplicates, the chromosome duplicate, it increases to 92. Then when it splits, those 92 chromosomes, we end up with 46 chromosomes and 46 chromosomes. So when we say they're genetically identical, the parent cell, the original cell, and the daughter cells are genetically identical. They have the same number of chromosomes. They are both diploid. So diploid is when they've got a full set of chromosomes. All these facts that I've just said are important to remember, especially when you have questions where you're comparing haploid to diploid, when you're comparing meiosis to mitosis. Now, describe what's happening. And what's actually happening is the cytoplasm and the cell membrane are dividing to make two identical daughter cells. So from an AQA perspective, you don't have to name this process, because this is anaphase. But you don't have to say anaphase. All you have to do is say what's happening. And what's happening is the cytoplasm and the cell membrane dividing to make two identical daughter cells. One thing that's not written here and would be part of the mark scheme is that the DNA is being separated and taken to the opposite poles using the spindle fibers. So spindle fibers are pulling the DNA, the chromosomes, to opposite ends. Okay, which stage is the fastest in the cell cycle? And if you look at the data that's in front of you, they looked at 36 cells, 20 of them were non-dividing, 9 were in stage 1, 4 in stage 2, 3 in stage 2, and only 1 in stage 4. So the fact that there's only 1 out of 36 cells in stage 4 means stage 4 must be the fastest part of the cell cycle, meaning they've entered that stage and they've very quickly moved on. Again, if you look at this question, it's not a question that required memorization. It required a good understanding. Okay, stem cells. I know I've already touched on stem cells a little bit, but now we'll go through it in detail. So stem cells are undifferentiated cells, and that's the key word here, they're undifferentiated. They don't have a specific role. Their main role is just to duplicate. At that point, if you have an egg cell and you've got a sperm cell, as soon as fertilization occurs, that original cell, that's what we call an embryonic stem cell. And it is undifferentiated. It has no specific role other than to start multiplying. So one cell will become 2, 2 will become 4, 4 will become 8, and so on. They're undifferentiated cells that have the ability to divide through mitosis. So this is a key bit of learning there. It's mitosis that causes them to divide. And then they develop into specialized cell types. They play a crucial role in growth, repair, and regeneration in both plants and animals. And we're going to go through that in more detail as we go through the PowerPoint. So animals and plants contain stem cells. Stem cells are cells which are undifferentiated, which means they have no specific job other than to multiply. Stem cells multiply by mitosis. And when they do multiply, specific genes in the stem cell are turned on or off. Now, when a stem cell actually differentiates and specializes, what you've got to remember is within the stem cell, the stem cell contains all the chromosomes, it contains all of the DNA. So it has genes for every single potential protein our body can make. When it becomes, for example, a red blood cell, then that particular gene that has the coding for the red blood cell, that's the part that turns on, and other parts will turn off. So that's how the gene actually specializes. Someone in the comments wrote mitosis is diploid. That is correct. Good, thank you for adding the answer. You are more than welcome to answer each other's questions as well. I will check on the answer to make sure it's correct, but if it is correct, I'll just mention that it's correct. So now there's 3 types of stem cells. In plants, we've got meristems. And in adults, in human beings, or in other organisms, non-plant organisms, we have embryonic stem cells and we've got adult stem cells. So they're the ones that are found within animals. So what are embryonic stem cells and what are their functions? So cells become specialized when genes are switched off or on. So that's what I've just explained to you. Most cells in the human embryo become specialized after the 8-cell stage. So once they've reached 8 cells, the genes start to switch on or off and they start to become specialized cells. And however, some cells remain. These are known as adult stem cells. Now, if these are the stem cells, when they specialize into cells, those cells work together to make tissues, tissues work together to make organs, organs work together to make organ systems. Now, once our stem cell has differentiated and specialized into one of these cells, it will be that cell forever, as in that cell will keep multiplying, and it can only multiply to become that one specific cell. So a nerve cell can only multiply to become a nerve cell. A red blood cell can only multiply to become a red blood cell. A muscle cell will only multiply to become a muscle cell. Meaning, initially, embryonic stem cells are very versatile. They can become any cell in the body. But once they've specialized, that's it. They've specialized. They can't become any other cell. Now, the adult stem cell is not as versatile. It's found in the bone marrow, and it can specialize only into a few different types of cells, predominantly white blood cell, different types of white blood cells, red blood cells, and platelets. But like it says here, there's a limited number of cells that they can differentiate into. So they're not as versatile. So if you look at embryonic stem cells, they're versatile. They can become any cell in the body, but once they've specialized, they can't become any other cell other than the one they've become. If we look at adult stem cells, we've still got access to our adult stem cells, but they're not as versatile. You can only use them to become a specific certain number of cells. Now, meristems, found inside plants. Meristems are the stem cells of plants. Only meristems in plants undergo mitosis. So we find them in the roots and we find them in the shoots. Now, the meristem produce unspecialized cells that can develop into any kind of plant cell. That means a meristem can become any cell in the plant organism at any point. And they're found in the roots and shoots. What that actually means is, if you look at this as an example, you can see here that the stem cells in the green part are becoming a leaf, as in is producing more and more green cells. The roots are producing more root cells. Now, what if I took this plant and I turned it upside down? So here's the ground. If I turn it upside down, we've got our shoots on this side, buried under the earth, buried under the mud, and we've now got our roots at the top. What would actually happen, let's see if I can change my color to, there we go, is the roots will start to produce leaf cells, because they need to very quickly photosynthesize, and in other words, they'll start to produce shoots that eventually become leaves as well. And the roots, on the other hand, would start the shoots, which are now under the earth, under the ground, will start to become roots. That's how versatile plants' meristems are. So our stem cells, not as versatile. But our plant stem cells, very versatile. So what can we use meristems for? So stem cells from meristems in plants can be used to produce clones of plants quickly and economically. Because what that essentially means is you can go to a stem, you can take a few stem cells. So just from one leaf, you can imagine there's going to be lots and lots of cells, and you can develop each cell into a separate plant. The only issue would be that there would be clones and there'd be no genetic variation, but it means you can you can stop a species from going extinct. So you can use this process to clone rare species to protect them from extinction. Like I said, the issue is that there would be clones and you reduce the genetic pool, but it would still prevent extinction. Crop plants with special features such as disease resistance can be cloned to make large numbers for farmers. So that means farmers can, if there is a plant that is genetically superior, for example it has disease resistance, then farmers can then clone that one plant and make lots of them. Right, so let's summarize our 3 different stem cells. We've got embryonic stem cells, which are found in early embryos. Embryonic stem cells are highly versatile and can develop into almost any cell type in the body. So they're found in the early embryo, they're highly versatile, they can develop into any cell type in the body. This ability makes them valuable for regenerative medicine and research. So for example, someone has diabetes, embryonic stem cells potentially could be used to create a cure. Adult stem cells are found in specific body tissues such as bone marrow and can only differentiate into certain cell types. They're less versatile than embryonic stem cells, but still essential for tissue repair and regeneration. So they're still important within our body. We need them, but we couldn't make a new kidney out of it. We couldn't cure diabetes with it. If someone has a heart condition, if you had an embryonic stem cell, potentially you could take that cell and you could grow it into a new heart. But with adult stem cells, you can only make specific things, like for example, white blood cells and red blood cells, platelets. Meristems in plants are located in regions called the meristems, found at the tips of the shoots and roots. Plant meristems allow for continuous growth and enable plants to regenerate parts because they are very versatile. They can become any cell in the plant organism. So, describe the function of the meristem in the growth of a plant. The meristem produce unspecialized cells that can develop into any kind of plant cell. All stem cells multiply and divide due to mitosis. So this was a question. That's an actual GCSE question. This is what we've learned. We've learned that meristems produce unspecialized cells that can develop into any kind of plant cell. All stem cells multiply and divide due to mitosis. The mark scheme answers here. Meristem cells are undifferentiated. Meristem cells divide by mitosis. Gets you the last 2 marks. So we mentioned that here. Unspecialized, undifferentiated, you will still get the mark. So what are stem cells and what are the advantages and disadvantages of using them in medicine? Stem cells are cells that can divide to make copies of themselves and can become different types of specialized cells. So that's an advantage. They're not rejected. Now, if we were to use stem cells from a patient's body, the advantage would be that they're not rejected by the patient's body. And they can be used to treat diseases like paralysis and diabetes. Also, the disadvantages, however, are there's a risk of passing on viruses. So this is in the case where stem cells have come from a different person. And usually to deal with something like paralysis and diabetes, we're talking about embryonic stem cells, not adult stem cells. Because you do use adult stem cells for certain treatment, but adult stem cells are not as versatile. But if they were to start doing more research on embryonic stem cells, then potentially they could learn how to use embryonic stem cells to treat things like paralysis and diabetes. And there is research being done on adult stem cells too. So there is a risk of passing on viruses. The bigger issue is this. Some people disagree with the use for ethical or religious reasons. So there are some religions that consider as soon as this egg cell and sperm cell combine, they consider that to be conception. They consider that to be the point where the soul enters the body, and at that point it's a living human being. So if someone was to have an abortion, they consider that to be murder because that unborn child hasn't got a voice and may have wanted to live. So for that reason, there's a lot of issues with doing stem cell research, particularly embryonic stem cell research. So exam style question. Evaluate the use of stem cells from embryos or adult bone marrow. So this is an embryonic stem cell that we're talking about, and we're talking about adult stem cells to treat human diseases. So embryonic stem cells. So when we're saying evaluate, we're talking about advantages and disadvantages. So what are the advantages of using an embryonic stem cell, and what are the disadvantages? What are the advantages or disadvantages of using an adult stem cell to treat a human disease? So embryonic stem cells can treat more diseases, so that's an advantage, but they raise ethical issues, so that would be a disadvantage. Adult stem cells are safer to use, albeit painful to extract, and they are more accepted, as we don't have ethical issues, but they are less flexible, so that would be a downside. So overall, adult stem cells are more ethical, while embryonic ones could be more effective. So if we compare the two, embryonic stem cells, and I would advise memorizing this. So if an evaluate question comes up, because it's a very common question to evaluate, the best way to answer a question like this is to memorize the advantages and the disadvantages. So embryonic stem cells potentially could treat lots of disorders. Adult stem cells would only treat a limited number of disorders. Embryonic stem cells is painless, in the sense that removing them isn't going to cause, getting hold of embryonic stem cells isn't going to cause pain to the patient. Removing adult stem cells is painful because they are in the bone marrow. So when the injection goes through the skin, the injection needs to be strong enough not to just go through the skin. So that's supposed to be an injection I'm drawing there. It's not a particularly good one, but it's an attempt. That injection, not only does it have to pierce the skin, it has to pierce through the bone to get to the bone marrow. So you can appreciate how painful that would be for the donor. The embryonic stem cell, removing it could cause harm to the embryo, because again, either it's taken from a child that's been aborted, or if they were to take it from stem cells from a lady that's pregnant or is expecting, then you'd have to, again, this is not a very good needle that I'm drawing here, the needle will have to go through the amniotic fluid, the amniotic sac, and reach that initial ball of cells. So this would have to be before it turns into a fetus. So I've drawn, what I was attempting to draw there was a baby bump, but I think at the point where you'd be extracting it, there wouldn't really be a baby bump. So that would cause harm to the embryo, because you would try to remove from that ball of cells, that initial ball of cells, which are stem cells. They could attempt to remove one of the stem cells, but that could harm the embryo overall. The embryo can't give consent. Now with the adult stem cell, it's safe, and the donor can give consent. So like I said, important to go through that, important to memorize these key points. Okay, diffusion. So we're going to go through now diffusion, osmosis, and active transport. This is the last part of topic 1, and then we're hopefully going to try and cover the starting point of topic 2 as well. Now, diffusion is the spreading out of particles of any substance in a solution or particles of a gas, resulting in the net movement, now this is the key part here, from an area of high concentration to an area of low concentration. So it's the movement of gas particles from a region of high concentration to a region of low concentration. And it is a passive process. Now, whenever this comes up in a question, these are the three things you've got to write down to get your marks. Diffusion is the net movement of particles from an area of high concentration, second mark, to an area or region of low concentration, and diffusion is a passive process. So when you're answering questions and diffusion comes up, always mention all 3 points. Don't think that, never assume the examiner just understands. What I usually find, and I find this more with AQA students, is, and I find this a lot with AQA students who are very capable and really good at science. The ones who are, like I'm assuming everyone that's online today, the ones who are aiming for grade 9s, what happens is there's an automatic assumption everyone understands what you understand. But what ends up happening with that assumption is you miss out all the basic facts. You miss out all the easy things. So always put the obvious into your answer. So if your answer is linked to diffusion, write down what diffusion is. If your answer is linked to osmosis, write down what osmosis is. Active transport, write down what it is. Write all the steps. Don't assume just writing diffusion is enough. And sometimes that's the difference. I've seen I have had students where that was the difference between a grade 7 and a grade 9. They just weren't writing the obvious. Examples of diffusion include oxygen diffuses from the alveoli to blood in the capillaries. Carbon dioxide diffuses from the blood in the capillaries into the alveoli. Urea diffuses out of the blood plasma into the kidneys to be excreted. So let's go through these examples one at a time. So we already know what diffusion is. Diffusion can occur through a cell membrane. So we consider that's a cell membrane, and I'll just draw my alveoli really basic like this. Got red blood cells flowing through. Now, as the red blood cells arrive, they arrive oxygen deficient. We've got lots of oxygen here. Actually, I'll just draw it as circles rather than adding the two. So we've got more oxygen here. Here we've got less oxygen. But in the plasma, we'll come back to carbon dioxide. Now, because we've got more oxygen, so if we're answering a question here, what we would say is the alveoli has a high concentration of oxygen. In the blood cells, there's a low concentration of oxygen. So that means the oxygen diffuses from a region of high concentration to a region of low concentration, and it's a passive process. Now we have more carbon dioxide in the plasma than we have, so I'm just doing these dots as carbon dioxide, compared to the carbon dioxide that is in the alveoli. So what happens? We go from a region of high concentration, we've got high concentration of carbon dioxide in the plasma, and the carbon dioxide goes to a region of low concentration, which is the alveoli, and it's a passive process. Now, if you look back to last lesson, we said what the cell membrane was. The cell membrane's function is to control what goes in and out of the cell. And this is exactly what happens here. The cell membrane, you should look at a little bit like a door or a window. So if I look at my kitchen door, I can't fit my kitchen table through the kitchen door. My kitchen table will not fit through that space, but I can fit through that door. And that's what's happening when we have diffusion. Your oxygen is small enough to fit through the gap, but something big like a protein would not fit through the gap. Okay, similar thing would happen in a single-celled organism. Single-celled organisms have a large surface area to volume ratio so they've got large surface area compared to the volume. Now, what this actually means is this: if we compare an organism, and we assume let's just assume it's a cube shape, just for ease, and every length, width, breadth is 1 centimeter. So if everything is 1 centimeter, the surface area is going to equal, so if you just look at one side, it's going to be 1 centimeter times 1 centimeter, so that's 1 centimeter squared. But we have 6 faces on that cube. So the surface area is 6 centimeters squared. So let me repeat that. If we have an organism which has a 1 centimeter height, 1 centimeter width, 1 centimeter length, then its surface area of one side is going to be 1 centimeter squared, but this cube, because it's a cube-shaped organism, just for our ease, there's 6 sides, so it's 6 centimeters squared. Now if you work out the volume of that same organism, we're going to do length times width times height, because that's how you work out the volume of a cube. So it's going to be 1 times 1 times 1 which is 1 centimeter cubed. So our volume, surface area to volume ratio is 6 to 1. What that actually means is there's 6 cells on the outside feeding 1 cell on the inside. Now, if you look at us as human beings, do we have more cells on the outside or more cells on the inside? We've got more cells on the inside. So our cells on the outside can't fulfill the needs of all the cells on the inside. As in, if we were doing diffusion through our skin could not take across enough oxygen for every single cell, because we have more cells on the inside. That means if we were diffusing all our oxygen into our body through our skin, there would not be enough oxygen to feed every single cell inside the body. Same thing with glucose. We won't be able to absorb enough glucose for our respiration. So small organisms, they take in all their nutrients through diffusion. Okay, just quickly look at these questions. It said, what do you mean by passive process? And Saima and Coding G both have answered what you've written is correct. So I think it means that no energy is used. That's correct. Passive just means it doesn't require active transport. It doesn't require mitochondria or respiration. It naturally occurs. So if you think about, if you're in a classroom at school and your classroom usually has 30 students, and instead of you having 30 students, I filled it with 1,000 students, and I've just crammed that classroom up with student after student after student, and then I've shut the door. After a minute, if I open that door, naturally, which way will all the students go? They will naturally move out of the classroom. And that's what's happening in diffusion, where when you've got lots and lots of particles in one area, they naturally move into an area where there's less particles, and it doesn't require energy or an additional energy source. So yeah, that's excellent. Now, in multicellular organisms, surfaces and organ systems are specialized for exchanging materials. This is to allow sufficient molecules to be transported into and out of cells for the organism's needs. So here, the gills of a fish have a large surface area. The gills are well ventilated, so they've got lots of water flowing over it that's full of oxygen, and there's an efficient blood supply which surrounds the gills. And exchange surfaces within our lungs. So if you look at our alveoli, if you were to flatten all our alveoli out, then automatically, if you flatten out all the alveoli in our body, it would cover around a tennis court, if not more. So we're bigger on the inside. Now we need that increase in surface area from our alveoli. And also if you look at our digestive system, we've got villi and microvilli. We need those villi and microvilli to increase the surface area. That increased surface area allows us to take in enough nutrients for all the cells in our body. So our lungs are well ventilated, meaning there's lots of air coming in and out. It keeps a steep concentration gradient. What that means is, because we're constantly breathing in, we're taking in fresh amounts of oxygen, so that our alveoli are always full of oxygen. And because we're breathing out, we're removing a lot of excess carbon dioxide, so there's always more carbon dioxide in the plasma. So let me just explain that with the same kind of diagram we showed before. We always want there to be more oxygen in the alveoli. So that way, it always goes to where there's less oxygen, which is the red blood cell. And we always want there to be more carbon dioxide, I'll do this as a bigger circle, want more carbon dioxide in the plasma than inside the alveoli, so that it's always moving out of the plasma and into the alveoli. And that is helped with our breathing in and out. Good blood supply. So every alveoli is surrounded by lots of capillaries, and the capillaries are 1 cell thick, and it has a good flow of red blood cells flowing through. That means once one red blood cell is full, it continues to flow past, and then the next red blood cell comes which is deficient in oxygen. Then that gets oxygen and flows through. Then the next red blood cell comes that's deficient in oxygen, and so on and so on. It's got large surface area, which I've explained. It's got thin walls. That's really important. Thin walls provide a very short distance for oxygen and carbon dioxide to diffuse through. That means the oxygen doesn't have to travel far, the carbon dioxide doesn't have to travel far to get to its destination. And in our small intestine, what I've already just mentioned, a good blood supply helps keep a steep concentration gradient for quick diffusion. It's got thin membranes, which allows substances like glucose and amino acids to diffuse quickly, and it's got lots of mitochondria to provide energy for active transport, which moves nutrients even when diffusion isn't enough. Okay, in the roots have got large surface area as well. So, like I was saying to you guys yesterday, if you consider a root hair cell, if the root hair cell just looked like that, then only this part here would have any exchange of water and minerals. But actually a root hair cell looks like this. So the exchange can happen anywhere here. So you can see now that's an increased surface area. So through those root hairs, more water, more minerals can be taken in. It has a thin membrane, so that's a short diffusion pathway. So the water and the minerals don't have to travel far. It's got lots of mitochondria, because one of the things that come into the roots are nitrates. Very important. Now usually there's more nitrates inside the root hair cell than there is outside. So nitrates are actively transported in. We're about to cover active transport as soon as we finish this diffusion section. So it needs lots and lots of mitochondria to provide the energy for those nitrates and for other things. Now what the plant does with the nitrates is, once photosynthesis has occurred, so photosynthesis makes glucose molecule, it adds the nitrogen onto a glucose molecule, and now the plant has an amino acid. If it joins lots of amino acids together, it now has a protein, and proteins are needed for growth and repair. Exchange surface in the leaf, they have large surface area, which helps absorb more carbon dioxide for photosynthesis. Because on the underside of the leaf, you've got stomata, which are tiny gaps through which carbon dioxide can diffuse into. And it's a very thin structure, which provides a short diffusion pathway. So gases move in and out quickly. Now, of course, having a large surface area helps absorb sunlight as well, but because it's linked to diffusion, it doesn't mention that. Okay, quick exam style question. Why are lungs good at gas exchange? So number one, they've got lots of alveoli, which creates a large surface area for diffusion. The alveoli and capillary walls are 1 cell thick. They're well ventilated, and they have a good blood supply. So what we've just covered. Okay, osmosis. Osmosis is the diffusion of water from a dilute solution to a concentrated solution. Now, if anyone ever forgets which way this works, just think of the capital of America, which is Washington, DC. So it always goes from dilute to concentrated. So D to C. When we say dilute, again, it's good to understand. When we dilute something, what does it mean? So for example, if you've got Vimto or Ribena, if we dilute it, it means we've added loads and loads of water to it. And so it means when there's more water. And what do we call the Ribena initially before we add the water? We call it a concentrate, meaning you've got more of the Ribena, less water. Now again, when osmosis comes up, it's always the 3 steps that you need to mention. Osmosis. Now this is slightly, the PowerPoint has moved out of alignment slightly, but the 3 marks are: osmosis is the movement of water from a dilute solution, as in where there's lots of water, that's your first mark. So that's a high water concentration. So you know how diffusion goes from high concentration, low concentration? That's exactly what's happening in osmosis. It's just we're using a different word. But if you want to say high water concentration for dilute, that's fine. You can say high concentration. Better say high water concentration. And for concentrated, you can say low concentration. That's absolutely fine as well. So first mark, osmosis is the movement of water from a dilute solution, where there's lots of water, to a concentrated solution, where there's little water. So it's going from high concentration to low concentration. And your third mark, through a partially permeable membrane. So this bit here should have been lower down there. So 3 marks. High concentration, so dilute solution. It goes from dilute to concentrated through a partially permeable membrane. And I would always add this as a 4th mark. It's a passive process. It does not need energy. Okay, how can we determine the concentration of a potato or a piece of potato? Now, in the AQA spec, they can ask you about any tissues. So they might not ask you about potatoes. In fact, a few years ago, I had some students that came out of the exam, and their first response was, "You didn't teach us about carrots." You guys may have come across it in the news or not, but it was all over social media that AQA asked a question on carrots rather than potatoes. The theory was exactly the same, but instead of using the word potato, they just used carrots. And in the triple paper, instead of using potato, they used the word eggs. And it threw so many people off. So my point is, it doesn't have to say the word potato, but the principle is exactly the same. You do exactly the same process. So step 1, you get your equipment. So here, step 1, we're taking the potato, from the potato, using this potato borer or core borer, you put it into the potato and you take out cylinders. And these cylinders are then measured. So for example, you might decide to do it for 3 centimeters. Then you weigh it. And here it says it's 12 grams. Then in these different test tubes, the initial one you put pure water and you add your cylinder. Now you do the same thing, you get more cylinders of potato, you measure and weigh them, and you do it for different concentrations of sugar solution or salt solution. Now you leave that for a known amount of time, you remove the cylinder, you blot dry and reweigh. Now the reason why you blot dry means where you just dab it with a cloth, with a tissue. That means it absorbs only the surface level water. If you really squeeze it, if any water has entered the potato, then you're going to squeeze all that water out, and then you reweigh. And then you do that whole process again for different concentrations, and you repeat the whole experiment 2 times more. Now, that is not needed. So what we've got here is a change in mass. So we know how the mass has changed from the initial point to the point after. So you can work out the percentage change in mass for different concentrations. Now, if you put the potato into pure water, then you've got more water on the outside, less on the inside. So the water will go in, and most likely the potato will increase in size. if you have it the other way around, where you've got, for example, an 80% solution where you've got a lot of sugar in there. Now you've got more water on the inside, so water will come out, and that potato tube will decrease in size. So what are the potential sources of error in this experiment? So there might be an inaccuracy in the mass pan balance. Some cylinders might be over dried. Some people really start squeezing them, and that ruins the whole experiment. Sometimes the water can evaporate from the tubes, and when it evaporates, that means the concentration changes. So how can we determine the results from the experiment? What we do is we calculate the percentage change in mass. So that means if it's gone from 3 grams and now it's gone up to 3.5, that the percentage change will be, when you look at how much it's increased by 0.5, and we divide by the original, which was 3 grams. So 0.5 divided by 3, times that by 100, and you now have the percentage change. If it was initially 3 grams and now it's gone down to 2.5, then what's the change in mass? It's minus 0.5. So now you're going to do minus 0.5 divided by the original, which is 3, and then times that by 100, and you've now got your percentage. So you can get a positive percentage and a negative percentage. And then you plot the percentage change in mass against concentration. And once you've plotted it, concentration against percentage in mass, this point here, where you have a percentage change mass of 0, even though it's not from one of your concentrations, that point there is the concentration of the potato cell. So whenever this experiment comes up in GCSE exams, so if this question was in a GCSE exam, you might be asked to plot the graph. They might give you the graph and just say, what is the concentration of the potato cell? And what's the only thing that we need to do? We just, where that pink dot is, we write down whatever that number is. That is the concentration of the potato cell. Now I know I'm overrunning with biology, but with the amount of chemistry we're doing today, it's a little bit less, so it's okay for us to use this extra time. Now, the potato changes mass when it gains or loses water. Now, this is a common question. It might say to you, why does the potato gain in mass? Or the question might say, why did the potato lose mass? It can ask the question in lots of different ways. But what most people say is osmosis, and that's it. But if you think about it, you've got to go back to what I said to you before. Why does it increase in mass? Because water has entered the potato tubes. Why does it increase in mass? Write down the most obvious thing. And this is where people lose marks. This is where students who are aiming for grade 9 lose marks, because they don't point out the obvious. Why has it increased in mass? Because water has entered the potato. So water has entered the potato. But why has the water entered the potato? Because of osmosis. And what is osmosis? It's when water goes from a dilute concentration to it goes from dilute to concentrated solution through a partially permeable membrane. Yeah, all membranes are partially permeable. This is just an answer to one of the questions in the chat, which I think everyone's already worked out the answer to. So yeah, partially permeable membrane. Every membrane is partially permeable. Every single membrane is partially permeable. So active transport, osmosis, diffusion, they all occur. There's a partially permeable membrane there. But the only one you have to mention it for is osmosis. And what we're going to be doing, as soon as I finish this, we're going to have a 5-minute break, and then we move straight on to chemistry. Just for those that weren't here yesterday. It's actually supposed to be 1 hour sessions, but I'm going to overrun slightly with biology today, because we started a little bit late yesterday, and I'm just catching up. So we will be having a mini break. Yeah. Right, now I've covered this already, but when you download the resources, then you'll be able to go through that. Now I'm just going to finish on active transport. So active transport is the movement of substances from a more dilute solution to a more concentrated solution. So here, and it's against the concentration gradient. So your 3 marks, whenever you mention active transport, are is substances moving from an area of low concentration to an area of high concentration against the concentration gradient, meaning it's going in the opposite direction to which it would naturally go. So if you think about diffusion, diffusion is the movement of particles from a, actually I don't even need to draw it, it's already there. Diffusion, in naturally occurring, diffusion would go this way. And that's a passive process, it doesn't require energy. But what's happening with active transport is it's taking, for example, nitrates. So it's taking it from a region where there's less of them, and it's taking them to where there's already a higher concentration. So, and that means it's going against the concentration gradient. And the fourth thing that you need to add, so we could say a 4th mark, is this requires energy from respiration. So if something does active transport, what does it have to have? Lots of mitochondria. Why does it have lots of mitochondria? Because it needs energy from respiration. Why does it need energy from respiration? Because it's moving substances from a low area of low concentration to an area of high concentration against the concentrated gradient. So all these things have to connect. So this example I've already gone through. So the example of mineral ions being absorbed through plant roots from very dilute solution in the soil, I've already gone through. Naturally, we have diffusion occurring, but the plant needs lots and lots of nitrates for growth. So active transport occurs, but it has to occur faster than the diffusion. So for that, we need lots of energy. And where does the energy come from? It comes from the mitochondria. What happens in the mitochondria? Respiration. And what happens in respiration? It's glucose, which comes from photosynthesis, plus oxygen reacting together to release energy. Another example, sugar molecules are absorbed from a lower concentration in the gut into the blood. The sugar can then be used for respiration. Okay, we're going to take a 5-minute break now. So we're going to restart at 12 minutes past 11. So I'll see you all in 5 minutes. Thank you for staying on that little bit longer. I just want to get through this full presentation before we finished. So actually we're going to come back in 5 minutes, so it's 13 minutes past 11. So you'll see you all in 5 minutes. Thank you. Okay, welcome back. Let's continue. Now what we're going to do in this session, because we had a slightly longer session in the last one, this one will be a slightly shorter session. And we'll take a break at 12 o'clock, unless we finish before then. So what we're going to cover today is how the periodic table developed. So yesterday I've already covered how the periodic table is currently laid out, the different rows, different columns, the the fact that the periods show how many sorry, excuse me. So the fact that periods show how many electron shells something has, an element has, and the fact that groups show us how many electrons they have in the outer shell. So we've covered all that yesterday. Now we're going to look at developing the actual periodic table. So how did the periodic table develop over time? This is one of the first versions of the periodic table, because what scientists were trying to do, they were trying to pair things up and put things in groups that had similar reactivities, or they reacted in a similar manner. So for example, fluorine and chlorine reacted in a similar way. So that makes sense that they were in the same group. Now if you look at sodium, lithium, sodium and potassium, that makes sense that they were in the same group. Beryllium, magnesium, calcium, it makes sense that they were in the same group. But the patterns they couldn't fit the pattern across the whole thing. The reason being, it was incomplete. And some elements were placed incorrectly. The other thing was, they were doing in order of atomic mass. So if you think of carbon 12, that top number there is what they were basing the periodic table on. We now do it based on the number of protons. They were doing it based on the overall mass. Now we don't call that mass number, because mass number is the number of protons and neutrons. At that point they didn't know about neutrons. They just knew that the overall mass of the atom was 12, and they would have put it in order of mass number. And because they didn't know about isotopes, it means, so if we look at, for example, the example I gave to you yesterday about chlorine 35 and chlorine 37. Now, if they were to look at things from a chlorine 35 perspective, then chlorine would have been one element earlier on the periodic table. If they looked at chlorine 37, then the position may have been one element to the right. So for example, here carbon 12 puts it in order over here, but carbon 14 has the same atomic mass number as nitrogen. So that would have caused confusion. So how did the periodic table develop over time? Mendeleev's periodic table looked like this. Now, some of the problems he had and how he solved them was that some elements were in the wrong order if they were arranged by atomic weight. So what Mendeleev did was he changed the order to fit patterns in properties. So for example, if sodium and magnesium, and in this case sodium and magnesium would be in this order, there's no examples on this particular one, but let's say sodium and magnesium were in the wrong order, he would have swapped them around, because he would have had the understanding that sodium reacts similar to lithium and reacts similar to potassium. So he made some swaps. So the first thing he did was he made some swaps. The second thing, some elements didn't fit in any group. So he understood that there was elements that were undiscovered. So he left gaps for those elements that hadn't been discovered yet. And thirdly, some elements seem to be missing. So same thing, he predicted the properties of the missing elements, and then they were discovered later. So because he made predictions on their properties, he was able to fit them in to those gaps and say that once something is found, it will have similar properties to this. And he was really, really accurate. Now, Mendeleev's periodic table was accepted by scientists. Why? Because elements he predicted were later discovered. So all the predictions he made, even the predictions made in terms of properties, were very close. Scientists learnt about isotopes. So the understanding of isotopes came later, which proved why he was right to change the order of some elements based on atomic weights. Because like I just said to you guys, when you've got isotopes, one isotope's heavier, one's lighter, one will be one place in, or will have one place in the order, and the other isotope will have a different place in the order. So he did the right thing. He swapped things around based on their properties. Now today, the periodic table is arranged by atomic number. Now, when you get questions where you're doing comparatives, that's one of the big comparatives that you've got to remember. We do it based on protons. We do it based on number of protons. So how did Mendeleev, so this is an exam style question now, how did Mendeleev know that there must be undiscovered elements, and how did he take this into account? How did he take this into account when he designed his periodic table? So Mendeleev noticed gaps in his periodic table where elements with similar properties should be but were missing. He predicted the properties of these missing elements based on their position in the table and left spaces for them. So that's something that needs to be memorized. That's a very common question. Now, by the early 20th century, protons and electrons had been discovered. Describe how the discovery allowed chemists to place elements in their correct order and correct group. So the discovery of protons allowed chemists to arrange elements by atomic number. So as soon as protons were discovered, some scientists started to look at the periodic table again, and they realized that order made more sense, and it was more in line with what Mendeleev had predicted. The electronic configuration, because now they'd also discovered electrons, helped place elements in the correct group as well. Because, for example, if something's got one electron in the outer shell, like lithium, sodium, potassium, they all react in a similar manner. And the fact that reacting in a similar manner shows that they should be in the same group. And this system led to a more organized and accurate periodic table. Hence, we now have a modern periodic table. Metals and non-metals. So, what are the differences between metals and non-metals? Metals are a substance that forms positive ions. Now that's something we're going to cover in more detail when we get to bonding. And they are found on the left bottom part of the periodic table. Non-metals, on the other hand, form negative ions. So what do we call, and actually I'm going to let people have a go answering this, what do we call a positive ion, and what's the word for a negative ion? If you want to quickly type it in, I'll give you guys some time. I can't give you guys this much time. So come on, quick. If anyone knows the answer, what's the scientific word for positive ion, and what is it for negative ion? Unless you've answered and there's a bit of a delay coming across. There must be a delay. It's fine. So it is a cation. A cation is positive. And how do we know cations are positive? Because cats have pores. So because cats have pores, a cation is positive. And an anion, now if we cut off the word, we cut off the A and put an O there, it spells onion. What do onions do? They make you cry. So that means they're negative. So an anion is the negative ion. Well, this will get covered again. So cations are positive, anions are negative. Cats have pores. An anion sounds like onion makes you cry, that's negative. We'll cover this again when we do electrolysis. Group 0. What are the noble gases and how do their boiling points change? So the elements in group 0 of the periodic table are known as noble gases. As you move down the group, the boiling points of these gases get higher. This is because the molecules become larger, which makes the forces between them stronger. As a result, more energy is needed to break these forces. What they mean by this is they have very, very low boiling points. We're talking minus 100, minus 200. But helium has a very, very low boiling point, whereas as you get to the lower ones here, this is getting closer to 0. It's still negatives. Xenon is still in the negatives, but it's closer to 0. And that's because if you consider helium is only one electron shell, whereas 1, 2, 3, 4, 5, xenon is 5 electron shells. Actually, I'm going to do argon because it's easier to draw 3 shells than 5. Okay, now that's a larger atom. Now, noble gases, because they've got full outer electron shells, they don't form covalent bonds. They don't bond with anything. Now, if you've got 2 argons, you're going to form a weak intermolecular bond in between. And if you've got 2 heliums, you're going to form a weak intermolecular bond in between. But this weak intermolecular bond here between the argons is stronger. So that requires more heat energy to break. This one requires less heat energy to break. And that's essentially what this slide is saying. Now, why are noble gases unreactive? So helium is the exception because it's only got 2 electrons, but every other noble gas has got a full out. So helium's got a full outer electron shell, but thinking back to yesterday, the first shell can only take 2 electrons. So helium has a full outer electron shell. Now every single, and we're going to cover this in more detail when we do bonding next, every single element wants a full outer electron shell. Now to do that, it will either lose electrons, and then when it loses electrons it becomes positive, because when we lose a negativity we become positive, and if it has got, or it can gain electrons. So if it gains electrons it becomes negative. Now with helium and everything else in group 0, they've already got full outer electron shells. So they do not need to gain or lose electrons because they're already there. They already have a full outer electron shell. So noble gases are very unreactive and they rarely form molecules because the atoms have stable electron configurations. For a long time, we didn't even know they existed because they're so unreactive. So noble gases already have a full outer electron shell, which means they are very stable and unreactive. Group 1. So why are noble gases unreactive? So group 1 elements in the periodic table are called alkali metals. Now, that's because they have unique properties due to the single electron in the outer shell. Now these are not the correct atoms. So we've got lithium, which has got 1 electron in its outer shell. Then we've got sodium, that's in period 3, so it's got 3 shells, and again 1 electron in the outer shell. Potassium, which is in the 4th period, so it's got 1, 2, 3, 4 shells, but it's got 1 electron in the outer shell because they're all from group 1. Now, when anything from group 1 reacts with water, so for example lithium reacts with water, I'm not going to do a balanced equation right just yet, although we can. It makes lithium hydroxide plus hydrogen. Now lithium hydroxide is an alkali. That's why we call them alkali metals. So they have unique properties, and they have unique properties because they only have 1 electron in the outer shell. So what all of group 1 does, so let's just do it over here. Lithium has got 1 electron in the outer shell. It will lose that 1 electron. Sodium will lose that 1 electron. Potassium will lose that 1 electron, meaning they all react in a specific way. So when they react with oxygen, lithium plus oxygen makes lithium oxide. So what does sodium and oxygen make? Sodium oxide. What does potassium and oxygen make? Potassium oxide. Now here we've got 4 lithium plus 2 oxygens makes 2 Li2O. So if I said sodium and oxygen, then it would be 4 Na plus O2. It would be the same state symbols, so S for solid, G for gas, S for solid, and it makes 2. Now instead of being Li2, I'm going to write Na2O. Now if I did it for potassium, it would be exactly the same thing. All I'm doing is replacing the Na with a K. So they all react in exactly the same way because they're all losing 1 electron. Now how do alkali metals react with oxygen? Apologies, same issue on this slide. Okay, so that's what we've already covered. How do alkali metals react with chlorine? So lithium plus chlorine makes lithium chloride. So that should be a small 2. Now it's the same thing as I said before. We've got lithium, sodium, potassium. Now they might ask you about rubidium, they might ask you about cesium. Doesn't matter which one it is. It doesn't matter which element it is from group 1, but all you're doing is, let's write the whole thing out. 2 plus Cl2, arrow 2, leave a gap, Cl. Now if it's sodium, all I'm doing is swapping the Li with sodium. So I'm just putting an Na there, and an Na there. Same formula. Same thing. Now if I said, instead of chlorine, I said fluorine, then you just swap the Cl2 with fluorine, with F2. So it's the same thing. So now if I did potassium, it would be 2 potassium plus Cl2 becomes 2 KCl. It's exactly the same. We're just swapping the group 1 metal. So how do alkali metals react with water? So lithium's got 1 electron in the outer shell. Sodium has 2 shells and it's got 1 electron in the outer shell as well. And lithium's got 2 shells. Sodium's got 3 shells. Potassium's got 4 shells. I've left myself enough space there. So group 1 elements in the periodic table, now when they react with water, they always make a hydroxide and hydrogen. This is why we call them alkali metals. So 2 lithium, now that 2 there should be small, and that 2 there should be small as well. So it is, if we look at it with sodium, it'd be 2 Na plus 2 H2O becomes 2 NaOH plus hydrogen. Now, quick question. While I go through the next example, if anyone can answer this, how do we test for hydrogen gas? So you can put your answer straight into the live chat if you know it. So for potassium, it would be 2K, exactly same thing, plus 2H2O becomes 2KOH plus hydrogen H2. Excellent, thank you Coding G. Squeaky pop test. And thank you, Farah. Thank you, Night Shadow. There was a question on here that was linked to displacement. I will have to wait till we get to that point to go through it. But if we don't have any extra questions to answer between 9:45 and 10 a.m., if you want to log on early and ask any questions like that, I can go through it at that point. Even if it's paper 2 content, between 9:45 and 10 a.m, that's question and answer time. So if we don't have any questions and answers linked to what we're covering, I'll either be going through 6-mark questions, because a few people have asked for 6-mark questions, or I can go through whatever questions you want to go through. Just one thing I just want to quickly reiterate is that this, because we're covering every single specification point, we are covering combined foundation, combined higher and triple award. But every time we come across any content that's combined higher, I'll let you know. So those people that are doing foundation, as soon as I say this is higher content, then you know that it's not something that will come in your exam. As soon as I say separate science, then the combined students know that it's not going to come up in your exam. Okay, so how does alkaline metal reactivity change down the group? It's more reactive down the group. As you go down group 1, the metals become more reactive. Why? Because the atoms get bigger. So if you think about lithium, it's got 2 shells. Sodium, 3 shells. Potassium, it's got 4 shells. So the atoms getting bigger as you go down the group. So that means the outermost electrons are further from the nucleus. And what's the charge of the nucleus? It's positive. And then we have the electron on the outer shell. So there's an attraction between the two. But as the distance between that attraction becomes bigger, what happens? It becomes easier to take that electron away. It becomes easier to lose that electron. So if this is lithium, sodium, and potassium is the one that is easiest to take the electron from, because that electron does not feel the attraction of that positive nucleus as much. So the outer electrons are easier to lose. So let me just reiterate this, because this is usually a 3-mark question. As you go down the group, the atoms become bigger. So that means the outermost electrons are further from the nucleus. The nucleus is positive, so you have an attraction between the outermost electron and that positive nucleus. The further the distance, the further the gap between the two, the easier it is, as in this, because of there's less attraction between the nucleus and the electron as you get further down the group. As the atom gets bigger, that means the electron becomes easier to lose. So, group 7. The elements in group 7 of the periodic table are known as halogens. So by halogens we mean fluorine, chlorine, bromine, iodine, astatine. The main 4 that you'll be dealing with is fluorine, chlorine, bromine and iodine, but astatine does come up. Halogens are non-metals and made up of molecules that consist of pairs of atoms. So they are diatomic. What that means is you won't find F by itself, you'll find F2, Cl2, Br2, I2. And what you've got in between them is a covalent bond. So both fluorines, both chlorines, both bromines, both iodines are sharing a pair of electrons. Each one is sharing 1 electron each. And we'll cover that in more detail when we do covalent bonding. Now, if you look at the colours of fluorine, chlorine, bromine and iodine, fluorine is a pale yellow gas. So the 2 things to look at here, it's look at the colour and look at its state. So it's yellow and it's a gas. Chlorine is green and it's a gas. Bromine is orange and it's a liquid. And iodine is a grey solid that then sublimes. So as soon as you heat it up, it turns into a purple gas. Now, if I was to say to you, what happens? Can you tell me what about astatine? What do you think the color would be? So it's gone yellow, green, orange, grey. So it would be fair to assume that it's black or dark grey. Because what's happened to the colour as you go down the group, it gets darker. And it's gone from gas to liquid to solid. So we would assume that astatine is also a solid. So these are the trends in colour. This is a trend in state down group 7. How do the properties of halogens change as you go down group 7? So they become less reactive when you go down the group. As you go down group 7, the halogens become less reactive. Why? Because the atoms get bigger. And that's because the outermost electrons are further from the nucleus. So if you see here, that's where your positive charge is. And if you have an incoming electron, because if you look at group 7 here, it's got 7 outer electrons, it wants to have 8. That fluorine wants 1 extra electron. And the chlorine wants 1 extra electron. But which one's going to attract it to its nucleus? Now, the outermost electrons are further from the nucleus as you go down the group. So now you've got a bigger distance here to that electron compared to that fluorine to that electron. So it becomes harder to gain electrons in the outermost shell. So that electron is that electron that fluorine wants to take on, that the fluorine wants to gain, is closer to the positive pull of that nucleus. Chlorine has got 1 extra shell, which means it won't attract it as strongly. And this comes into displacement. So if you, for example, have fluorine and let's just use sodium chloride as an example, which one is more reactive? The fluorine or the sodium? In this case, the fluorine is more reactive. So what the fluorine does is it displaces the I've rubbed out the wrong thing. It displaces the chlorine and it takes its place. So what you're left with is sodium fluoride and chlorine. Now this way around, this chlorine is not as reactive as the fluorine, so the chlorine can't displace the fluorine. So how do the properties of halogens change as you go down group 7? So melting point and boiling points go up as you move down the group. So the boiling point, melting point increases as you go down the group. The halogens get darker as you go down the group, and the halogens react in similar ways as you go down the group because they all have 7 electrons in the outer shell. So because they've all got 7 electrons in the outer shell, each one is trying to gain an extra electron. Yes. So for just a quick answer to your question, if it's reactive, the more reactive it is, that's the one that displaces the less reactive one. So it's a little bit like, not a nice example, but if one person's got a pen and the other person likes it and says, I want that pen, if that person's stronger, they can snatch the pen off them. If they're not stronger, then they can't snatch that pen off them. And that was the same in that example. If you've got fluorine and you've got sodium chloride, that sodium is the equivalent of the pen that the two boys or two students are fighting over. And because that fluorine is more reactive, it takes that sodium for itself. So you end up with sodium fluoride and chlorine. Okay, so this is just a repetition of the previous slide. So, how do halogens react with hydrogen and metals? So halogens can react with hydrogen. For example, chlorine reacts with hydrogen to make hydrogen chloride, which is an acid. So again, when we cover the topic of acid afterwards, it's an acid because HCl, when it enters a solution, it dissociates and it breaks them to H plus and Cl minus. It's the H plus part that we call an acid that causes the acidity. So now if here we've got Cl2 plus H2 becomes HCl, and there should be a 2 here to balance it. Now if instead we react it with fluorine, then it would be F2 plus H2 becomes 2HF, and these would be the same, the big gases. Now if I wrote bromine, what would be different? Because it would Br2. So if anyone wants to write down the answer, they can. I realize when I'm asking you guys questions, there's like a 10-20 second delay between when I say it and you guys hear it. So I'm gonna have to wait a little bit longer to get the answer. So Br2 plus H2 becomes 2HBr, but there's a difference between this one and this one. What's the difference? I'll start filling in the rest of it slowly, and you guys can tell me what I need to put next to that Br2 that's different. Yes, thank you, Coding G. It is the state symbol. That's correct, it's the state symbol, yeah. It is a liquid. So that's the only thing that's going to be different. The overall layout of the equation is going to be exactly the same, but that state symbol, there's the difference. Now, halogens also react with metals. So let me erase this first. So chlorine reacts with lithium to make 2 lithium chloride. Now, if it was fluorine, it's going to be exactly the same. So fluorine plus 2 lithium becomes 2 LiF. If it's with bromine, it'd be exactly the same. The only difference with bromine would be the state symbol. Iodine, exactly the same, but the only difference would be the state symbol. So here's an exam style question. Explain why potassium is more reactive than lithium. So this is exactly what we've covered already. It's just putting it into the answer. And you should answer in terms of electrons. So here's what I would call an ideal answer. So potassium is more reactive than lithium. Point out the obvious. Because it has one more, so I've put a tick there, but that wouldn't get you a mark because that's already in the question. It has one more electron in its outer shell. Now it doesn't have one more electron in its outer shell. It has one more electron shell, that should say. So it has one more electron shell. This electron is further from the nucleus. So it's easier for the potassium to lose it. The easier it is to lose an electron, the more reactive the element is. So that part just there, if you've already been sent these notes, that part needs to be deleted. It's because it has one more electron shell. That means the electron is further from the nucleus, so it's easier for potassium to lose it. The easier it is to lose an electron, the more reactive the element is. The halogens are in group 7 of the periodic table. Explain the trend or in reactivity of the halogens. So the reactivity of halogens decreases down group 7. Okay. At the top, halogens like fluorine are highly reactive because the outer electrons are closer to the nucleus. So the outer electrons closer to the nucleus, making it easier to gain an electron. As you go down the group, the outer electrons are further from the nucleus, making it harder to gain an electron. And that means the reactivity decreases as you go down the group. So, how does a more reactive halogen displace a less reactive one? So, a more reactive halogen can replace a less reactive halogen in its salt solution. For example, chlorine and potassium bromide become potassium chloride and bromine. So it's exactly what we were showing before. Chlorine, so if we look at the order of group 7, it's fluorine, then chlorine, bromine, iodine. And fluorine is the most reactive and iodine is the least reactive. So chlorine comes before bromine, so chlorine is more reactive. The chlorine displaces the bromine. So it becomes potassium bromide plus potassium chloride plus bromine. So here you can see as well, the Cl has swapped places with the Br. So it's become 2KCl plus Br2. Chlorine is more reactive than bromine, so chlorine has replaced bromine in this reaction. Bromine is a red-brown color. So when it's displaced, you will see a red-brown color change. Now that's something that's important. If you think back to the beginning part of group 7, we spoke about the different colors. And we spoke about how the colors get darker. We've gone from light yellow to light green to reddish brown to a purplish to a grey/purple color. So when something gets displaced, the color of the solution changes based on what's been displaced. So now you've got bromine in that solution, which means that everything will change to a bromine color. That chloride is reacting with potassium, so that color won't express itself. The thing that will express itself is the element. So that's something to keep in mind, because what some people do is these kind of questions come up, and when it says what color changes occurred, they look back through the notes or they look back through their teaching and think, no one's ever told me about a color change. But it all links back to the original colors of the elements. Okay, now the remainder of this PowerPoint, that means the next 3 slides are separate science chemistry only. So if there is anyone who's only doing combined, if you want to start your break now, you can do. We're going to restart physics at 12 o'clock. So I'm going to go through this for the next 3-4 minutes, and then we're going to take a break and then restart at 12. So if you're only doing combined, you can start your break now, but please make sure you're back at 12 because we're going to start at 12 o'clock promptly. So, separate science. Where are the transition metals located in the periodic table? This, if you look at the specification online, this isn't that complicated additional topic. What I'm about to cover is essentially what you need to know. The transition metals are a set of metals that have similar characteristics, but they're different from metals in group 1 and group 2. It's the metals that you find in that middle section. The middle section of the periodic table. There. So they're not in a specific group. They're still in periods, but they're not categorized as a group. Usually, you know how with group 1 usually form +1 ions and group 2 form +2 ions. Most metals most transition metals are usually +2. But what you'll find is in an exam question, it'll have Roman numerals. Those Roman numerals tell you what ion it is. So if you've got copper 2 oxide, then you know it's a copper 2+. If you see this Fe followed by 3 lines, then that means it is Fe 3+. If you see Ag 1 something, then you know that is Ag+. Now, how do group 1 metals compare with transition metals? So it's important to know the comparison because you can get some comparative questions. So melting and boiling point is low in a group 1 metal, but for a transition metal it's high. Now this is relative because when we say low melting and boiling points, we're not talking as low as covalently bonded substances. So we're not comparing it to group 6 and group 7 and group 0. We're comparing it to other metals. So group 1 has a low melting and boiling point relative to transition metals. Now group 1 metals are very reactive. Transition metals are less reactive. For example, gold, silver, platinum, iron, copper. You can put these things in water and you won't get much of a reaction. Group 1 metals have low density, again relative to transition metals which have high density. Group 1 metals are silvery and shiny. Transition metals are often coloured. For example, iron has a reddish colour. Pure iron has a reddish colour. Hardness, group 1 metals are soft. Transition metals are hard. Uses, group 1 metals are often used in batteries. Transition metals are often used in construction or jewellery. So if you think of the pipes that we have in our house, the pipes in our house, now most pipes are plastic, but before copper became expensive, most pipes were copper. And that's because copper is quite an unreactive metal. And from an ionic charge perspective, group 1 metals have got 1 electron in the outer shell. When they lose that electron, they've got an extra proton compared to the number of electrons, so they now become a positive ion. Remember, when something, if as a human being, if we lose our negativity, we become positive. Same with atoms. If an atom loses an electron, it's negative. The negative part of the atom, it becomes positive. Now, most transition metals are +2, but part of the reason we call them transition metals is they can transition between different states. They can be +1, +2, +3. Iron can be +1, +2, +3. But it can vary. But usually it's 2+. If they don't give you any information in an exam, so for example, if it says copper sulfate, now SO4 is always 2-. Now if SO4 is 2-, what's copper automatically going to be? 2+, because it has to balance out. Now, what are the typical properties of transition metals? They form coloured compounds. Many transition elements have ions with different charges. Now, when it says coloured compounds, when we find metal oxide in mountains, for example, iron oxide is red. Copper oxide is like a greenish colour. So they form coloured compounds. Many transition elements have ions with different charges, meaning you can have Fe+, you can have Fe2+, you can have Fe3+. And they're useful as catalysts. So if you think of the Haber process and a lot of the processes that come up, particularly in separate science, it's always something like iron, platinum, things like that are used as catalysts. And they conduct electricity, as all metals do. Okay, brilliant. I'm so sorry. Because it's 57, we are going to restart at 2 minutes past. So you got 5-minute break, and then we'll restart at 2 minutes past 12. Okay, thank you everyone. See you all in 5 minutes. Okay, welcome back everyone. Let's get started with physics. So we're now going to have our 55-minute session on physics, where we're going to finish off topic 1 and then we're going to start electricity. Now, electricity is a fun topic, but I know it's going to be a topic that a lot of questions might be asked about. So just keep asking and I'll keep answering, hopefully. Let's really properly understand electricity. But that's going to start as soon as we finish the energy transfer section. So, how can we test the effectiveness of materials as insulators? So these are the materials that we need. So number 1, we boil water using a kettle, keeping the amount of water the same each time. So that's one of our controls. It doesn't have to be 200 centimeters cubed. That's just an example given here. And it doesn't have to be a 250 centimeter cubed beaker. Again, it's just an example. You pour the hot water into the container, like a beaker, and add a lid to reduce energy loss. Because automatically what you're going to get is convection currents. So within your container, the water will start to evaporate. And as it evaporates, those particles take some of the energy away with them. Then you place a thermometer in the water to measure the starting temperature. Wait for a fixed amount of time. So you've got your beaker, you've got your thermometer inside, you've got your lid over the top as well, and the thermometer just pokes a hole through it. You know what your starting temperature is. You wait for 5 minutes, then you measure the final temperature of the water. So if you do final temperature, and from that you take away the starting temperature, you know the temperature difference. Then you repeat the experiment with different materials wrapped around the beaker. So you do it with foil, wool, newspaper. Then you compare how well they insulate and reduce energy transfer. So let's have a quick look at this exam style question which is linked to this topic. The data logger records 10 readings every second. The student considered using a temperature probe and a data logger. Explain why it was not necessary to use a temperature probe and data logger for this investigation. And again, this shows why past paper practice is really important, because you've got one category of question that you're going to be able to answer because you've memorized. You'll have one category of question that you'll be able to answer because you've understood. So the first category is because you've understood. The second category is the ones that you've memorized. And the third one is through pure past paper practice. It's because you've done experiments linked to it at school, and those practice past paper questions, what they do is they take out the understanding from your experiments. And it's questions like this, which you're not necessarily going to have memorized from beforehand, but the learning that you take from questions like this, you can then apply to other similar questions. So if we look at these two, the digital thermometer and data logger, because a lot of you won't know the difference, or you might not know the difference. This kind of question is probably the kind of question that always causes difficulties when you're in exams. What is it that's the same when you look at them? Firstly, both of them measure the temperature. So both measure temperature. The second thing is either of, if they're both measuring temperature, is one more accurate than the other? No, they both measure to 1 decimal place. So for that reason, is there a need to have two? Because they do have the same purpose, they measure to the same level of accuracy. So for that reason, the answer is the thermometer, the digital thermometer and the data logger have the same resolution. By resolution, it means they measure to the same decimal points, as in they measure to the same accuracy. And in this experiment, you'll need to measure the start temperature and the end temperature, which means you don't actually need to have two. You could probably just use the digital thermometer. I mean, I can appreciate someone wants to be accurate using a data logger, but you can actually only need the digital thermometer, which is still accurate. So, efficiency. What is efficiency? Efficiency is useful output energy divided by total input energy. So, for example, if you have a light bulb, and through the electrical supply, you provide 1000 joules of energy, or it could be watts. Now, to light up the bulb, it uses up 300 joules of energy, whereas as thermal energy, it uses up 700 joules of that energy. Meaning 300 joules have been used in a useful manner, whereas the heat represents, because we don't want a light bulb to heat things up, we want the light bulb to light up the room. So that heat aspect of it is wasted. So from that 1000 joules of energy, 300 joules were used in a useful manner and 700 joules were wasted. So when you're working out efficiency of a device, you do the useful output divided by the total. So in this case, our useful would be 300 and our total is what we put in from the beginning, divided by 1000. Now you can write this answer either as a decimal, which is 0.3, or you can times it by 100 and you can write it as a percentage. So 0.3 times 100, you've got 30%. Either way is absolutely fine. Now, like I said, it could be 1000 joules, it could be in watts as well. Watts is joules per second. So you can do it in either format. Now, the total power input, so this is a practice question now. The total power input to the solar cell is 3.5 watts. When the efficiency is 0.25, calculate the useful power output of the solar cell. So let's have a quick look at this question. We know that efficiency equals useful divided by the total. Now, if we substitute our numbers in, we know what the total power input is. So that means we can swap our bottom number, our T, with 3.5, and we know the efficiency is 0.25. So we know 0.25 equals useful divided by 3.5. Now, that 3.5 there is dividing on this side. So what will it do on the other side? It will multiply. So that means useful equals 3.5 times 0.25. If you want to use this as a triangle, then the U goes at the top, efficiency and total go at the bottom. So efficiency times total. So what we're looking for is a quarter of 3.5, which is 0.875 watts. Because our total input, because it's total power input, and it was 3.5 watts, our useful has to be in watts as well. Okay, energy resources. So this is, I think, a topic that a lot of people find straightforward. So if you do have any questions, please ask. Otherwise, I'm going to go through 1.3 quite quickly, because a lot of it is just memorization. If there's anywhere where I feel it needs extra clarification or explanation, I will pause. Not pause, so I'll slow down. Otherwise, I'll go through it quite quick. So coal, oil, and natural gas are fossil fuels that have developed over millions of years from the remnants of deceased organisms. So don't forget, organisms, all living things are, on Earth, carbon-based. So what's happened is, once they've died over millions and millions of years, that carbon has reformed into a different format. So they've turned into coal, which is made of carbon and hydrogen, oil made of carbon and hydrogen, and natural gas, which is also carbon and hydrogen. So there's our 4 non-renewables: coal, oil, gas, and here we've got radioactive metals, so nuclear. What are the advantages and disadvantages of these non-renewable energy resources? So coal has a high energy output, oil is easy to transport. Gas, it produces less carbon dioxide than coal and oil. And nuclear has a very high energy output, and it also has no carbon dioxide emissions. Now, disadvantages: coal, oil, and gas all produce carbon dioxide, so they all lead to global warming. Now, coal, additionally, in a lump of coal, predominantly you've got carbon atoms, but in amongst those carbon atoms, you also have sulfur, and the sulfur is minimal, but because of the amount of coal we burn, it becomes significant. But when that sulfur burns, and when we say burn, we mean combustion, which means reacts with oxygen, we make SO2. When that SO2 reacts with the water, it's a longer process, and those doing A-level biology will cover it in more detail. But when that sulfur mixes with the water in the rain, so SO2 plus water, and it becomes H2SO4, and that causes acid rain. And of course, it's non-renewable. So once we've used it, it's not going to come back to us anytime soon. It takes millions of years to produce, so it's not sustainable. Oil produces carbon dioxide. There's a risk of oil spills, and oil spills float on top of the water, which means any animals that land on top of the water can become injured, die, etc. And it's non-renewable. Gas, it does produce less carbon dioxide, which is good, but it still does produce carbon dioxide, so it still emits greenhouse gases. Nuclear, very expensive to build, and there's a risk of radioactive waste, and it's a long decommissioning time. So if anything wrong happens, and Chernobyl's an example of this, it can cause massive radioactivity issues. So Chernobyl everyone had to exit Chernobyl because of what happened there. What are renewable energy resources? So an energy source that won't run out and is naturally replaced. So for us, we consider the sun to be renewable because it'll be around for billions of years. The wind and water. So what uses do we have? So I should say we have for energy resources. So heating, like radiators and boilers, electricity, and transport. We use renewables the same way we use non-renewables. We would use non-renewables for all three, and now we're able to use renewables as well. The main issue with renewables is they're just not as reliable. They're not as reliable as non-renewables. So we've got solar, which uses sunlight to make electricity. We've got wind, which uses wind to spin turbines and generate power. Now, the way these actually work and how they actually generate power is something that we'll cover as part of the motor effect, as part of paper 2. So that's something that we'll be covering in AMALathon 2, which will be in May. Hydroelectric, moving water like rivers, turns turbines, because all of this links to turbines, and it all links to the fact that within these turbines you have magnets and wires, and when a wire cuts across a magnetic field, it produces electricity, it produces current. And that's, again, if anyone understands the motor effect or understands that part of understands electromagnetism, they'll see the connection. Tidal, the rise and fall of tides spins the turbines. Waves, so when we have waves on the sea, that moves machines to generate power. Again, it spins turbines. Geothermal, that's heat from underground, and that turns water into steam. So it works the same way a coal-powered power plant turns water. So the coal is burnt, it turns water into steam. The steam makes the turbine spin, and that turbine spinning generates electricity. That's same. All these do exactly the same thing, but it's just different ways of making it spin. And when it spins, most of the time it's a magnet that's spinning on the inside, and that's then generating electricity within the wires. And biofuel, that's made from plants or waste, and as it ferments, so biofuel, as it ferments underground, so it's lots of waste products that are bioorganic So we're talking about banana peels, etc. Food that would just get thrown in the bin, gets put underground. As microorganisms feed off it, they release methane gas, and then that gas can be used within homes. So there are countries that do use that quite a lot. Okay, so what are the advantages and disadvantages of these renewable energy resources? So biofuel, it can be made from waste. It's carbon neutral if managed properly. Now when we say carbon neutral, what that means is the amount of carbon dioxide it takes when it's growing. So don't forget, during photosynthesis, what does a plant take out of the air? It takes out carbon dioxide. So it's taken carbon out of the air. Now it then fixes that carbon into a glucose molecule. It takes 6 carbon dioxides to make 1 glucose molecule. Now that glucose molecule is part of the plant. When a plant takes up nitrates, so this is our glucose molecule. Each one of these corners is a carbon from carbon dioxide. Now, when that plant takes up nitrates from the soil, it adds the nitrogen part onto that glucose, and now we have an amino acid. If you attach all these amino acids, we have made a protein, which helps the plant grow. That means that carbon is now locked in that tree. It's locked in that plant. If we now burn that plant, or fruit, or vegetable, whatever it is, if we turn it to biofuel, what we're actually at that point releasing is carbon dioxide. But it's the same amount of carbon dioxide that it took in. So if it took in 1 million atoms of carbon dioxide, when it burns, it can only burn the same amount. So in that sense, it's carbon neutral. What it takes in is what it releases back into the atmosphere. Now, the issue with biofuel is it can take up land that should be used for growing crops. And when you burn it, it releases carbon dioxide. Now, in this case, burning it still leads to carbon neutrality. So it's not as big an issue as burning fossil fuels, but it does take up land that should be used for growing crops. And again, we can use this for electricity generation, heating, transport. Then we've got wind. No fuel costs. It doesn't produce carbon dioxide, so it's renewable and clean. But the issues that people have with it is that it's noisy. It spoils the view for some people. So if you imagine someone's living in a nice green hillside type area, and every time they look out the kitchen window, they look out one of the windows, they see this beautiful view. Then all of a sudden, someone comes along and puts all these windmills, wind turbines in front of them. It ruins the view. On top of that, when they spin, they make a lot of noise. We don't realize it because a lot of the time we only see them when we're driving past on the motorway and we see them in the distance. But someone that lives next to it, most people that live next to it complain. And it only works when it's windy. Not only does it work when it's windy, so it doesn't work when the wind level is too low. It also doesn't work when it's too windy. If it spins too fast, the wind turbine won't work. So it's good, but there are issues. Okay, hydroelectric power. It's reliable, no fuel needed. It's quick to start and stop, no carbon dioxide. So brilliant. The issue with hydroelectric, however, is, so you can see here in this picture, you've got water at a higher level, and that water then, as it drops, or as it goes through these floodgates, it turns a turbine. So that means to make such a massive expansive area, such a massive area of water, because lakes don't exist in this manner where you've got one lake just above the other one. So they have to make their own lakes. So it's man-made. You would have a naturally occurring lake, and they would have to make a man-made version next to it to make this work. So that means they're destroying habitats. It's expensive to build, and it only works in hilly and wet areas. So if you don't have a natural hill, then the water's not going to move downwards. And it's that water moving down that's important here. Now, if we think about this, what's actually happening here is the higher up it is, the more potential energy it has. Then as the water goes down, it increases in kinetic energy. That kinetic energy then helps the turbine spin. So it's all about transfer of energy. So geothermal energy, it is reliable, no fuel costs, no carbon dioxide emissions. The issue here is that if you look at the Earth, the outer layer, I'm not talking about gas here, I'm talking about solid outer layer, is the crust. The crust in most places is about 20 kilometers. But there are some areas where it's less. It can be up to 6 kilometers, 7 kilometers, 8 kilometers, a few kilometers. So in those regions where it's less, where you can, if you can dig that space up and you have access to the mantle, or you can go lower down into the Earth. The lower down you go into the crust, the closer you get to the mantle, the hotter it is. Maybe should have done that with a red pen. So the hotter it will be. Now, if you throw water onto that, it will turn to steam, and that steam can be used to turn a turbine. So some countries, for example in Iceland, where you've got lots of semi-stable volcanoes and you've got that kind of atmosphere and environment, you can use things like geothermal energy, and it's very, once it's been made, because it's expensive to initially drill and build, and it only works in volcanic areas. So a country that does have that kind of volcanic area and they invest in something like this, once they've made it, it's really cheap energy and it lasts for a long time. So tidal energy. It's reliable, no fuel costs, no carbon dioxide emissions, but it is expensive to build, it may harm marine life, and it only works in tidal areas. Yeah, everything I'm going through is for higher and foundation, and it's for separate science, unless I specifically say. So wave energy, no fuel needed, no carbon dioxide emissions, and it is renewable. Unfortunately though, it can be unreliable because it depends on wave strength. It may affect marine ecosystems, and it's expensive to build and maintain. Now, some energy resources are more reliable than others because they produce energy all the time, no matter the weather or time of the day. So unfortunately, that means fossil fuels like coal and gas and nuclear energy, they're the ones that are reliable. They are the ones that generate electricity whenever needed. The issue with those are that there's lots of negatives. Because the issue, and if you look at solar and wind, they're less reliable because they only work when it's sunny or windy. And although solar panels are getting better and better and they can work on a cloudy day, they don't work at night time. Okay, now I'm just going to load up the next PowerPoint, the next presentation, and we'll move on to electricity. Now, I just want to say a really big thank you for everyone that joined yesterday as well, because when we were doing the live session, I think we had, similar today, around 30, 40 students, but afterwards, by this morning, we had over 900 views. So I really appreciate everyone that took the time to like, to subscribe, to forward on, and to share with others. So yeah, we really appreciate that. And like I said, yesterday when I spoke to you, I think we had 7,000 subscribers, and this morning we had 12,000. So again, anyone that's been forwarding and sharing with others, I really appreciate it. Like I said to you guys yesterday, for those that weren't here, we have got long-term plans, and the thing that's going to be coming out soon, we're doing AMALathon 1, AMALathon 2. For those of you that are in year 10 and joined and you're in year 10, from September we'll be starting the weekly AMAL. The weekly AMAL is where we'll be doing we'll be covering paper 1 and paper 2 biology, chemistry and physics on a weekly basis. We'll have a maths teacher that will cover grade 5, grade 7 and grade 9, and we'll have teachers teaching Edexcel and AQA English language, possibly literature as well. Hopefully around the same time we'll be launching our app, and our app is, the way we've designed it, it's been designed to cover all gaps. The issue as a teacher, and I'm not sure if you guys have had the similar concerns or not, but for example, I won't say names. I don't want to degrade a different company. Companies are excellent. There's loads of different fantastic apps and services available. But the issue I've always found as a teacher is that a lot of them give you the question, and then if you get it wrong, they tell you the answer. But what they don't do is tell you where you've gone wrong. So this is a big part of our app. This is a big part of why we're making animations right now, is so that as soon as you get a question wrong, you have an animation to tell you where you've gone wrong. And it's specific. So we'll have questions for example, right now we're doing 2.1.1. We'll have questions just for 2.1.1, so you can check your understanding just on this one topic. So if you get a question wrong on 2.1.1, circuit diagram symbols, then we'll give you an animation specific just to this one section. Then on top of that, we've got live online sessions. So if you're still struggling after the animation, we'll have tutors to help out. So we'll have teachers on the other side. We'll have specific sessions on a weekly basis, and depending on your data, the data will tell you which session to attend. And then we'll have mock exams. There's a lot more features that help you kind of develop. It finds the gaps, it closes the gaps, and it allows you to test yourself as well. So there's a few more phases that we're looking at beyond that point as well. And we'll be covering AQA, Edexcel. There's a few other examples that we'll be looking into as well. So I'm sorry if I've just said that to year 11s who can't take benefit from it. But I'm hoping you're still taking benefit from this. And we do really appreciate everyone that's joined us live. And anyone that's listening to this afterwards, there was 900 of you yesterday. We really appreciate that you've taken the time to watch our sessions as well. So now this part here, 2.1.1, I'm not going to give too much time to, because you need to memorize these state symbols. These electrical component symbols, sorry. There's no other way around it. Because you will get questions where you might be asked to complete a circuit. And you will just need to know how it's drawn. Now, one thing to keep in mind is this. If you look at the diode, and you look at the shape of the diode. So just excuse me 1 second. Yeah, if you look at the shape of the diode, it's got that triangle shape in the middle. So that triangle shape is linked to diode. And it's not just the triangle shape, it is the circle that goes around it. Now if you look at an LED, that D stands for diode. And look what it's got. It's got circle, it's got the triangle in it. Now this here, and then if you look at a resistor, it's this rectangle shape. A variable resistor, it still has that rectangle shape. If you look at the thermistor, which is a type of resistor, it still has that rectangle shape. Now here, that R stands for resistor, and again it's got that rectangle shape. So there are things to help you, so make use of that, as in just these little bits of, this little bit of advice. This you do need to know all of it, but I wouldn't, it's only worth a few marks. So I'm not going to spend too much time. We're not going to spend 5-10 minutes trying to memorize them right now. Okay, so what is electrical current? Now, I am going to explain electrical current first. Now, electrical current is how much charge passes a point in a circuit each second. So let me just say that again. So it's how much charge. Now, if you think about if you have a wire, what is inside the wire? It's a metal wire. So you've got positive ions. And we'll learn about this in more detail after when we do metallic bonding. You have in metallic bonding, what actually happens is the outer electron becomes delocalized. It becomes free to move around wherever it wants. So because it's lost that electron, the remaining atom now becomes an ion, a positive ion. And then you've also got these electrons. Now, if this is attached to a battery pack, so I'm just going to draw a rectangle here and just put 6 volts on it. What the voltage does is it gives those electrons a bit of a push. Those electrons already have a charge. Now, depending on how many of those, so let's say the point is here. If I put an ammeter here, what the ammeter will do there is it will measure how many of those electrons have passed that point with a charge per second. So it's the amount, how much charge passes this specific point in the circuit each second. So you can think of it like a flow of water through a pipe. The more water flowing each second, the bigger the current. So let me repeat that. You can think of it like the flow of water through a pipe. The more water flowing each second, the bigger the current. But here, instead of water, what are we comparing it? What are we looking at? Electrons moving through wires. So the more electrons flowing through the wire at a specific point, then automatically it's got a greater electric current. Now, you could look at it as people walking down a hallway. So you're at school, you're walking through the canteen, or you're walking down the hallway. The current is you could compare it to how many people pass a certain spot every second. So the more people passing a certain spot per second, the bigger the current. I hope that clarifies what current is a little bit, because it is one of those topics that's poorly understood. I'm just going to erase this, and then we're going to go through this formula. So charge flow, or charge, we don't actually need the word flow, equals current times time. So Q equals IT. Now Q represents charge, which is measured in coulombs. Now, like I said to you guys yesterday, you're going to be given all the formulas from beforehand. You're going to be given a formula sheet. So you will already have Q equals IT. What you need to make sure you know is the units. So I stands for current, that's measured in amperes, and T indicates time, and that's measured in seconds. So you need to remember coulombs, amperes, seconds. And electric current is the flow of electrical charge. And hopefully the explanation I just gave puts more context into that. So it's the electrons that are carrying the charge, and it's the amount of electrons passing a point per second. Now this is just a quick question to test our understanding of that formula. A torch worked for 12,000 seconds. So that means we've got time. And before needing to be replaced. The current in the LED was 30 milliamps. So that's I. Calculate the charge flow. Now we know, and I'll just do it at the side here before I show the full answer, Q equals IT. Now do we need to change anything here? Because first thing, we've got to get the units right. And the first thing we've got to change is milliamps. So remember what we said yesterday? Here we're going from milliamps to amps. We're going from smaller to bigger. So what we're going to do? We're going to divide by 1,000. And that's why we get 0.030 amps. The next step, we substitute into the formula. So we know I equals 0.03. We know time is 12,000. Multiply them together, and what's the charge? 360 coulombs. Now in the exam, if you write it as a symbol, as in a capital C, or you write just the word coulombs, both ways you're going to get the mark. But it is probably easier to just write capital C. Calculate the time when charge flow is 5 coulombs and current is 4 milliamps. Now, for those who prefer to use a triangle, Q goes at the top, that's charge, I for current times time. Now this time we're trying to work out the time. So if you cover time, time equals Q divided by I. Now we know Q is 5, and 4 milliamps, we've got to divide by 1000 again. So we know this is 0.004. So it's 5 divided by 0.004. Okay, now what's on here has been done incorrectly. Should be the other way around. So you should have got 5 divided by 0.004, 1250 seconds. So 2.1.3, current, resistance, and potential difference. So the amount of current flowing through a component depends on the component's resistance and the potential difference across it. But I'm going to explain that in a bit more detail. So how much current flows in a circuit or through a component like a bulb. So if we use this example, we've got our circuit. For example, we've got a 6-volt battery pack. We've got our light bulb. Okay, so now current flows in a circuit or through a component like a bulb, and it depends on two things. So here we've got little electrons flowing through this wire carrying a charge. When that charge reaches the light bulb turns on. But how bright that bulb glows or how bright that bulb becomes depends on two things. One, the potential difference. This is the potential difference here. The potential difference is the voltage. That's like the push that makes the electrons move. Yeah, so let me just repeat that. The potential difference is that the push that helps those electrons move. That's why if you've ever done this kind of experiment at school, if I add another cell to this battery, what will happen to the brightness? So if we go from 6 volts to 9 volts, the bulb becomes brighter because now you've got a bigger push. If you've got a bigger push, you've got more electrons going through that bulb, which means that bulb will be brighter. Then the other thing that has an impact is resistance. This is how much the component resists or slows down the current. So what that means is, if we go back to our positive ions, now if you think about your TV at home, or think about your laptop, your phone, anything. Phone is probably the easiest thing to relate to. Now, has anyone ever used the phone for so long that the earwax started to melt? I've come across a few people who spend so much time on the phone, or they're on the phone talking to someone for so long. Actually, your generation probably spend more time on social media, so maybe that's not the case. But I've come across people who've said, I was using the phone, my earwax started melting, which means their phone overheated. But why did their phone overheat? And that's because as the electrons are being pushed through the circuit, and what pushes them through the circuit? Through the wire. It is the potential difference. But as they're going through the circuit, what are they automatically going to collide into? They collide into these positive ions. And when they collide into them, what gets transferred? Energy. So these positive ions now start moving faster. And the longer you have your device running, the hotter, the more energy is transferred to these positive ions, the hotter the device gets. Now, if this has got increased thermal energy, it's going to have an increase in kinetic energy. It's going to move faster and faster, which means it's going to be more difficult for the electrons to get through. That's resistance. So resistance is how much the component resists or slows down the current. So the hotter something becomes, firstly it's become hot because the electrons keep colliding with it. But once it's hot, it means the positive ion is going to move more and more and more. If it's moving more and more and more, it's going to impede those electrons. It's going to stop the electrons from going through. That's resistance. Okay, so I hope that makes sense. Now, if you increase the voltage, you get more current because more push equals more flow. If the resistance increases, then what happens to the current? You get less current. Because if you've got resistance, you're stopping the electrons from going through. So you get less current. Because now you've got more opposition, it's harder for the electrons to move through. It's a little bit like, if I give an example, I think most schools will have some similar kind of situation. When I go into my school, there's a communal area, there's a canteen. And if I come in at school at 7 a.m, the canteen's empty. It's very easy to go from one side of the canteen to the other side of the canteen. But if I go to the canteen at break time, and if you go to the canteen at break time, is it as easy to get from one side to the other side? No, because what's there now? Lots and lots of people, lots and lots of movement. That's resistance. So if you take yourself as the electron trying to get from A to B, initially when there's less resistance, you get there quickly. So that's the electron getting through quickly. But when there's an increase in resistance, or at break time, lunchtime, there's more people, it takes longer to get through. Okay, let's go back to the PowerPoint. Let me just quickly remove these images, and then it'll make it easier to quickly read through what's on the slide. Now, that means voltage, current, and resistance are connected to each other. V equals IR. Now, it says here voltage. What would be a better word to use other than voltage? Voltage is not the word that we should be using. We should be using potential difference. So potential difference equals IR. The symbol for potential difference is V, voltage. Is V, and it's measured in volts. So V is voltage, but better than voltage, we should be writing potential difference. I is current and R is resistance. So I is current, which is measured in amperes, and resistance is measured in ohms. So let's have a quick look at some example questions. A filament lamp has a current of 600 milliamps. So let's quickly convert that quickly. So milliamps, we're going from a smaller unit to a larger unit. So we're going to divide by 1000. So we divided by 1000, we've got 0.6 amps. We know what the resistance is. So we've got current, we've got 3 ohms, which is the resistance, and we're trying to work out the potential difference. So we know that V equals IR. What do we do next? We substitute our numbers in. So one thing, just to explain why I always take out the numbers first, it's the easiest way to answer maths questions when you're doing physics. Always on one side, write down all the information that you have. If you know what information you've got, it's easy to find the correct formula that goes with it. Always write down your key information. Same thing with chemistry, same thing with physics. You can do the same thing with biology. It just brings a lot of clarity to everything. So we know V equals IR. So we're going to do 0.6 times 3, which equals 1.8 volts. Let's move on to the next part. Another example question. So in this question we have got current. So we know I equals 26 amps. We know potential difference is 230 volts. Calculate the resistance. Now, for those that like to use a triangle, we're trying to work out the resistance, so let's just cross that out. So resistance equals V divided by I. So in this case it is 230 divided by 26. So our answer is 8.85, and don't forget the units, ohms. So how can we actually investigate the relationship between the length of a wire and its resistance? So you arrange the equipment as shown in the diagram. Now I'm going to explain this in a little bit more detail. So you set up the circuit. The wire is attached to a meter ruler. So here we've got a wire that's connected with crocodile clips. But the easiest way to do this is to get a meter ruler and then put a wire along it. That way you can measure at the same time. You make sure your ammeter is in series. You make sure your voltmeter is in parallel. And then you make sure your wire attached to the meter ruler is part of the circuit. And that way you can move the crocodile clips to different distances, so you can change the distance of the wire, the length of the wire, sorry. Now you connect the first crocodile clip at 0 centimeters and the second clip at your chosen length. So on your ruler you might start, for example, that might be 50 centimeters. You turn on the power and record the current. So you have a table, you record the current and you record the voltage. Let's call them X and Y because we don't know what the numbers are. Turn off the power and now take this crocodile clip and move it closer. So now you might do it at 40 centimeters. Then you do it again, you follow that same process, write down your results and now move the crocodile clip so it's 30 centimeters apart. Write down your results. Now move the crocodile clips even closer. Write down your results. Now, if we've got I and V, what can we record? What can we put in our third column? We can calculate resistance because it's V divided by I. So we can calculate resistance as well. So you record all the values in a table. Now, what do we expect to find from this kind of experiment? What we expect to find is the longer the wire, the greater the resistance. Now, the way you've got to see this is, you know the example I gave you of walking across your cafeteria. Now, if your cafeteria was 100 meters long and there was resistance and it was full, and if your cafeteria was only 20 meters long and it was full, which one would cause you a bigger problem getting across? It would be the longer one. Because it's longer, it's going to take longer for you to go through. There's more resistance, there's more blockages along the way. So if you've got a longer wire, then the electrons have to travel through more material. And that means even more collisions with the ions, the positive ions, which slows them down. So with us, if you're traveling a longer distance through that cafeteria, there's going to be more bumps with people that are in the way, or having to patiently wait for them to move to one side so you can get through. So it slows you down further. Now, if we plot a graph for this, the way we plot the graph is we put resistance on the y-axis and we put the length of the wire, so length in meters, resistance in ohms, we'd put that on the y-axis and then we plot our graph. Now you should get a straight line because resistance is directly proportional to length. Now in this experiment, to keep it fair, you need to make sure you use the same type of wire. You need to make sure that the thickness of the wire stays the same. So you don't use different thickness of wire because the thicker the wire, the less resistance. the temperature needs to roughly stay the same as well. Because what happens when you increase the temperature, if this is your metal wire, what will happen? The positive ions will vibrate faster and they'll stop the electrons, they'll impede the electrons even more. So it increases the resistance. Increase the temperature means increase in resistance as well. Okay. This is the last part we're going to try and get through, which is 2.1.4. So for some resistors, so I've not had a look at the questions for a while. I just want to make sure there's no questions here. So on that point, those points that I've just gone through, it's really important that it's properly understood, the difference between current, voltage and resistance. It just opens up the rest of electricity as a topic. And what a lot of students, they don't understand, what a lot of students don't understand is the clear difference between the three. So if there's anyone that hasn't fully understood, you can ask now, or tomorrow when we log in at 9:45 for the question answer session, you can log in and I can answer your question specifically. But I hope that's really helped. Now, for some resistors, R is constant, while for others, it varies with current. So the current through an ohmic conductor is directly proportional. So when we say something's an ohmic conductor, we mean the potential difference and the current is directly proportional to each other, and it stays directly proportional. And we can see that with that straight line. It means it's following Ohm's law. That this indicates that the resistance stays the same even when the current varies. That when the temperature stays the same, then you don't get an increase in resistance. As soon as you increase the resistance, that line will change. And we'll be able to see that as we go through some of these examples. So if you set up your equipment correctly, and I'm just going to show you the setup for the equipment and then come back to this. So if you set up your equipment like this, so you've got your cells, you've got your component, so your variable resistor, you've got your ammeter, you've got your voltmeter. And let's say you want to check how current and voltage change based on resistance. Then what you can do is you can use your variable resistor to keep changing the resistance. So you might have resistance of 1 ohms, then 2 ohms, 3 ohms and so on. And then you're measuring the current and the voltage. Now, if we go back to this original part, that's how these graphs were made. So what they've done is, they've got the circuit, they've added the variable resistor, they've added the component, which in this case is the filament bulb, they've added the ammeter in series and the voltmeter in parallel. Then as they've changed the resistance, they've recorded the current from the ammeter and the voltage from the voltmeter, the potential difference for voltmeter. Then they've drawn the graph. Now as you can see here, it's curved off at this point here and it's curved off at this point here. And that is because as the potential difference has increased and the current has increased, what have more and more electrons are now colliding with ions, so you have an increase in resistance. As you get an increase in resistance, you get to a point where less and less electrons can go through, so the current stops increasing. You can keep increasing the voltage, you can keep pushing the voltage as much as you want. So it's a little bit like you're in the cafeteria and you're trying to get from A to B. Now, if someone grabs you from behind, grabs your back and pushes you, you might get through quicker. That's someone increasing the voltage. However, at a certain point, you can have as many people as you want pushing you, but if it's blocked in front of you, you're not going to get through. And that's why it curves off. Now, if you look at this, so that's what the IV profile. So this is called an the current potential difference profile. That's what it looks like for the filament bulb. Now, if you look at the diode, on this side here, the current hasn't left zero, meaning it's close to 100% resistance. The resistance is so high that the current doesn't flow in that direction, and it's easy to flow in the other direction. And that these diodes are used to make sure the current only flows in one direction. Okay, now I'm conscious that we've now reached 1 o'clock. I really appreciate everyone's time today. What I'm going to do is from tomorrow, I will go through this all again. So I'm going to start from 2.1.4. Like I said, if anyone's got questions, you can. This will be live. This will be uploaded to YouTube soon, so you can just post your question directly on there. Any questions you put, it doesn't matter which animation is put next to, we get those questions as a compiled list afterwards. So we'll go through those questions. We'll either answer it on the YouTube channel, or I'll answer it in the question answer session. So again, thank you very much, guys. Have a lovely evening. I'll see you all tomorrow. Good afternoon, everyone. Welcome to AMALathon 1 2025 Day 2. What we're doing today is we are hopefully going to finish off topic 1, biology. So that's what we're doing for this first hour, and then we're going to make a start on biology topic 2. Now I'm going to do my best to cover some of the triple content as well. Otherwise, the triple content session is as soon as the first 3 sessions finish. So we'll have 1:30 till 2:30 is biology. What we're doing today 2:30 till 3:30 is chemistry. 3:30 till 4:30 will be physics. That's what we're doing for this. Then we'll have a 15-minute slot on which was set aside for questions and answers. Topic 2. And I'll keep that 15 minutes just in case there's questions I don't answer today. I'll do my best to answer questions live. Otherwise, we'll have a 5-10 minute break and then we'll start the triple content. There isn't too much triple content to cover today. So anyone that's seen the timetable, for those that do triple award, it's just from topic 2, topic 1, a little bit from topic 1, a little bit from topic 2, and hopefully a small lot from topic 3 that we're going to be covering. And I'll keep that 15 minutes just in case there's questions. So let's make a start. I'll do my best. Any questions you can ask them in the live chat. Otherwise, we'll have a 5-10 minute break. We're looking today at the core practical. There isn't too much triple content to investigate the effect of pH on enzyme activity. This is a core practical, so you need to know this inside out and try some past questions into this. And as I mentioned yesterday, I obviously can't go through the practical, so it's worth watching the videos online. The best ones are the ones made by Pearson and Edexcel live chat. Otherwise, we'll have a 5-10 minute break. We're looking today at the core practical. There isn't too much triple content to investigate the effect of pH on enzyme activity. This is a core practical, so you need to know this inside out and try some past questions into this. Okay, as I mentioned yesterday, I obviously can't go through the practical, so it's worth watching the videos online. The best ones are the ones made by Pearson. So I think there's an issue with the mic. We're just going to check what's wrong with it. So we're looking today at the core practical. There isn't too much triple content to investigate the effect of pH on enzyme activity. This is a core practical, so you need to know this inside out and try some past questions into this. Okay, as I mentioned yesterday, I obviously can't go through the practical, so it's worth watching the videos online. The best ones are the ones made by. Thank you for those that have told us straight away. I think there's an issue with the mic. We're just going to check what's wrong with it. So we're looking today at the core practical. There isn't too much triple content to investigate the effect of pH on enzyme activity. This is a core practical, so you need to know this inside out and try some past questions into this. Okay, as I mentioned yesterday, I obviously can't go through the practical, so it's worth watching the videos online. The best ones are the ones made by. Thank you for those that have told us straight away. I think there's an issue with the mic. We're just going to check what's wrong with it. So we're looking today at the core practical. There isn't too much triple content to investigate the effect of pH on enzyme activity. Okay, is that clearer now? Can everyone hear me? Is that clearer, or is it still repeating? Brilliant. Thank you, Sarah, for letting me know. Okay, so let's continue. So this is a core practical. Thank you, Hafsa. Thank you, Saif, Muqaddas, Zoya. So the core practicals, the best place to find the core practicals are, the best place is to use Edexcel's own YouTube channel. Well, I'm not sure it's the YouTube channel, but it's made by Pearson's. So if you Google the name of the core practicals, so even if you write 1.10 core practical, it should take you straight to one of their videos, and they're really well presented, and it goes through all the different aspects. So I'll still go through the theory now, so it'll help. And when we have the AMALathon 2, when we have the pre-exam workshops, at that point we'll also go through some past paper practice linked to this. So, one step at a time. Step 1, how can we investigate the effect of pH on enzyme activity? So yesterday we looked at how enzymes work, And what we're doing today is we're going to go through this whole practical one step at a time. So step one, add a few drops of iodine solution to each dimple in a spotting tile. So we've added our iodine here. And that's your dimple tile. Or sorry, that's your spotting tile. The dimple is the actual groove within the plastic spotting tile. Now, you add a fixed volume of starch, amylase, and pH buffer to a water bath at a set temperature and leave for 5 minutes. Now, the reason you do that is to make sure that all the solutions we're using all have the same starting point temperature. So one isn't providing, so because if you increase the temperature, it increases the rate of reaction. If you increase the temperature too much, it can denature the enzyme. Now, one thing to note here is amylase breaks down starch into glucose. Now, if you add starch to iodine, it turns it blue-black. When that starch gets broken down by the amylase, then it gets broken down into glucose. That glucose has no effect on the iodine. It stays that orangey colour. So step 3, mix the starch solution and amylase solution together. Now, I've just said this, but I want to recap it. What happens, what does amylase do to starch? It turns it into glucose. Now, every 30 seconds, you take a pipette to take a small sample from the test tube. So what we're saying here is, we've got a test tube. Inside that test tube, we've put starch solution, and we've also put amylase. So now what's starting to happen in there is that the amylase is starting to break down the starch into glucose. Now that's not going to happen straight away. So initially, our mixture there, when we get a pipette and we take a drop out and we add our first drop in here, it's still predominantly going to be starch. So it still makes the iodine go blue-black. And because you're doing it every 30 seconds, after 30 seconds again, it will most likely still be blue-black. After 1.5 minutes, so 90 seconds, it still be blue-black. Eventually, you're going to get a result where it's not blue-black. And then that tells you how long it took for the starch to be broken down into glucose. So then you repeat the experiment, but with different buffer solutions. So that means you still add the starch, you still add the amylase solution, but you also add a buffer. Now the buffer solution, all it does is it doesn't react with anything. Well, it will react with things, but it just changes the pH. So now you're adding a third thing in here, which is the pH buffer. So you do a pH buffer of 3, then you do a pH 5, 7, 9, 11, and you do the same process. You time how long it takes for the starch to be broken down to glucose. And how do we know the starch has been broken down? How do we know the starch has been broken down to glucose? Because when it's starch, it makes this orange iodine turn blue-black. So the starch makes the iodine turn blue-black. However, once the starch has been broken down to glucose, it doesn't change its color. So if I add, if I take some of this solution here, that's in the test tube, if I take some of this and I add a drop of that into my iodine, it will turn blue-black. But once that starch has been turned into glucose, then it won't turn blue-black. So at that point, because you've put it in every 30 seconds, you can work out roughly how long it took. And at each pH, you're going to get a different timing. It's around between pH 7 and 9 that amylase works best. Now, why did we use a water bath? The water bath is used in enzyme experiments to maintain a constant temperature. That ensures fair and reliable results. Because like I said before, if the temperature goes too high, then what's going to happen to the enzyme? It's going to denature. And if the temperature is too low, there won't be enough energy for the reaction to occur. And reactions will still occur, but it'll slow down. The faster particles are moving, they collide more often, and you get more of a reaction. If the temperature goes too high, on the other hand, the reaction completely stops because the enzyme denatures. That means this active site changes shape. So think back to what we said yesterday, the active site changes shape. If the active site has changed shape, the substrate no longer fits. And you don't have a reaction. Okay, 1.11. Demonstrate an understanding of rate calculations for enzyme activity. How can we determine the rate of reaction? Now, whenever we say rate of something, it means a quantity, for example, in centimeters cubed. So either a quantity in centimeters cubed, like a volume, or it means something as a mass in grams or kilograms. So here is the quantity of reactant used divided by time taken, or quantity of product formed divided by time taken. Now, rate of reaction is the speed at which the chemical reaction occurs. So all you're doing is you're taking the volume or the mass, which is in grams, and dividing it by the time taken. So let's have a look at this example. It says calculate the rate of reaction when 234 grams of a substrate is used in 2 minutes. So first thing we're going to do is convert the 2 minutes into seconds. So it took 120 seconds. Now if we look back at our previous formula, it's quantity divided by time. There's our quantity and there's our time, which we've now converted to 120 seconds. So what we can do now is divide those, substitute the numbers in. 234, which is the quantity, divided by time, which is 120. Do the maths. It's 1.95. But here, the quantity was measured in grams and time was measured in seconds, so it's grams per second. Okay, it's just a question online. When is the Biology Edexcel class? This is the Biology Edexcel class, it's just started. Okay, now this is triple award. So I'm going to go past this, and if we've got time at the end of this session, I'll come back to it. However, I'm going to be covering this in the session at 4:30. So when we finish physics, it'll be a triple award session. And like I said yesterday, those that can make the triple award session, that's brilliant. If you can't, it's not a problem. The recording will be there for when you're able to attend. And 1.13b is also a core practical, which is for triple award also. So we're going to move straight on to 1.14. Which again is a topic I'm going to go through with triple at the end of the session. So we're going to continue from 1.15 to 1.17, and then we're going to start topic 2. So 1.15 is about how molecules diffuse, osmose, and actively transport. So diffusion is the spreading out of particles of any substance in solution, or particles of gas, resulting in the net movement from an area of high concentration to an area of lower concentration. Now, whenever you get a question like this, there's always 3 marks available. And the first mark is for saying diffusion is the net movement of particles from an area of high concentration. So your first mark is for saying high concentration. Your second mark is for saying it goes from high concentration to a lower concentration. So the second mark is saying lower concentration. And the third mark is for pointing out that it's a passive process. Now, what do we mean when we say passive process? Passive process means it doesn't require energy. So you've got lots of particles here, that's high concentration. And then they spread out into the emptier space. So they've spread out into the space where there's less of them. So they've gone from high concentration to where there's less of them, low concentration. And it's a passive process, meaning it doesn't need mitochondria. It's no active transport involved. There's no energy needed to make it do that. Now, an example you could look at for that to understand that better is in a classroom usually fits 30 students. Now, if a teacher, instead of 30 students, fits in 200 students, then there's going to be very little space for the students. As soon as the door opens, so if the teacher opens the door, fits in 200 students, then closes the door, everyone's going to be really crushed inside that small space. So the second the teacher opens the door, what's going to happen? The students automatically, they go out through the door because they want to enter a wider space. And that's what's happening with diffusion as well. The particles, when we say high concentration, it means there's loads of them. Then eventually, they go from where there's loads of them to where there's less of them. Human beings would move in the same way. And that's why it's a passive process. It doesn't require any additional energy. So examples of diffusion include oxygen diffuses from the alveoli to blood in the capillaries. So if you have an alveoli and then you have the capillary next to it, there's more oxygen inside the alveoli. And the red blood cells which are passing through, they've got less oxygen because they've been throughout the body. They've been passing the oxygen on to different cells for respiration. So because we've got high concentration in the alveoli and low concentration in the red blood cell, the oxygen diffuses across into the red blood cell through, and it's a passive process. Now, carbon dioxide, there's more carbon dioxide. I'm just going to show it as a little C. There's more carbon dioxide in the plasma, in the capillary. There's less in the alveoli. So we're going to go from a high concentration of carbon dioxide, and it's going to enter the alveoli, where we've got a low concentration of carbon dioxide. And another example is urea. This is a triple example, which I'll go through in a lot more detail when we cover this topic. So urea diffuses out of the blood plasma into the kidneys. So urea is what we get when we break down amino acids. So if we eat lots and lots of protein, any extra protein initially is broken down into urea. And with the urea, it enters the blood plasma and eventually it enters the kidneys to be excreted. Now, there's just a quick question I'm going to answer. How do we make notes while watching? If you join our WhatsApp channel, you'll be able to get more information from the WhatsApp channel. They'll be able to pass you on the notes. So if you go to the WhatsApp channel, you'll get more information there. And then someone from the admin team will contact you. So, osmosis. This is the diffusion of water from a dilute solution to a concentrated solution through a partially permeable membrane. And when we say dilute solution, we mean high water concentration. So a little exactly like diffusion, it goes from high concentration to low, except here we call a high concentration a dilute solution, and we call where there's less water a low water concentration. A low water concentration, we call it. We call a low water concentration a concentrated solution. So if you consider a glass of, an empty glass. If I put at the bottom of this empty glass some Ribena, now how much water at the moment is in that Ribena? Not much. So we call that a concentrate. That's concentrated because you've got loads and loads of that Ribena. Now, if I add water to that, what do we call it when we add water? We're saying we're diluting it. Now we're making those particles spread out amongst the water. Now there's more water than there is Ribena or Vimto. So that's your dilute solution when you've added loads of water. And when you haven't added the water, that's your concentrated solution. And what does it occur through? A partially permeable membrane. Now, you can write this answer as dilute solution to concentrated solution, or you can say high water concentration to low water concentration. Now, if you forget dilute and concentrated, just remember what the capital of America is. It's Washington, DC. So it goes from dilute to concentrated. Now, when you're answering a question linked to this, to get the 3 marks, it's always the same thing that you've got to write down. And people miss out these 3 marks. Particularly those students that are doing higher, they always miss out. Higher students always have a tendency to miss out the obvious. So your first mark comes from saying dilute solution, as in where there's lots of water. So osmosis is the movement of water. To join the WhatsApp channel, there's a link in the posts. And if you join the WhatsApp channel, you'll be able to get all of today and yesterday's notes. For those students who have joined through from Eden, your teachers will also put it onto Teams. So the first mark is saying dilute solution, and second mark, it goes from the movement of water from a dilute solution to a concentrated solution through a partially permeable membrane. That's your third mark. And again, this is a passive process, so it doesn't require respiration, it doesn't require mitochondria. Active transport. This moves substances from a more dilute solution to a more concentrated solution against the concentration gradient. So what this means is, if we look at the image first, now from a diffusion perspective, naturally diffusion goes this way. It doesn't require any energy. But when we're talking about active transport, it goes in the opposite direction. Now, naturally things go from high to low. So to take it in the opposite direction, we need energy. And where does the energy come from? It comes from the mitochondria. So any organelle or any cell that does active transport, it will have lots of mitochondria. Now, what process occurs in the mitochondria? Respiration. And what does respiration release? It releases energy. And that energy is then used to pull that particle across. Now, if we use the example that I gave before of a classroom and a mean teacher that's forcing all these kids into one room. Now, that class is full. It's got 200, 300, 400 students in. Naturally, if the teacher opens the door, which way are the students going to go? They're going to go out through the door. Active transport is where you open the door, but you grab more students and you pull them in. And you're forcibly pulling them in. So in other words, you're exerting energy to get them to come in. Now, if you open the door, a teacher could just take a step back and the students will automatically go out. You don't even need to say anything. They want to enter that open space. But if you ask a student to come into that crowded space, they're not going to come in. So you'd have to pull them in. And that's what's happening here. The active transport, they're using energy and they're taking it in the wrong direction. They're going against the concentration gradient. So here, again, whenever active transport comes up, it's 3 marks. And your first mark is that the substance moves from an area of low concentration to an area of high concentration, and it goes against the concentration gradient. This means it requires energy from respiration. So that would be your 4th mark. It requires energy from respiration. So examples of active transport include root hair cells, because in root hair cells, plants need nitrates from the soil, which is NO3 minus. It's important to know that for chemistry as well. And what happens is there's always more nitrates inside the root hair than there is compared to outside. So naturally, these nitrates are going to diffuse out. So what the plant has to do, using these mitochondria, because it's full of mitochondria, it allows the process of respiration to happen. That energy then is used to bring these nitrates into the root hair cell. So there's always more nitrates inside. Now, why are those nitrates really important? Because when photosynthesis happens, 6 carbon dioxide molecules make a glucose molecule. So each one of these corners is a carbon. So it's taking carbon dioxide from the air and it's trapping that carbon within glucose. Now, this nitrogen from nitrate, if you take that nitrogen, you add it to a glucose molecule, what has been made is an amino acid. Now, if you take those amino acids and join them to each other, eventually, if you have enough amino acids, you have made a protein. And we need proteins for growth and repair. So this is really important that these root hair cells do active transport. Sugar molecules are absorbed from the gut. And we need sugar for respiration. So this happens in our digestive system. Examples of active transport. So this is another example. Sugar molecules absorbed from the lower concentration in the gut into the blood. So our blood at times can have, so it's within the plasma, not the blood cells, at times can have more glucose within the plasma. But we need that glucose for respiration. So our small intestine, which has microvilli, and those microvilli have capillaries, so I've drawn this capillary way too big, has less sugar, so active they can be, so that glucose, that sugar can be actively transported into the plasma. And it's important that happens because we need the sugar for respiration. Every single cell in our body, needs that sugar for respiration. So just previously, I'll just go back a couple of slides here. So in this particular, what it says here, mineral ions are absorbed through plant roots from very dilute solution in the soil. What that means is, if you think back to osmosis, what does dilute mean? It means it's full of water. So this is surrounded by water. Now, you know the example I gave you before, you got the ribene at the bottom. Now, if you've got lots and lots of it outside this root hair cell, then which way are the nitrates going to go? They're going to go straight in. Let me just erase this so it can be seen more clearly. So what I mean by that is, if you had lots and lots of nitrates on the outside, and I'm just drawing it as these purple lines instead of doing loads of dots, then it would go in. But when it's dilute, it means you've added lots and lots of water to it. So now you've got more water and the nitrates are spread throughout that water. So because the nitrates are spread everywhere because they've been diluted, you now end up having a higher concentration of nitrates on the inside. So now your nitrates are spread around because they've been diluted by the water. So this usually happens in, for example, a waterlogged football pitch when it's rained a lot. Sometimes we get muddy puddles. That's not a Peppa Pig reference. You sometimes get muddy puddles, and in those muddy puddles things don't grow as well because the nitrates have become diluted. So at that point you would have to actively transport them in. Okay, 1.16, investigating potatoes. So how can we determine the concentration of a piece of potato? Now, this is a very common question, and it's very common and it's also poorly understood. So actually it's not that poorly understood, it's getting better, but if you understand how to find out the concentration, it can be a really simple topic. So step 1, take your equipment. So that means you take your potato, you take this borer, you put that borer into the potato, and out comes a nice cylinder of potato. Then we weigh it, so now we know its initial mass. We record all this data, so we know initially it was 12 grams, and then because we're going to reweigh it afterwards, then we add it to a test tube full of water, leave it for a set amount of time, and then we take it out, the potato tube, put it in a piece of tissue, and we blot dry and reweigh. Now blot dry means just dabbing it with some tissue. When you dab it with some tissue, what that actually does is it just gets rid of the surface water. What we don't want to happen is where all the water from inside gets squeezed out. So that's why it has to be blotted dry. You have to blot dry it and not squeeze it dry. Now you reweigh. So let's say it weighed 12 grams, but now it weighs 12.5 grams. You know that the change in mass is 0.5 grams. Now you repeat this for different concentrations. So you might do this with 10% sugar or salt solution, 20% sugar, salt solution, 30% and so on. Now that only gives us one reading for each concentration, so you make sure you repeat it twice more. So that gives you 3 readings. Now in your 3 readings, if one of those readings don't make sense, that means it's anomalous. That means you can discard it. So if you've got, for example, 0.5 grams, the second time you did it, it was 0.45 grams. But the third time you did it, it was 3.2 grams. Automatically, what we're going to do with the third one, we're going to discard that data. We're just going to make an average of these two. And step 7, that's not a step for us. Now, so what we're trying to find out here is we're putting this potato, this tube of potato, into different concentrations, so different solution concentrations here in the test tube. We're leaving it for 20 minutes, half an hour, 1 hour, and we're seeing how much water enters or how much water leaves that potato. Now, if water enters, what's going to happen to the size of our potato? It's going to get bigger. If water leaves it, then it's going to get smaller. So we're seeing in which concentration, and what we're seeing, what concentration has what effect. So generally speaking, if it's in pure water, what's going to happen? Water is going to enter inside because it's surrounded by lots and lots of water. There's more water outside than inside, so water enters. If it's put into a solution that's mainly sugar, if you've got lots of sugar on the outside, that means you've got more water on the inside. So now water will come out. So in a sugary solution, you've got more water coming out. So that means the size decreases. Now, what are the potential errors in this experiment? So one is inaccuracy in the balance. The more decimal places the mass pan balance has, the more accurate it is. Sometimes people over dry the cylinders. So there's always an overexcited student that squeezes all the water out of it. Evaporation from tubes. So when you leave these tubes open, depending on the warmth of the room, some of the water vapor, some of the liquid will turn to gas and it will diffuse away. Now, if some of the water leaves that test tube, then the concentration has now changed. So now you've got inaccuracy in the concentration of the solutions. It's important to learn this slide because when these kind of questions come up, students usually struggle when it comes to finding out common errors or potential errors. So how can we take results from this now? How can we actually work out what the concentration of the potato is? So number 1, you calculate the percentage change in mass. So I'm going to cover 1.17 now. I'm not going to cover it in our session. I'm just going to explain it as I'm going through. So the example I gave before, let's use a different example. Initially, a potato weighs 3 grams, and now it's been put into pure water So I'm just going to show this. It's in the cylinder. It's in a measuring tube, sorry, or boiling tube or test tube. And now there's lots more water on the outside. So what happens? The water enters. Now one thing to note here is water's entered, so now it's 3.5 grams. So it's increased by half a gram. So how do we find out percentage change? We take the change. So what's 3.5 take away 3? That's 0.5. So that's our change in mass divided by the original. What's our original? 3. And then you multiply that by 100 to turn it into a percent. Now, let's assume again we had another 3 gram potato, and after it was put into a sugary solution, it was 2.5 grams. Now with a sugary solution, there's more water on the inside than the outside, so the water leaves because of osmosis. Now whenever we say the word osmosis, what should we automatically also add? So if we look at this one here, water entered. If the question says, why has the mass increased? You say, water has entered the potato tube. If water has entered the potato tube, through what process? Osmosis. And what happens in osmosis? Water goes from a high concentration to a low concentration through a partially permeable membrane. That's how you get the full marks. What most people, when they get a question that says, why has it increased in size? Most people just say osmosis and just stop there. So just this example again, 3 grams becomes 2.5 grams. That means our mass has changed by minus 0.5 grams. If we divide that by 3, the original, this time we're going to get a negative percentage. So if the mass increases, you get a positive percentage. If the mass decreases, you get a negative percentage. So it's okay to get a negative percentage because you're going to see in the next slide that you're drawing a graph from this information. So you need the positives and negatives. So you need to calculate the percentage change in mass. Then you plot the percentage change in mass against concentration. Okay, so that looks like this. You got line of best fit. Now, see that point there where the little pink dots come? That is the concentration of the potato. Because when the concentration of the potato was that particular concentration, look what happened to the change in mass. It didn't change. Now, if I've got my potato tube, okay, and it's inside water, if I've got the same amount of water outside as I've got inside, then what's going to happen? Water will exit and water the same rate, the same speed, meaning the mass doesn't change. So that point there is the concentration of the potato. Now, what was the whole point of this core practical? To find out the concentration of the potato. Now, that 1 mark is missed by so many students because they don't learn that one fact. So it's important that you remember that. Now, this is a really common question linked to this. So whenever potato comes up, this question comes up as well. Why does the potato gain in mass, or why does the potato lose mass? So what you've got to be able to say, and I'm going to explain this in my own words. If it gains in mass, what's actually happened? Now, many students miss out the obvious. It's the students that are aiming for grade 9, they're really good at science, and they just make the assumption that everyone already understands what they're talking about. So how does that increase in mass? What has to happen? Water enters. So people miss this fact out. Water enters the potato. Why does the water enter the potato? Because of osmosis. And in osmosis, the water goes from a region that is dilute to a region that is concentrated through a partially permeable membrane. Same thing every single time. Now, here it says, why does the potato lose mass? So this time, it's a little bit different. Which way is the water going? Water leaves the potato. Why does water leave the potato? Because of osmosis. This time, which way is the water going? It's going from, it's still going from dilute to concentrated, except this time dilute is inside the potato. Before it was outside the potatoes. This time is the dilute part is inside the potato, so water exits from dilute to concentrated through a partially permeable membrane. So this is why I was saying to you before, whenever you have this kind of question, you have to remember to include everything as part of your answer. Okay, if you bear with me, I'm just going to quickly put up the next presentation. So we're now on to topic 2. Now, Edexcel Biology, Chemistry and Physics, topic 1 is really big. As in paper 1, is really big. When you get to paper 2, it calms a little bit. Even with paper 2, biology is quite a big topic. But it is not, but paper 1 is much bigger. That's why if you look at our timetable, it's quite kind of crammed together. It's quite congested, and that's why I've added the additional lesson for triple. So triple students are given proper time, and they can go through the theory properly. Okay, so I'm just reading one of the comments. It said the mic's breaking up. Okay, so another person said it's fine. That's okay. Anyone that misses anything, don't forget it's all being recorded, so you can come back to it. Okay, topic 2. So topic 2 is predominantly cells and control. Now if you look at the timetable, we're starting cells and control today, and we'll finish it tomorrow. And then we'll start genetics. So the first part of our understanding that we need to learn about is mitosis. And it's important that you can recognize the pictures and you can recognize the different stages of mitosis, correctly order the stages, and also describe the different stages. So it is really important to memorize this. So if we look here, let's start with genes within itself. So with mitosis, you have a parent cell. And that one parent cell provides splits to become 2 daughter cells. Now, mitosis plays an important role in growth, tissue repair, and asexual reproduction. Now, this is what I was saying, this is important for everyone to know, is that if we take a cell, what we've got here is the nucleus. Now, if you look inside the nucleus, we have chromosomes. Now, if you unravel the chromosomes, it unravels into DNA. Don't need to worry about the word centromere and telomere. It's of benefit for those students who are doing A-level biology, but you don't need to know for GCSE. Now, if we take one section of this DNA, then we call it a gene. And a different version of the same gene we call an allele. So a gene codes for a specific protein, a specific characteristic within the body. And so, for example, we might have genes for eye color. Now, from eye color, we've all got different versions of that gene. So some people have got the gene for blue eyes, some people have got the gene for brown eyes, black eyes, green eyes. and that's what we refer to as an allele. It's a different version of the same gene. And we've got 23 pairs of chromosomes, 23 from the male, 23 from the female. Now the cell cycle's got 3 main phases. Now for Edexcel, it's important that we know all 3 phases and we know exactly what's happening. We know exactly what's happening in each phase. I'm just going to bring the mic a little bit closer so there's no issues. Now, the first phase is interphase, then mitosis, and then cytokinesis. Now, we also need to know all the different steps involved in mitosis. So those people that have already put IPMATC into the live chat, you are correct. And this PMAT, prophase, metaphase, anaphase, and telophase is what makes up mitosis. And interphase, so let's start with interphase. Interphase is when the cell's DNA replicates. Now remember, the whole point of mitosis is to take this original cell and to make 2 genetically identical cells. So the first thing that has to happen is the 46 chromosomes that are there have to all be replicated, duplicated. Because afterwards, when it splits, we want our 2 daughter cells to also have 46 chromosomes each as well. We want it to be genetically identical. Then also what's going to happen is all the subcellular structures, all the organelles will start multiplying too. And overall, our cell will increase in size. And the reason it's increasing in size, is so that it has the additional cytoplasm and the additional space for the structures that eventually will split into 2, because those 2 daughter cells, although it doesn't show in the picture, are exactly the same as this original parent cell. So it's not that 2 smaller cells are made, they're identical in every way. Right, let's go through each step one at a time. So what actually happens during mitosis? We've already said interphase. Prophase is where the chromosomes condense. Now if you look carefully, what's happened from interphase to prophase? The nuclear membrane has broken down. That's why you can't see this white area in prophase. The chromosomes condense. So at this point, if you looked at it through a microscope, you'd be able to see it. Then we've got metaphase. Now, metaphase is when the chromosomes align in the center of the cell. And they're attached to the spindle fibers. Then we've got anaphase. The paired chromosomes are pulled apart by the spindle fibers towards opposite ends of the cell. And then we've got telophase. Now look what's formed again in telophase. A nuclear membrane is formed again. It's just getting ready to actually split and become 2 separate cells. So the chromosomes reach opposite ends of the cell and a nuclear membrane is formed around them. And then finally, what happens in cytokinesis? The cells divide. So, which is the correct order for these stages? These are the kind of questions you've got to be able to answer. Usually only 1 or 2 marks. So the spindle fibers is a type of protein which is made within the cell. The cells have ribosomes, and then those proteins are made from there. But you don't need to know that for the Edexcel specification. But I'm just answering Safe's question. So initially we've got prophase, which is R. Then we've got metaphase, which is Q. Then we've got anaphase and telophase. Now we know it's telophase because you've got 2 separate sections of DNA, chromosomes. State which phase of mitosis this image represents. So you can see how they're being separated apart and they've gone to opposite ends. So this actually represents anaphase because you can see they're still being pulled to the other side. If they were in a random kind of mix like this on opposite sides, then you would have said telophase. And you'll start to see something distinctly like a nucleus. But here, you can't see any membrane around it, which shows that it's anaphase and not telophase. 2.2. Describe the importance of mitosis in growth, repair, and asexual reproduction. So the cell cycle's got three main phases, as we've already said: interphase, mitosis, kinesis, cytokinesis. But why is it important? So it's important because it's important for growth. We need it for repair, and we need it for asexual reproduction. Because what we're doing here is we're taking our original cell and we're making two cells which are identical, which is important throughout the whole of our body. So if we want to replace skin cells, we don't want different versions every single time. When we're replacing our muscle, we don't want different versions. The cells in our eyes, we don't want different versions every single time. We need the cells to be exactly the same. If we damage some skin, we don't need a different type of skin growing across. So if we have a cut, we don't want the new skin to be a different type. So growth is one of them. That's because we produce more cells for the organism to grow. Repair. So if someone's got a cut, it replaces damaged or old cells. And asexual reproduction is something that happens in, for example, bacterial cells. It happens a lot in it happens in single-celled organisms. But essentially, mitosis is a type of asexual reproduction. But some organisms use it to reproduce, such as bacteria. 2.3. Now, in 2.3, we're going to compare the parent cell and the daughter cells produced due to mitosis. Now, in mitosis, now it's important you memorize these key facts because it's a very common question when mitosis is compared to meiosis. Now, mitosis we're covering now, meiosis we will cover tomorrow when we cover genetics. It's part of topic 3, and then we'll do a comparison between the two. So the two daughter cells are genetically identical to the parent cell. They have the same number of chromosomes. So for human beings, the daughter cells have got 46, just like the parent cell. They're used as body cells. So our cells in our body all go through mitosis. The only cells that don't go through mitosis are the sex cells, the gametes. So the egg cells and the sperm cells. And they're all diploid. Diploid means they've got a full number. Now in meiosis makes egg cells and sperm cells, and meiosis forms haploid cells, where it's half the number. And they're not genetically identical. So meiosis is the opposite of everything that we've got for mitosis. So let's quickly complete this table. What's the number of daughter cells produced by mitosis? And the number of daughter cells is 2. And the number of chromosomes in each daughter cell. So now this question here says, human body cells, except gametes, have 23 pairs of chromosomes. So when it says number of chromosomes in each daughter cell, it's asking to talk about 23 pairs. What's 23 pairs? 46. So 23 pairs or 46 chromosomes. So parent cell turns into 2 daughter cells. Okay, next one. Describe cancer as a result of changes that lead to uncontrolled cell division. Now the key learning from this part here is this bit. Cancer is a result of changes in cells that lead to uncontrolled growth and division. That's what gets you the marks. When it says what is cancer, it's uncontrolled growth and division that is caused due to a mutation. And why does it happen? It happens because of a mutation. Now you've got 2 different types of cancer, benign and malignant. Now benign are the ones that don't spread to other parts of the body and usually are not life-threatening. So that's a tumour that's just in one part of the body. That means it can be cut out or it can be dealt with in that one area. The issue is when, because don't forget, a tumor is, if we let's look over here, it's this. That should say malignant. It is this. It is the uncontrolled growth and division of cells. Now, if it's happening in one area, we refer to it as benign. Now, if one of these cells breaks off and it goes through the bloodstream, then wherever it lands, after going through the bloodstream, wherever it lands, it starts multiplying again. So it could end up in the kidneys, the liver, any part of the body, but it starts multiplying again rapidly. And then if a bit of that breaks off, it can end up in another part of the body, meaning a person ends up with multiple tumors. So malignant tumors are what we call cancerous, and they have the ability to spread to distant parts of the body through the bloodstream. So once they've spread, they start to form secondary tumors. And that's where it becomes much more difficult. It becomes much more difficult to control. So if you look here, again, if you look at the page before, in normal cell division, you've gone from 1 to 2 to 4. But in cancerous cell division, we've gone from 1 to 2 to 6 to many. So yeah, so for the exam, you do need to know the difference between a benign tumor and a malignant tumor, but the most common question is this one: what is cancer? But benign and malignant come up too, so it's important to know that also. So benign tumors are non-cancerous. So this is your typical question that could get asked. Benign tumors, as in what's the difference? As a comparative question. So benign tumors are non-cancerous. Yes, it can come up with combined. So when I'm going through the PowerPoints, if something is linked to triple, I'll let you know it's linked to triple. If something is higher only, I will also let you know when it's higher only. So at the moment, everything we're covering is for everyone. Foundation, higher, triple. So benign tumors are non-cancerous. They don't spread to other parts of the body. They grow slowly and stay in one place. Often they have a capsule around them. That you don't need to know. Malignant tumors are cancerous. They can spread, as in metastasize, to other tissues and organs. You don't need to know that word. If you want to learn it, memorize it, that's absolutely fine. You can use it in the test, you'll get a mark, but you don't have to use it. You can just say it spreads to other tissues and organs, and it grows quickly and uncontrollably. It can invade surrounding tissues. Okay, this is going to be the last specification point we're going to cover. Then we're going to take a 5-minute break after that. So what are the examples of growth in animals and plants? So animals, you can get cell division or differentiation. But in plants, you can have cell division, cell elongation, or differentiation as well. Now, what does each one mean? Cell division, we've already covered mitosis. Cell division occurs in both animals and plant cells. We need that for growth, repair, and reproduction. So meiosis is also a type of cell division that occurs. Cell differentiation is when you have a normal stem cell. It has no specific job, but then it specializes and has a specific job, a specific role, specific function. So our bodies also go through our cells also go through cell differentiation. Now in plants, cell elongation occurs as well, and it only happens in plants. And that's during cell elongation, the cells grow and become longer. So literally what you can see in the picture can happen in a plant cell. Okay, thank you everyone. It is now 30:30. We're going to continue at 35. So at 2:35, we'll continue with chemistry. Thank you everyone. See you all in 5 minutes. Okay, welcome back everyone. So firstly, I just want to say a really big thank you for those people that have subscribed and shared, because I think 2 days ago we were on 440 subscribers, and right now we're on just over 11,000 subscribers. So thank you for those that have supported us and have helped us grow. Really, really appreciate it. So really, really appreciate the support. And even yesterday's session, I think when we did the live session, we had maybe 30, 40 students. Not too many, but just overnight, we've had over 1,000 views of yesterday's session. So again, really, really appreciate the support. Really appreciate those people that watched it. Those people that weren't able to watch it live, but still watched it afterwards. And really appreciate those people that have shared and subscribed and liked the videos. Okay, so we're on chemistry now. Ionic bonding. We're going to go through the 3 different types of bonding today. Got a lot to cover now in the next hour. So I'm going to go a little bit faster. But what I want you guys to do is slow me down when I'm going too fast. So if there's something I say that doesn't make sense, or it makes sense but you just want to understand it a bit better, just mention it in the live chat. I've got the live chat open as well, so I know exactly what people are asking and saying, and I can respond to that. So let's get this understood. Or I could say, AMALearning, you're about to. Now, type of bond first that we're looking at is ionic bond. The particles involved are oppositely charged ions. And when a metal joins with a non-metal, you get positive and a negatively charged ion, which is going to come up in the next couple of slides. And we're going to explain that in a bit more detail. So an ionic bond, examples of substances with ionic bonds include sodium chloride and magnesium oxide. All this will start to make sense as we go through charged ions. Now I'm going to explain. Let me go through this first. So let's look at lithium. Now this does come on another slide. So when you're revising this afterwards, if you're using our notes, there are a few amendments that we need to make in the notes at the moment. So it might be a couple of days before we start releasing them out to people. But if you're going through the notes afterwards, everything I'm about to explain is on a separate slide, but I want to explain it here so it links. Now, if I look at lithium, lithium's in group 1. Now, if it's in group 1, automatically what can we say about lithium being group 1? We can say that it has 1 outer electron. It's got 1 electron in its outer shell. So if it's got 1 electron in its outer shell, done. It's got 1 electron in its outer shell. Now, if it loses that 1 electron, so let me just start this again. So lithium looks a bit like this. On your periodic table, it's got 3 protons and it's got a mass number of 7. Now it's got 1 electron in the outer shell. So initially, it has got 3 protons. So that means it's got a charge of 3 plus, and it's got 3 electrons, which means it's got a charge of 3 minus. So what's its overall charge? 0. As atoms should be. All atoms should have an overall charge of 0. Now, once it loses that electron, so let's look at this lithium here. Let's put square brackets around it. It's lost an electron. Now let's compare what it has. Has the proton number changed? Because it's lost an electron. It's not lost a proton. So it's still got 3 protons. So it's got a 3 plus charge. And it did have 3 electrons, but it's now lost 1. So how many electrons does it have now? It has got 2 electrons. So that's a 2 minus charge. So what's 3 plus and 2 minus? It equals not 0, sorry. What does it equal? 3 take away 2. It equals plus 1. So the charge of a metal is plus 1. So lithium has a plus 1 charge. That's why all of group 1 is plus 1. All of group 2, because it's beryllium, magnesium, calcium, they've got 2 electrons in the outside shell. They lose 2 electrons. If they've lost 2 electrons, what's their charge? So always think of it like a human being. If we lose our negativity, then we become positive. So when an atom loses its negativity, its electrons, it becomes positive. So it becomes 2 plus. Now let's look at group 7, fluorine. Fluorine's got 7 electrons in the outer shell. So I'm just going to rub all this out so we can use this space. So now let's look at fluorine. Fluorine looks like this. The top number is 19, the bottom number is 9. So fluorine's got 9 protons and 9 neutrons. Now it's in group 7. So how many electrons does fluorine have? It has got 7 electrons in its outer shell. So what does fluorine want? It wants 1 more electron. So initially it's got 9 protons. So that is 9 plus. It's got 9 electrons. So that is 9 minus. So just like every single atom, it has an overall charge of 0. But if it gains an electron, now it's going to look like this. It's going to have 1, 2, 3, 4, 5, 6, 7 electrons, and it's just gained this extra one. Now if it's gained that extra electron, let's see what its charge is going to be. It's now still got 9 protons, 9 p's. So that's 9 plus charge, but now it's gained 1 electron, so it's got 10 electrons. Just like human beings, if we surround ourselves with negativity, we become negative. So now it's gained that electron, so 9, take away 10, it's going to have a -1 charge. So fluorine will have a -1 charge. Now oxygen is in group 6. So it's got 6 electrons already, and it gains 2 more electrons. So it becomes -2. Now if you understood what I've said, brilliant. It means you can work it out whenever in the middle of an exam. But it is worth just memorizing and keeping in mind that group 1 gives you a +1 ion. Group 2 gives you a +2 ion. Now it says here group 17. That's wrong. It's group 7. And not group 16, group 6. Group 7 gives you a -1 charge, and group 6 gives you a -2 charge. So how do positive ions form? It's exactly what we've just gone through. But one thing to remember is, what do we call positive ions? We call them cations. How do you remember that? Cats have pores. So cations are positive. Cats have pores. So what's happened from lithium atom to this lithium ion? It has lost this electron. Because it's lost the electron, it's become positive. So positive metal ions are created when metals lose electrons. Now, this bit we've already gone through, but it's there for you when you get the revision notes. How do negative ions form? So first thing, an anion sounds a bit like, if we turn the A into an O, it sounds a bit like onion. Onions make us cry, so that is negative. So anions are negative. Cations are positive. So it's something to keep in mind. Now here we've got fluorine. It's already got 1, 2, 3, 4, 5, 6, 7 electrons. It gains 1 more electron. And because metals lose electrons, non-metals gain electrons. So when the non-metal gains the electron, so at the moment you don't need to worry about the top bit, we've just discussed that already. The non-metal gains that electron from the metal, and a negative ion is formed because now it's got more negatives than positives. Now, magnesium chloride is an ionic compound containing magnesium ions Mg2+, which means it's from group 2, and chloride ions Cl-. Figure 10 shows the electronic configuration. So there's the electronic configuration of magnesium and chlorine. Explain how magnesium and chlorine atoms form the ions in magnesium chloride and how the ions are arranged in solid magnesium chloride. So let's go through this one step at a time. we've got magnesium, and it's got 2 electrons in the outer shell. We've got chlorine. Chlorine's in group 7. It's got 1 electron. It's got 7 electrons, sorry, in the outer shell. Magnesium wants to lose both electrons. Chlorine needs to gain 1 electron. So what does magnesium do? It gives its 1 electron here. Chlorine is now happy. Chlorine is happy. But magnesium isn't. Magnesium wants to lose both electrons. So we need to bring another chlorine. This chlorine also has 7 electrons. And this electron is transferred to this chlorine. So now we have magnesium has lost 2 electrons. So what's the charge on magnesium going to be? 2 plus. Now here, magnesium's got 12 electrons altogether. So like it said, magnesium is 282. It said that on the previous slide there. Magnesium is 282. If it loses 2 electrons, then it loses this bit here. So it just becomes 28. Chlorine gains an electron, so it becomes, instead of 7, it's got 1 more, it becomes 288. So here this magnesium is 2,8. And you put a bracket, that's the electronic configuration, you put a bracket around it. So you can put the 2 plus there. Now with chlorine has gained an electron, so it's become minus, and that's become 288. And what do we write for the electronic configuration? 288, put a square bracket around it, and put negative on the outside. And we've got 2 of these chlorines. So I'm just going to draw the second one. Now, in the exam, if you just draw this out, you'll just get the full marks. You're going to see the description in a second, but everything that's in the description is fulfilled by what we've just drawn. Or you can explain it in words. And what in words we would say magnesium has 2 electrons in the outer shell. It gives 1 electron to one of the chlorines, because chlorine has 7 electrons in the outer shell and wants to gain 1 electron. Magnesium's got 2 electrons, so it gives 1 to one chlorine and 1 to the other chlorine. Magnesium has an electronic configuration of 282. When it loses the 2 electrons, its electronic configuration becomes 28, and it now has a 2 plus charge. Chlorine gains an electron, so now it has a negative charge, and it has, because it's gained that 1 electron, it goes from 287 to 288. Okay, now let's look at the mark scheme answer. Now, one thing to keep in mind when we're doing ionic compound and ions comes up again and again and again. So it's something that will come up in electrolysis. It comes up in a lot of different topics. It comes up in redox. So the better we understand this now, the better we're going to do when those topics come. So magnesium chloride is made when magnesium and chlorine atoms form ions. So we've already shown that it formed ions. Magnesium uses 2 electrons to become Mg2+. We put that there, and it becomes a +2 charge. We put that there. Chlorine gains 1 electron. We've put that there to become Cl- with a -1 charge. So our diagram showed all of this. Now we've got Mg2+ and Cl- ions are oppositely charged, so they attract each other, forming an ionic bond. Now we didn't mention this, that when you have oppositely charged ions, then you have an electrostatic attraction between them. Now in solid magnesium chloride, the ions are arranged in a regular pattern with Mg2+ surrounded by chlorine, you don't put the 6 chlorine minus ions, and each chlorine minus surrounded by Mg2+ ions. Okay, so how do we determine the number of protons, neutrons and electrons in an ion? So this is something I've already shown you, but I'm going to go through it with you anyway. So let's look at the example of sodium. And initially when we've got sodium has 1 electron in the outer shell. Now as on your periodic table, sodium looks like this, 23, 11. So that means in terms of protons, it's got 11 protons. In terms of electrons, it has 11 electrons. So we've got 11 positives, 11 negatives. So what's our overall charge? 0. Now, because it's got 1 electron in the outer shell, so sodium looks like this. It's got 3 shells altogether because it's in period 3, but it's got 1 electron in the outer shell. Now that 1 electron, if it loses that 1 electron, now how many protons do we have? Still 11, because it's not lost any protons. So we've got 11 positives. What about electrons? It's just lost an electron. So we've gone from 11 electrons to 10 electrons. So 11 plus and 10 minus. So what's 11 plus? Take away 10 is plus 1. So that means our sodium will have a charge of plus. And that's what this whole slide is trying to explain. So let's get rid of this, and I can show you what's already written there. So sodium atom, it has 11 protons, which are positive, and it's got 11 electrons, which are minus. So now we've got overall charge of 0. Here we've still got 11 positives, and 10 minus is because we lost an electron, so our overall charge is positive. Okay, now for this question, I know we have a slight delay in timing. So if anyone wants to have a go and answer this, I am going to keep moving on. But if you can answer this question in the live chat, then I'll see your answers as you put them down. So we've got Mg2 plus. I'm going to start going through the answer, but if you want to write it down, we can just see who's understood. So magnesium is 282. But it's got 2 electrons in the outer shell, so it loses those 2 electrons. So Mg2 plus is 28. Now O2 minus, oxygen, has an electronic configuration of 2,6, but it has an electronic configuration of 2,6. But what does the oxygen do? It gains 2 electrons. So when it gains those 2 electrons, because it's got 6 electrons in the outer shell, 1, 2, 3, 4, 5, 6. It gains 2 more, so its electronic configuration becomes 2,8. It's now got a full outer electron shell, so 2 minus. So both have an electronic configuration of 2,8. So describe how the following ions form. So lithium ion loses 1 electron because it's in group 1. Calcium ion is from group 2, so that means it loses 2 electrons. Oxide ion is in group 6. In group 6 it gains 2 electrons. And chloride ion, it's in group 7, so it gains 1 electron. Now, when sodium loses 1 electron, it becomes Na+. Here. When the chlorine gains that electron that it lost, it becomes Cl-. The electrostatic attraction between those two ions is what we call an ionic bond. So the metal loses electrons to become positive. The non-metal gains electrons to become a negative ion. And these oppositely charged ions form ionic bonds. So let me just repeat that again, because this is very important. The metal loses electrons to become a positive ion. the non-metal gains electrons to become a negative ion, and the oppositely charged ions, they attract to each other. There's an electrostatic attraction, and that's what we call an ionic bond. That's what forms an ionic bond. Describe what happens when the following metals and non-metals react. So these are lots and lots of different examples that you can go through in your own time. So hopefully, like I said, we've got a couple of things that we just want to sort out with the revision notes, and as soon as that's sorted, we'll pass them out to everyone. So for the time being, if you can join our WhatsApp channel, as soon as the issue is corrected, we'll start sending them out to people. And like I said, I've had conversations with teachers from Eden, so that's a different process. So you can look at how sodium and fluorine react with each other, magnesium and oxygen react with each other, calcium and chlorine react with each other. So these are really good examples to go through. Right, ides and ates. So ionic compounds are called ides if they contain only two elements. So for example, if you've got Na, which is sodium, and Cl, which is chlorine, if you react them together, it makes sodium not chlorine, but sodium chloride. So you change the N to ide. Now, if I said sodium and chlorine, which is sodium chloride, but now if I add oxygen to it, instead of being sodium chloride, it becomes sodium chlorate. So let me give you another example. Let's do lithium and fluorine. So that would be lithium fluoride. But if I do lithium, fluorine and oxygen, what we're going to call it? Lithium fluorate. So no oxygen, lithium fluoride. If you add the oxygen, lithium fluorate. So, quick exam question. Two compounds of magnesium are magnesium oxide and magnesium bromide. Give the names of the elements combined in magnesium oxide. This is the kind of questions that are more likely to come up in a foundation paper, but it can come at the beginning of a higher paper too. So the elements that make up magnesium oxide are magnesium and oxygen. Okay, so now let's look at 1.26, which is the formula of ionic substances. So cations are formed when an atom loses electrons. So it's something we've gone over multiple times now. If a metal loses an electron, we call it a cation or a positive ion. So we can call it either. It's because cats have pores. We can call it a cation or a positive ion. Now an anion occurs with non-metals. This is where they gain electrons to become negatively charged. So M has written, what about bromine from bromide? That's correct, yeah. So magnesium bromide would be magnesium and bromine. So well done, yeah, that's correct. Now, anions are non-metals when they've gained an electron. So remember what we looked at before? Group 1 make positive +1 ions. Group 2 make +2. Group 3 makes +3. Group 7 makes -1 ions. Group 6 make -2. And group 5 make -3. What this means is, if you, for example, got magnesium and oxide, and it says, what's the formula of this? Magnesium is from group 2. Now, if you go back 1 second, group 2 makes a +2 ion. Oxide, so that means we know magnesium is 2+. Now, oxide is from group 6. This is why you should just memorize this. Group 6 is -2 ion. So oxygen is from group 6. So oxygen is 2 minus. Now, what we want to do is balance these out so the overall charge is 0. So how many magnesiums do we need and how many oxygens? Well, magnesium's 2 plus, oxygen's 2 minus, so they cancel each other out. So we need one of each. Magnesium oxide, MgO. Now let's look at a different example. In fact, let's use the example that we had on the other slide, which is magnesium and bromine, magnesium bromide. Now magnesium's from group 2 and bromine is from group 7. So as we already know, magnesium is Mg2 plus, whereas bromine is from group 7. If it's 7, it's got 7 electrons already, it gains 1 more. So it becomes Br minus. Now, if you've got 2 plus, so you've got 2 pluses on one side and only 1 minus on the other. So how can we balance this out? The way we balance it out is we need to add another bromine. Now we've got 2 negative charges to balance out the Mg2 plus. So that means our formula will be MgBr2. So magnesium bromide is MgBr2. There's a really good question that's been asked. What about group 4? Now group 4, I suppose if you were looking at the same pattern, if it's got 4 electrons, it either wants to lose those 4 electrons or it wants to gain 4 electrons. Both are very, very difficult. So usually group 4 only covalently bond. In theory, they can ionically bond, and if anyone does A-level or does it at university, they'll see examples. But from a GCSE perspective, it'll be something that comes in more detail when we look at ionic bonding. So here we've got an example. We've got ammonium ion, which is NH4 plus. And if you don't know that's an ammonium ion, write this down separately and memorize that. An ammonium ion is NH4 plus. A nitrate ion is NO3 minus. So what's the formula of ammonium nitrate? Well, NH4 is already a plus. NO3 is 1 minus. So that means we need one of each. So the formula will be NH4 NO3. That is the incorrect answer. That is the correct answer. Oh, sorry, these are giving you examples and you have to pick one. Sorry. I didn't realize it was a multiple choice question. So which would be the correct answer? It would be NH4, NO3. Good. So in this question, we've got SO4 2 minus and we've got Ti, titanium 4 plus. So what would be the correct answer here? We've got 4 examples, we've got 4 choices. Now, if you've got Ti 4 plus and you've got SO4 2 minus, how many of these do we need? How many of the 2 minuses do we need? We need 2 of them. So it becomes 4 minus. So the correct answer would be this one. Because you've got 1 titanium and you've got 2 SO4 2 minuses. So Saif, did that answer make sense? What I just explained? So what is an ionic compound? A large structure made of positively and negatively charged ions that are held together by strong forces. And the strong force that holds them together is the positive and negatively charged ions, and it's the electrostatic attraction between the two. Now we can show it as this 2D model. We can show it like this as the ball and stick model, and you can show it like this as a 3D model. Now these kind of models have the positives and negatives, and that's something that will come up later. So ionic compounds have a giant lattice. So this is the kind of stuff that comes up quite often and you should be aware of. So they've got a giant lattice the anions and the cations are attracted to each other in a regular repeating pattern. So if you have a question about the structure with the ionic compounds, you say giant lattice, negative and positive ions are attracted to each other in regularly repeating patterns. Now, and then the electrostatic force of attraction between the oppositely charged ions hold everything together. Those two paragraphs are paragraphs you need to know. Okay, covalent bonds. A covalent bond happens between two non-metals. It's when atoms share electrons to fill their outer shells. So what are covalent bonds? Some substances with covalent bonds have very large molecules, like polymers. Substances with covalent bonds have huge structures, like diamond and silicon dioxide. Now, silicon dioxide is what we commonly call sand. These are huge structures. These have trillions upon trillions of atoms. But we can also have small structures like this, carbon dioxide, NH3. Now the answer is already shown here, I think. Sodium reacts with chlorine to form sodium chloride, which contains ionic bonds. Hydrogen reacts with chlorine to form hydrogen chloride, which contains covalent bonds, which you can see in figure 6. Figure 6 shows the dot and cross diagrams for these compounds. Sodium chloride, which is ionic bonding. Hydrogen chloride, which is covalent bonding. Describe the differences between an ionic bond and a covalent bond. This is a really good question to get an understanding of what kind of questions come up in exams. This is the kind of question I would memorize. This is a 4-mark question. And look, if you look at the question, it says here, describe. So describe does not mean explain. Describe just means say what you see. What is an ionic bond? What is a covalent bond? So what's an ionic bond? It's the electrostatic force between oppositely charged ions. That's your first 2 marks. Electrostatic force, 1 mark, between oppositely charged ions or between anions and cations. And what's our 2 marks for covalent bond? It's a shared pair of electrons. Atoms share a pair of electrons, but it's the word share and pair. That's where you get your marks from. And it gives you all the other additional marks you can get there as well. But that's how straightforward that question can be. Now, if you look at the question, how many people would have seen that question and thought, oh, I'm not going to answer that? A lot of students would have looked at that question and would have thought there's no point me answering that question, because not everyone. I'm not suggesting anyone that's listening online at the moment, but a lot of students look at wordy questions and they get put off, which is why initially go straight to the main part of the question, which is describe the differences. But as soon as you say describe, there's no explanation involved. You are just saying what you see. What is the ionic bond? What is a covalent bond? So don't get put off by questions like that. Okay, what is made through covalent bonding? So here's lots of covalent bonds. When covalent bonds form between atoms, they create molecules. Now you need to be able to draw all of those. So I'm going to go through a few of them just as an example. So let's start with something simple. Let's start with chlorine. So chlorine, the easiest way to draw covalent bonds is this. Chlorine is in group 7. If it's in group 7, how many more electrons does it need? It needs 1 more electron. Whatever the number of electrons it needs, however many electrons it needs, that's how many electrons it's going to share. So now you just draw 2 circles. How do we know it's 2 circles? Because it'll say Cl2. It'll let you know that there's 2 chlorines involved. Now, how many electrons does chlorine need? It needs 1. So from its 7 electrons, if it needs 1 more electron, it's going to share 1 of its electrons. So this chlorine here is going to share 1 electron here. Now, it's got 7 electrons altogether. So if it shared 1 in the middle, it's got 6 more left. So there's 1 in the middle. This is number 2, number 3, number 4, number 5, number 6, number 7. Now this next chlorine also has 7 electrons. It also needs 1 more electron. So it shares 1 in the middle and the remaining 7, so number 2, number 3, number 4, number 5, number 6, number 7 are around the outside. Now look at both chlorines. It's got 1, 2, 3, 4, 5, 6, 7, 8. Chlorine on the left has got 8 electrons. So it's got full outer electron shell. The chlorine on the right hand side has got 1, 2, 3, 4, 5, 6, 7, 8 electrons. So now they've both got full electron shells. Remember, a chlorine is a shared pair of electrons. So we've got 2 electrons in the middle there. So you can draw it this way as well. That one line represents 2 electrons. That's a pair of electrons being shared. Now let's look at a slightly more tricky one. Let's look at this one. Nitrogen. Now nitrogen is in group 5. If it's in group 5, so we're talking about nitrogen N2, it's in group 5. So how many more electrons does it want? It wants 3 more. So that's how many we're going to share in the middle. So let's do our overlapping circles. Let's look at nitrogen on the left first. It wants 3 more, so it's going to share 3 in the middle. Let me change my color to make it really clear. So the one on the left is going to share 3. 1, whoops. So it's going to share 1. So let me just erase this again. It's fine, I'll just stick to one color, make the circles a bit bigger. So the nitrogen on the left, it wants 3 more electrons. So it's got 5 in its outer shell, but it's going to share 3. 1, 2, 3. And it's got 2 more to go on the outside. The nitrogen on the left has also got 5 electrons. So it's going to share 3 in the middle. 1, 2, 3. And it's got 2 more. Now if you count the electrons for both nitrogens, how many electrons do they have? 6 are being shared. 6, 7, 8. So that's got full electron shell. The nitrogen on the left has got 6 in the middle being shared, plus 7, 8. It's got full electron shell. So they're both happy. So this is the process. This is how you've got to understand it. Now, a slightly more tricky one, I suppose, would be this one. But let's quickly understand it. So we've got carbon in the middle. Now you'll be told that it's CH4. That means 1 carbon is attached to 4 hydrogens. Now an easy way to do this kind of question is, don't forget that, what does this one line represent? It represents a shared pair of electrons, meaning hydrogen gives 1 electron and carbon gives 1 electron. But using the principle I explained before, let's add our 4 circles. Now carbon has got 4 electrons and it wants 4 more electrons. So it's going to share all 4 electrons. Hydrogen looks like this. It's got 1 electron and it wants 1 more. So it wants to gain 1 electron as well, so it's going to share 1 electron. So this hydrogen is going to share 1 electron and the carbon is going to share 1 electron. This next hydrogen shares 1 electron and the carbon shares 1 electron. This hydrogen shares 1 electron and the carbon shares 1 electron too. This hydrogen shares 1 electron and the carbon shares an electron. Now carbon, we said, needs 4 more electrons, so it shares 4 electrons. So it shared 1, 2, 3 and 4 electrons. Hydrogen, each hydrogen needed one more, so each hydrogen has shared one electron. So now, each of our hydrogens have got a full outer electron shell because they've got these access to these 2 electrons. Whereas carbon has got access to all 8 electrons because it's sharing electrons with all 4 hydrogens. So that carbon now has a full outer electron shell as well. Now everything is drawn out here, so you can zoom into these and just learn them directly from the slide. But this is something that I really would advise that you practice. Now, the size of these atoms are all similar, but they're not exactly the same. The diameters are slightly different, but they're all around 10 to the minus 10 meters, meaning they are 0.1 nanometers wide. Now, I have seen in foundation questions sometimes that can come up as a multiple choice question, but it's not something that needs to be that we need to spend too much time on. Okay, simple covalent molecules. When atoms share electrons, they form covalent bonds. So when atoms share electrons, they form covalent bonds. These bonds hold atoms together really strongly. Substances with covalent bonds are often small molecules, such as these small molecules. Now all these covalent bonds you need to learn how to draw. So these are nicely drawn out examples for you. And some practice questions for you to check your understanding with as well. Okay, different bonds. And when do they occur? So ionic bonding occurs between metals and non-metals. And this is where the metal transfers its electrons. So electrons move from the metal to the non-metal, meaning the metal becomes positive and the non-metal becomes negative, and then you have electrostatic attraction between the two. Covalent only happens between non-metals. So this is where atoms share electrons to get full outer energy levels, outer shells. So this is a paired share of a shared pair of electrons. So covalent bonding is a shared pair of electrons. And metallic bonding only happens between metal atoms. And this is where metal atoms share free moving electrons. So metal atoms share free moving electrons. Now what happens, for example, let's say you've got a metal wire. I'll just show it here first. Let's say you've got sodium. If you had a metal wire, and you don't have metal wires made of sodium, so let me use copper as an example. So let's say we've got copper and it's got 2 electrons in its outer shell. Within that metal, those 2 electrons become delocalized, and meaning they separate from the atom. They're free to move everywhere. Now if copper loses 2 electrons, that means what you're left with is a 2+ ion. So there's a whole of this metal wire is made up of 2+ copper ions, and you've got all those electrons that are free now just to move through the metal. This is why metals can conduct electricity and they're good conductors of heat, because all those free electrons can transfer the heat through. Now in a metallic bond, it's the attraction between these free electrons and the positive ions. That attraction is what we call a metallic bond. Now, it's important you memorize all these compounds, the properties of ionic compounds. You need to know the properties of ionic compounds, covalent compounds, and metallic compounds. You need to know examples. For example, in covalent bonding, you need to know about diamond, graphite, graphene, but we're about to go through that now. So it has high melting and boiling points. That's because ionic bonds are really strong, and that means you need a lot of heat energy to break them. They're solid at room temperature because they don't melt easy. Now, they conduct electricity when they're melted or dissolved. So melted, another word, we usually use the word molten, but melted is a similar thing. Now, when they're dissolved, the ions are free to move. So it says can move, but I prefer the word free. They're free to move and carry the charge. So when it's a solid, let's say I've got a battery pack here and I've got a bulb here. If I sprinkle some salt here, will the bulb turn on? No, it won't turn on because all the ions are within the solid, meaning all the positives and negatives that make up the ionic compound, they have strong electrostatic attraction, meaning these ions are not free to move around. They're part of the solid. But the second you melt or dissolve it, then the ions become free to move around. At that point, they are free to conduct electricity, so they would complete a circuit. Within that liquid, if this, for example, that negative ion becomes charged, it can move with that charge. It's free to move. Now, ionic compounds dissolve easily in water. So for example, you get some salt and you add it to water, give it a mix. And give it a mix, that salt, so if you add salt in water and it will mix it, it will dissolve into the water. And what actually happens is, for example, if you've got NaCl, it breaks apart, it separates in the water into Na plus and Cl minus. So the ions separate. Same thing if you've got HCl, which is an acid. It'll separate into H plus and Cl minus. It's the H plus part that causes acidity. But we'll cover that in a topic that's about, we'll be covering that soon in one of the topics. Now, brittle means it breaks easily if it's hit. So if you think of salt, if you hit it, you can crush it into finer powder. Okay, so why has it got high melting and boiling point? Because strong bonds need lots of heat. Why does it not conduct electricity when it's solid? Because the ions are not able to move, they're stuck in place, so no electricity flows. Why can they conduct electricity when they're melted or dissolved? Because the ions move freely, allowing electricity to flow. Now, this is a really good question for you to go through. I'm not going to go through it at the moment. We will be going through a lot more questions when we do AMALathon 2. So if anyone's not sure, is unaware about AMALathon 2, we will be doing AMALathon 2 in May and the beginning part of June. So what we'll be doing is similar to this. We'll have a 1 week session where we'll be ensuring that we have activated our brain. So if you remember what I said about AMAL yesterday, AMAL stands for A, activate your brain, M, memorize, A, apply, and if you do all those three things, you've learnt. So what we'll be doing what we're doing right now is we're activating our brain. We are making sure that we've understood all the content. In May, in the 1 week holidays, we'll be going through all the paper 2 content, making sure we've activated our brain, understood it fully. Now, the weekend before every single exam, we'll be doing a pre-exam work. I'll be running a pre-exam workshop, or AMAL will be running a pre-exam workshop. And in that pre-exam workshop, we'll be doing loads of past paper questions linked to our analysis of the last 7-8 years of past papers. So we're going to be covering the highest leverage topics, the topics that we feel come up year in, year out. Now, usually I'm around 80% accurate. So hopefully those that are able to join us, you'll take a lot of benefit from it. It will be almost like doing a walking, talking mock exam or a walking, talking predicted paper. So I would definitely advise that you join. Now, what happens to bonds when we boil water? So the heat is absorbed, water takes in energy from the heat. Now, when the water takes in energy from the heat, the molecules move faster. That breaks the intermolecular forces, and that means that some of the molecules can escape. So what we're actually trying to say here is, this is what a water molecule looks like. Now, if I've got another water molecule here, you can have weak intermolecular bonds that join the molecules together. Now, what happens is, if I heat up just an individual molecule of water, it takes a very high temperature to break these covalent bonds. but that's just one molecule, so it's already a gas. But to separate hydrogen from oxygen takes a high amount of energy. But if I heat up this example here, it does not take much energy to break this intermolecular bond. So at 0 degrees, it breaks enough bonds for it to become a liquid. At 100 degrees, it breaks enough bonds for it to turn into a gas, for it to boil. So the intermolecular bonds are weak, but the covalent bonds are strong. Now every single session that we're doing on YouTube up until Thursday, so we were on yesterday, we had a 3-hour session for AQA, 3-hour session for Edexcel. All of that, as soon as we finished, went straight onto YouTube. Same thing's going to happen today, and same thing's going to happen every day until Thursday. So it's going to be Saturday, Sunday, Monday, Tuesday, Wednesday, Thursday, 6 days, 18 hours. So that's what the AMALathon is. And like I said, because Edexcel's so content heavy, we'll be doing an extra hour of triple award for Edexcel. So those are in triple award, there'll be 1 extra hour, and you don't have to attend. Even if there's only a few, or even if there's no one, I'll still talk straight, teach it the way as though someone was there, and you can just watch the recording afterwards. And like I said, I was saying thank you guys before, because we did have over 1,000 views on yesterday's AMALathon, day 1. So again, really appreciate everyone that supported us. Now, in a simple covalent molecule, it's got low melting and boiling points because there's weak forces between the molecules. It means little energies need to break them. So the intermolecular forces that are between the molecules are really weak, and it does not take much energy for them to break. It does not conduct electricity. Why? Because there's no free electrons or ions to carry charge. Now, why can an ionic compound, so this is something I need everyone to really pay attention to and listen to and understand. Why can an ionic compound, for example NaCl, if I put that into water, why will it conduct electricity? Because it has free ions to conduct electricity. A lot of students always put the word electron. Ionic compound, we're talking about ions. Covalent bonding is a shared pair of electrons, so we're talking about electrons. Metallic bonding, we're talking about delocalized electrons, so again we're talking about electrons. But ionic bonding, the clue's in the name, we're talking about ions. Now, covalent bonds are usually liquids or gases. Why? Because what joins these molecules to each other is the weak intermolecular bonds. And it does not take much energy to break those weak intermolecular bonds. So again, there's a really good question here for you to have a go at once you've got the resources. I'm still going to just show it here and give it a couple of seconds. So afterwards, when you're watching the video, what I'd advise you pause, think about what the answer is, and then compare what you've written down to the answer. Okay, graphite and diamond. Whoops. Okay, now what I'm going to do is, because today there was supposed to be a question and answer session at 4:30, and it was going to be a 15-minute session. So I'm actually going to teach into that time slightly, because what I would like to do is finish this graphite and diamond bit. So we'll probably be about 5 minutes till we have a break. So hopefully everyone's okay with that, and I'll still make sure we do the right amount of physics as well. So we've got graphite and diamond. What are diamond and graphite? So diamond and graphite are both giant covalent structures. So everything we just went through on the previous slide, or the previous slides, the properties of covalent bonding, we were looking at small covalent molecules. So we were looking at simple covalent bonding. This is now covalent bonding that makes giant covalent structures. So both diamond and graphite are made up of carbon atoms, but whereas in water Each water molecule only has 2 covalent bonds. In diamond, we're talking billions and trillions of covalent bonds. In graphite, so this is diamond, and in graphite, we're looking at, again, millions and trillions of carbon atoms, covalent bonds between carbon atoms. So that's why they're giant molecules, the giant covalent structures. So diamond is a giant covalent structure where each carbon atom is bonded to 4 other carbon atoms. So each carbon is bonded to 4 other carbons. Then each one of these carbons is bonded to 4 carbons. Each carbon is bonded to 4 carbons. And that just continues and continues and continues. And that means it's very hard, because if you're going to break it, you've got to break through all these covalent bonds, and covalent bonds are very strong. That makes them strong as well, and that means all the electrons are trapped. They're part of the covalent bond. That means it will not conduct electricity. Now graphite on the other hand, is a giant structure, but it's a little bit different. A giant covalent structure where each carbon atom is bonded to 3 others, forming layers that can slide. So here's the layers. That's one layer. And then we've got a layer underneath it. And in between it, if you can see those dotted lines, those are weak intermolecular bonds. So in graphite, those weak intermolecular bonds can be broken and each layer can slide over the other. And that makes it soft, and it actually makes it a good conductor of electricity, because graphite only has 3 bonds. So each carbon, even though it's got 4 electrons in its outer shell, it makes 1 bond, another bond, and a 3rd bond. That 4th electron is just free to move around in between the layers. So for that reason, it's got a free electron that allows it to conduct electricity. So in graphite, 1 electron from each carbon is delocalized. It's delocalized, it's free to move around. So again, in ionic bonding, when it's molten, when it's melted or dissolved, what's free to move? An ion. But in graphite, what is free to move? A delocalized electron. In metal, what's free to move? A delocalized electron. So, uses of diamond and graphite. So diamond can be used for cutting tools, because diamond is the strongest known substance to man. So if you take diamond dust, and for example, if you've got one of those drills that spin around in a circle, if just on these sharp bits you put a little bit of diamond dust, it makes them a lot stronger. Jewelry and industrial coatings. And graphite we can use for pencil lead. So our lead is made of graphite. So whenever you use lead and you're drawing something or writing something, what you're actually putting on the paper is a layer of lead. Now when we say lead, we mean graphite. We don't mean lead, the metal. It's just called that because it has a similar colour. And it can be used for lubricants because, like we said, the layers slide over each other, because there's weak intermolecular bonds in between. So if you've got a squeaky door, you put a bit of graphite there, it reduces the squeakiness. It can be used in electrodes in batteries. We even use so that was a really good question from Saif. I'll answer that in just a second. Now when we're doing electrolysis, we use graphite electrodes because they are unreactive, number 1, and number 2, they conduct electricity. Now, how do we cut diamonds in the first place? We cut diamonds with other diamonds. The only thing that can cut a diamond is a diamond. So, quick explanation. Why is graphite soft and slippery? Because it has layers of carbon that slide over each other. That makes graphite useful in pencils and lubricants. It conducts electricity because, remember, the carbon only makes 3 bonds. It has 1 delocalized electron. So it has a free moving or delocalized electron. And why does it have a high melting point? Because it's got strong covalent bonds between the carbon atoms. Lots and lots of strong covalent bonds. And that means a lot of energy is needed to break those bonds. Properties of diamond, they're very hard because it's got strong covalent bonds, make the diamond useful in cutting tools and drills. They're transparent and sparkly, so that means they're beautiful and used in jewellery. They've got high melting points because they've got strong bonds. Remember, each carbon atom in diamond is bonded to 4 carbons. So there are trillions upon trillions upon trillions of, in fact, it would be 6 followed by 23 zeros and multiples of that. That's how many carbon covalent bonds you would have, carbon-carbon covalent bonds. Okay, fullerenes and graphene. Now this is where we're going to continue from tomorrow. So thank you very much. I really appreciate that you all were happy to stay for the extra 5 minutes. We will continue at 3:40. So I'll see you all in 5 minutes. Okay, welcome back everyone. Let's start our physics. The aim today is to finish the rest of this topic 2 in physics. It is quite a big topic, but hopefully we're going to finish all of it by the end of this 1 hour. So we're going to continue till around 4:35, 4:40 if we need to. But we're gonna finish this. Like I said, we don't have the question answer session today because we have been answering the questions live. So as long as there's no questions that are needed, we'll use that time. But again, please keep asking questions and I'll keep answering them live as we're going through. And if I'm not able to, then we'll answer it in the next session. Now there's a question here for AMALathon 2. Will that be live on YouTube as well? It will be on YouTube. It will be live, but that will be for members only. So there will be, because what we'll be doing is 18 hours in the May holidays, and we'll be doing 18 hours during, as in we'll be doing 3 hours before each exam. So that's another 18 hours. So for those 36 hours, we'll be charging £3 an hour. That hasn't been, we've not made that live yet, but as soon as we do, if you've joined our WhatsApp channel or you've registered with us, so you can register in the description below, then you'll get information regarding it. But that'll be 36 hours of learning, so we'll be covering everything. This session is completely free, the AMALathon 1 is completely free. It's just so that everyone gets a bit of a taste of what we're doing and how we're doing it. AMALathon 2 will have a small charge. So I'm hoping everyone can now see my screen. Yep, everyone can see the screen. Let's get started. So this group's been really good, asking lots and lots of questions. So please keep asking questions and I'll keep answering them. Like I said to you guys before, let me just really reiterate this. This is why we called our company Amal, because the first A stands for activate, which is understand. So what we're covering at the moment is Edexcel exam board. AQA is at 10 AM till 1 PM. It'll be exactly the same. So if anyone's asking about AMALathon 2, we'll be covering the Edexcel exam board and AQA exam board during the AMALathon 2 as well. So the A stands for activate. In other words, understand everything. And that's the first step to succeeding in your exams. So just through your understanding, you can already get yourself to a reasonable grade. Then memorize and apply. They're interchangeable. The more past paper practice you do, the better you're going to understand. Firstly, you're going to understand how many questions you can answer just from your understanding. There's lots of questions that you can answer without having to memorize anything. The second thing they'll show you is what kind of questions require memorizing. For example, whenever a question comes on genetic engineering, it's less to do with, you have to have understood it, but it's having memorized the process and writing the keywords. But you understand what those keywords are, and you understand what kind of question, how the question is going to be asked only from past papers. And then, so this way you understand what questions you can answer through understanding. You understand what kind of questions you can understand through memorizing. And thirdly, there are some questions that are linked to kind of like scientific inquiry, linked to core practicals, which again, you start to understand better when you do go through past paper practice. So what we're doing at the moment with the AMALathon 1 is we're activating. What we're going to do with AMALathon 2 is we're going to activate and understand everything for paper 2. With the pre-exam workshops, our main focus will be the application and identifying and helping you see the areas that need memorizing. But when you've gone through those 2 phases anyway, where you've gone through the process of understanding everything, you've gone through the process of applying to loads of past papers, you automatically start to memorize. Because anything you do more than once, the more often you do something, the more you repeat it, the more importance your brain attaches to it. And right now, hopefully we're at that point where our brains are attaching a lot of importance because it's GCSE. So automatically you're going to memorize and understand things a lot better than you maybe were when you were in year 10, because there was no pressure then. So the AMALathon 2 will have a lot more application, particularly before each exam. So Newton's second law, the main thing we need to understand from this slide is this formula. Apologies, I'm not sure why it's gone so far back. Is this formula F equals ma. So Newton's second law states that acceleration of an object is directly proportional to the net force. So acceleration is directly proportional to the force and is inversely proportional to the mass. Meaning, when the force increases, the acceleration increases. But when the acceleration increases, the mass decreases. So, in other words, if the mass increases, what happens to the acceleration? It reduces. So, I mean, that's, if you think of it like this, if you're running as fast as you possibly can, and you run 100 meters in 15 seconds. Now, if I say run again, but this time put your school bag on your back with all your books inside, are you going to still do it in 15 seconds? No, because your mass is increased. So what's happened to acceleration? It's decreased. They're inversely proportional. When you increase one, you're decreasing the other. So this formula needs to be, you won't need to memorize it because it's going to be in the formula sheet, but you need to know the units. The formula sheet is one bonus that you've got 100%, but we shouldn't become reliant upon, well, we're not, from the formula sheet, we're not going to get units. So force is newtons, we've got to remember the units. Mass is in kilograms, and acceleration is meters per second squared. So, real life examples linked to Newton's second law of motion. A lighter car accelerates faster than a heavier truck with the same engine power. Why? Because of what we just said. If the mass has increased, what happens to the acceleration? It decreases. They're inversely proportional. So let's have a quick go at this practice question. Now, we're only going to have a go at a couple of questions. Now, what's happened with these questions, just to fit a few questions into one page, they've been condensed. So let's say a car weighs 1,000 kilograms. That means mass equals 1,000 kilograms. And let's say the force equals 5,000 newtons. Then what does the acceleration equal? Now, if we are saying F equals ma, if we convert that to a triangle, for those that prefer using a triangle, F goes at the top, and we've got M and A here. So acceleration equals F divided by M. So for this question, it's going to be 5,000. So let's substitute the values in. 5,000, which is our force, divided by 1,000. So acceleration equals 5 meters per second squared. So every single formula for anyone who's currently in year 10 and for anyone that's currently in year 11 will be in the booklet. So everyone for the next 2 years, this year's year 11, next year's year 11, so this year's year 10, will get a formula booklet, and that will have every single formula on it. But the problem is with that formula, it's got so many formulas on. Whenever you answer a question, always answer it like this. So I'm just going to give you an example with the box one. You know how I just did this question? You always write down what you've got, your information you've got first. So here we know mass equals 20 kilograms and force equals 60 newtons. Now, if you've got those two units, you've got mass, you've got force. Now, when you look through the formula booklet, it'll be easier because you're trying to find acceleration. So now you've got F, M, and A. So automatically finding the right formula will become a lot easier. But if you practice from beforehand, it'll be a lot easier to get to the correct formula. Or you might not even need to look at the correct formula. Look at the formula booklet. Now, definition of weight. Weight equals mass times gravity. So weight is measured in newtons, mass is measured in kilograms, and the G is for gravity, gravitational field strength, which on Earth is 9.8 meters per second. Now, in an exam question, most of the time they'll give you 9.8 meters per second. If they don't give it to you, you can just use the number 10. Now, what is weight? If someone is falling from a plane because they have chosen to fall from a plane, they've got a parachute on the back, then what is pulling them down? Gravity. When something has particles, when you add up all those particles together, what do we call that? We call that a person's mass. So when gravity acts on someone's mass, that is weight. So it's gravity acting on our particles, gravity pulling down particles, mass times gravity, and that gives weight. So how is weight measured? To measure weight, we need to determine the force pulling an object towards the center of the Earth. So this is done using a Newton meter, a device that measures force in newtons. So you can use a Newton meter. It is also called a spring balance, but Newton meter is the correct term. Gravitational field strength. So gravitational field strength is just gravity, is what I've just explained. It's the force exerted by gravity on a mass of something. So it is, now this is the same formula as we showed before, W equals mg, except it's been rearranged so that gravity is the subject of the formula. And what does gravity do? It pulls down on someone's mass. It is the force that acts on a mass, on particles. So let's have a quick go at this question. So remember, first thing we've got to do is convert everything to the correct unit. So in this question, we're saying calculate its weight. So we want to find out what weight equals. Whenever you answer the question in physics, always start by writing down the units that you have. The next thing we have is mass. Now at the moment, it says mass is 245 grams. We need to convert that to kilograms. Now that means we're going from a smaller unit to a bigger unit. When you go from a small unit to a big unit, what do? We divide. When you go from smaller to big, we divide by 1,000. If anyone needs help with converting units, there is an AMALearning video, I think it's 2.4/2.5, that goes through how to convert units. No, sorry, it's 1.4, 1.5. And that shows you how to convert units. So if you're struggling with converting units and you're struggling with the understanding between nano and pico and micro and milli and how you convert from one to the other, that video, it's only about 3-4 minute long video, but it will bring a lot more clarity. Or in the AMALathon video is what I went through yesterday in the biology section. So now if we divide 245 by 1,000, we get 0.245 kilograms. Now what's the other unit we have? We've got gravitational field strength, which is G, as 10. So here they've given it as 10, so we just use 10. So what we're going to do, W equals mg, substitute the numbers in, mass is 0.245 times by G, which is 10, which equals 2.45 newtons. And we can simplify that to two significant figures, which is 2.5 newtons. In physics, generally speaking, 2 significant figures is preferred. But if you put 3, like here we've got 1, 2, 3 significant figures, that's absolutely fine as well. 2 or 3 is okay, but don't ever do 4. Okay, core practical. How can you study the impact of changing force on an object's acceleration? So what you do is you connect a light gate here, a light gate here, you put a piece of card there. So when it goes through the light gate, the timer starts. When it goes to the second light gate, the timer stops. So that means we have the time. You've also got the distance measured here. So we've got distance. And here we can keep adding more weight. So this way we can measure, using the light gates, you can measure the velocity here. The light gate will measure the velocity here. So it shows you the change in velocity and the time it takes. In other words, it calculates the acceleration. So the first time you do this experiment, you do it with just 1 Newton. Then you add another one on top. Then you add another weight on top. Then you do the whole thing again. You measure the acceleration. And the light gates do that automatically. Then you do it again, measure the acceleration. So what you're seeing is, what is the impact of the changing force on the object's acceleration? So what you do is you connect both light gates to the data logger to record velocity and analyze acceleration changes. So you're comparing the acceleration, the velocity from here to here. So this is your initial velocity, that's your final velocity. And using that, remember A equals V minus U divided by T. The final velocity minus the initial velocity. So the light gates do all that calculation for you. Then you arrange the equipment as shown in the diagram, ensure proper alignment for accurate results. The light gates A and B measure the velocity at each point, allowing acceleration to be calculated between them. Then you increase the force here by adding weights in 1 Newton intervals. Then you measure and record the acceleration each time. And each time you add 1 Newton. So you keep adding another Newton, and you keep working out what the acceleration is. So you would make a graph, and here you would put the weight that you're adding, because that's a force, weight is a force. And here you'd write down what your acceleration is. Okay, circular motion. Now this topic is higher only. So this topic is higher only. Now circular motion, and I'm going to explain 2.2 and 2.1 in the same slide. And 2.21 is also higher only. So it's for both combined higher and it's for triple as well. But it is a higher topic. So circular motion occurs when an object moves along a circular path. So that's circular motion. So for example, a car going round a roundabout, that's circular motion. Now it has a constant speed, but it's continuously changing direction due to centripetal force acting towards the center. So if you look at 2.21, that's centripetal force. And 2.2 is circular motion. And I'm just going to explain this in my own words first before I read through all the content. Now imagine you've got, not imagine, this is a real life example. There's the Earth and here's the Moon. Now the Moon, say, is moving this way. The Earth, however, has and this is what we call centripetal force. The Earth's gravity pulls the Moon towards its center, pulls it downwards. So now what we've got is the Moon going this way and the Earth pulling the Moon towards its center. So you end up with a force somewhere in between. So the Moon moves that way. Now the Moon is moving in this direction, but what's happening? It's being pulled towards the center of the Earth. So it ends up moving somewhere in between. So that pull towards the middle is what we call centripetal force. So the Moon is now here. It's moving this direction. But which way is the Earth pulling it? Towards the center. So it ends up somewhere in the middle. Now, if you look at it, how is the moon moving? It would continue moving that way. That's its momentum. That's its inertia that would continue making it move in that direction towards the right. The Earth is pulling it. The centripetal force is pulling it towards the center. So the moon goes somewhere in between, and it goes that way. In other words, the moon is moving in a circular motion. But why is it moving in a circular motion? Because of the centripetal force. So now the moon has gone round in a full circle. And if you think about when you're in a car, if someone goes round a roundabout really fast, which way is your body moving? Is your body also going around a circle, or is it moving in the direction the car was moving before? So you kind of get pushed, even when you turn a corner sharply, does your whole body move with the corner, or does your body continue moving forward? So the car turns to the left, but your body moves towards the right in the other direction, because that's the direction your body was already moving. So circular motion occurs because of that centripetal force. It's that force pulling things towards the middle. So I'll rub this out now. I'm just going to check the questions to see if anyone has a question about that. Because I know that's a trickier topic compared to some of the others. So, circular motion occurs when an object moves along a circular path with a constant speed, but is continuously changing direction due to a centripetal force acting towards the center. So the example that we just showed was the moon, and it's going around the Earth, and it's constantly changing direction. But ultimately it's still going around in a circle. Why is it still going around in a circle? Because the Earth is pulling it towards the center, that centripetal force. Now, that does not mean the moon's speed changes. So let's assume the moon's speed stays at 1,000, and I'm making this number up, kilometers per hour. It travels at 1,000 kilometers per hour. But why do we still call this velocity a change in velocity? Because it's going at constant speed, but it constantly changes direction. Now remember, velocity is a vector. A vector is something that has a size and a direction. So if something has a change in size, so it goes from 1,000 kilometers to 2,000 kilometers per hour, then we would say velocity is increased. If something is going at 1,000 kilometers per hour in this direction, and then it goes in a different direction, and then it goes in a different direction, but it stays at 1,000 kilometers per hour, the direction is still changing. That means the velocity is changing. So we would call that acceleration. So let's look at the key points. Speed stays the same. A car going around a circular track, but the velocity changes. Why does the velocity change? Because the direction changes. The velocity changes too, because that includes direction. Velocity is not just a size, it has a direction as well. So if the direction of changes, the velocity is changed. An example is a satellite orbiting Earth. Acceleration exists. So it's called centripetal acceleration, always points to the center. It's caused by gravity, tension, or other centripetal forces. So tension is, if you think of, if anyone's ever seen someone get, for example, like a bucket of water, and then spin it around in a circle, and the water does not fall. That's because when you reach this point here, where the water would fall, the force of the water is making it move in a different direction. Gravity is pulling it this way, but at that point the water is actually moving in a different direction. So it ends up being somewhere in between. The water actually moves in somewhere else. It doesn't fall down because of the initial force that was acting on it. And centripetal force is still pulling it towards the center, so it makes it go around in a circular motion. So centripetal force is the force that keeps an object moving in a circular path. So that's what keeps the moon orbiting the Earth by acting towards the center of the circle. Without this force, the object would move in a straight line due to inertia, as in due to momentum. So if something is moving in that direction, it's going to keep moving in that direction unless another force acts on it. In this case, the Earth has a force and it pulls it towards itself, which means the moon ends up moving in a direction in between the two, because now there's two different forces acting on it. Okay, inertial mass. So the definition of inertia. This is again a triple topic. 2.23 can come in foundation and it can come in higher, but the language that is used for 2.23 changes for foundation and higher. But this is inertial mass is specifically higher. So inertia is a tendency of an object to resist changes in its state of motion. So it's similar to what we would say momentum. So if something's moving in a certain direction, it'll just continue moving that direction. It means that an object at rest stays at rest, and an object in motion continues moving at the same speed and in the same direction, unless acted upon by an external force. So what that means, if you're in space and you're sat in a rocket and you have that initial burst of energy, and then that energy goes and you're now in space. Now let's say you're moving. Oops. So I'm just explaining this part here, and in the same direction unless acted upon by an external force. So let's say you're in a rocket and it's moving at 500 miles per hour. If there's no force acting on it, it will just keep moving at 500 miles per hour. If someone's driving a car on Earth and you let go of the accelerator, why does it eventually slow down? Because your force going forward is still going forward, but now you have a friction force slowing it down. But in space, you don't have that friction force that slows it down, so it just continues moving at that same speed. A better example of inertia is when a car suddenly stops. So as in, I'm assuming everyone's been through this scenario where we're driving a car, someone's driving a car and someone suddenly has to brake because possibly a cat's come in front of them or a child's come in front of them or a car's suddenly braked. And the second you brake, what happens? Your whole body moves forward. Why does your whole body move forward? Because when you're sat in a car, and you probably felt this again on the motorway, if you drive at 70 miles per hour, it doesn't feel like it's 70 miles per hour because you're sat in the car. So you're moving, your body's moving at 70 miles per hour and the car's moving at 70 miles per hour. So if you all of a sudden brake, the car, the friction that the brakes applied on the tires, so there's a friction force, the car stops. But we haven't stopped. Our body still keeps moving at 70 miles per hour. That's why seatbelts are really important. Because what the seatbelt does is it applies a force in the opposite direction to stop our bodies going through the windscreen. So inertial mass is a measure of how much an object resists a change in motion when a force is applied. And it's another way of looking at it is F equals ma. Okay, Newton's third law. So this is something that will come in both higher and foundation, but the questions will be different, asked in a different manner. In foundation, it's more about understanding the definition, whereas in higher, you need to understand the definition, but you're more likely to be asked to apply a question to apply your understanding to a question. So here, for every action, there's an equal and opposite reaction. This means that whenever one object exerts a force on another object, the second object exerts an equal force in the opposite direction. So here's two really good examples, because forces always come in pairs. These forces act on different objects but are equal in size and opposite direction. So let's have a look at the rocket launch. The rocket pushes exhaust gases downward. So when the flames come out the bottom, that pushes downwards. So what does the rocket do? The exhaust gases push the rocket upwards with an equal force, but in the opposite direction. So you have a force going downwards, and you get an opposite force in the other direction. That's the same size, but in the opposite direction. When we're walking or running, your foot pushes backwards on the floor, and the floor pushes your foot back, but in the opposite direction with an equal force. So if you want to run fast, the stronger your legs are and the more force you put into the ground, the more force the ground's going to put back, and you're going to move faster. Now, just quickly answer this question: is urea in combined science as well? Urea is covered predominantly in triple in the topic on the kidneys. So that's not, you need to know what urea is, but it's not something that you won't be asked questions on the kidney. Because what happens in the body is when proteins get broken down, the protein gets broken down to urea. And then when we add water to that urea, it turns into urine. That's all linked to the kidneys. 2.24. Momentum. So momentum is the amount of motion an object has. It depends on an object's mass and velocity, and is given by this formula. So that's momentum, mass, velocity. Now, the way you would write momentum is more like this, but it's easier just to put a P for the symbol. That's momentum, that's kilogram meters per second. Mass is in kilograms, velocity in meters per second. So what are the key properties of momentum? Momentum is a vector quantity. It has both magnitude and direction. If you increase the mass of something or velocity, the greater the momentum. Now, momentum is the amount of motion an object has. It's the likelihood that an object will just keep moving. So if you think about, if you've got a car and you've got a really big lorry, if they're both going at 70 miles per hour, so they're both going at the same speed, but obviously the lorry has a bigger mass, which one's going to be more difficult to slow down? It's going to be the lorry. So if they both take the foot off the accelerator, the lorry is going to have a greater momentum. So it's going to keep moving for a lot longer. So momentum, and the third thing that we need to remember from the key properties of momentum, momentum is conserved in a closed system, meaning total momentum before is the same as the total momentum after, like a collision. So momentum is the same before and the same after. So this is a question based on momentum. Now we're finding the momentum, so that's our first question mark. We've got mass, which we're dividing by 1000 because we're converting from grams to kilograms. So it's 0.45 kilograms, and we know the velocity, which is 12 meters per second. So all we're doing here is substituting our numbers in. Mass is 0.45, velocity is 12, and that gives us 5.4 kilogram meters per second. So here's two more questions for you to have a go at in your own time. 2.25, collisions. So again, collisions is a higher topic. In fact, 2.24 is also a higher topic. 2.25 is a higher topic. 2.26 is a higher topic. So a collision is an event where two or more objects come into contact with each other, exchanging momentum and energy. So you've got different types of collisions. You've got an elastic collision that you don't need to know. These are just examples. You don't need to know what an elastic collision is. You don't need to know what an elastic collision is or perfectly an inelastic collision is. You just need to know that momentum is conserved when a collision occurs. So this is a good example of a question I'm going to go through. What happens to momentum in the event of a collision? So if there's a head-on collision with equal momentum, so what that means is a car is going this way and it has a momentum of 10. Now, if it's going towards the right, let's call that plus 10. If a car is going in the opposite direction and it has a momentum of 10, it's going in the opposite direction, so we would call that minus 10. Remember, it's a vector quantity, so one direction is positive, one direction is negative. If these two objects collide, when they collide, what happens to the momentum? The momentum cancels out. Now, the momentum before the collision was plus 10 minus 10. So what was the momentum before the collision? So let's write here, total momentum before was 0. So now that they've hit each other and the collisions they've cancelled out, what's the total momentum after? It's also 0. In other words, the momentum has been conserved. So total momentum before was 10 minus 10, which is 0. After the collision, both cars came to a stop, meaning the total momentum after is also 0. So momentum is conserved. That's the main key fact that you need to keep in mind, that momentum is conserved. So what happens to momentum in the event of a collision? So now let's look at this one, two objects sticking together after collision. So here, we've got a moving truck, its momentum is 5,000 and it collides with a stationary car which had a momentum of 0. So what's the total momentum before? It is 5,000 because the truck had a momentum of 5,000, the car had a momentum of 0. So what will the total momentum after equal? It's also going to equal 5,000 kilogram meters per second. But what's the difference going to be? They stick together. Their combined momentum remains the same. So total momentum before the collision is 5,000 and the total momentum after is 5,000. But the only difference is this. The big lorry and the car have attached to each other. So now we've got an increase in mass. Remember, momentum equals mass times velocity. So if it equals mass times velocity, if your mass increases, because now they join together, what happens to the velocity? It decreases. But when these two numbers are multiplied, it still equals 5,000. So they move together with 5,000 kilogram meters per second total momentum, but because we've had an increase in mass, because they join to each other, we have a lower velocity because of the increased mass. But momentum is still conserved. So whatever momentum we have before is the amount of momentum we have after. Newton's second law. So as I said before, momentum, so 2.26 is also, I'm just going to quickly confirm this, 2.26 is also a higher only topic. Yeah. So 2.20, 2.21, 2.22, 2.23 in a way was, but it was for foundation and higher, depending on what kind of question you get. 2.24, 2.25 and 2.26. So this is also higher. So when does a change in momentum occur? A change in momentum occurs when a force is applied to an object over a period of time. So this follows Newton's second law. So a change in momentum occurs when a force is applied to an object over a period of time. So what we're saying here is change in momentum. So momentum, how do we work out momentum? It equals mass times velocity. So force equals the momentum before. So the final momentum, which is mass times velocity. So V stands for the final speed minus the initial momentum. So that's where the U comes in. U is the initial speed. So mv, mass times velocity, but that V stands for final, and mu, mass, M stands for mass and U stands for the initial speed, and then divide that by the time taken. So when does momentum change? When a football is kicked, a car crashes into a wall, a ball bounces off a wall, a cyclist speeds up or slows down. But now let's understand this in context of how, because this leads to safety as well. Actually, that will come afterwards. Now, because if we increase the time, automatically if you increase the time, you decrease the force. So the bigger the time taken for a collision to occur, the less the force is. So, numerical questions based on Newton's second law. So a tennis ball experiences a change in momentum of 4.8 kilogram meters per second. So we've just said force equals change in momentum divided by time. And here it happens over a time of 0.02 seconds when struck by a racket. So we substitute the numbers in. Force equals change in momentum divided by time. So that's 4.8 divided by 0.02, and that means the force that the ball experiences 2,400 newtons. How can we determine someone's reaction time? So this is a practical that you need to know about for the exam. Measuring reaction time, you can do the ruler drop test. So one person holds a ruler above another's hand. So that's between your thumb and another finger, and you put the ruler just above, so at the 0 mark. Without warning, they drop it, so the ruler drops, and the other person catches the ruler as fast as possible, and then you measure how far the ruler has fallen. So with this experiment, you would obviously need to do it multiple times, and you can also do it in different conditions. So you can, for example, do it using caffeine. So caffeine usually increases a person's reaction time, so you could check how much the person's reactions are improved when they've had coffee. So, but when you do this experiment, you want the valid results to be valid. So to make sure that reaction time measurements are valid and reliable, you follow these key steps. So one, you repeat the test 3 to 5 times and you take an average. But what do we do before taking an average? We remove anomalies. So you remove anomalous results. You use the same ruler and environment. You keep the hand position the same for all. So the ruler, you always have it at the 0 point just above the person's fingers. And you use accurate equipment. So if you can use digital timers or sensors, that will reduce human error. The problem with these kind of experiments is that you would have to use a timer. So, actually, sorry, you can use a timer as well to do this. So if you were timing how quickly the person catches, you could use a digital timer or sensor. Otherwise, there's no human error here. One thing that can happen is that if you're using the same kind of timing, then a person becomes used to the other person's timing. So what I mean by that is the one who's got the fingers ready to catch it, they become used to the person dropping it. They know they're going to drop it after 3 seconds, or they can drop it after 6 seconds. They start to understand the other person's pattern. Okay, stopping distance. Now, stopping distance, the key terms are, stopping distance is the sum of the thinking distance and the braking distance. Thinking distance is the distance traveled during the driver's reaction time. What that means is, I'm driving, I suddenly see a cat. The time it takes for my brain to send a message to my foot, to put my foot on the brake, the distance I've just traveled, that is my braking distance. And so that's my thinking distance. So I'll say that again. I'm driving, I see a cat. The amount of time it takes for me to process that information, send a message to my foot, and then my foot presses down on the brake, that's the thinking distance. The braking distance then is the distance the car travels once I put my foot down on the brake. Because the faster we're going, the longer it's going to take for the car to stop. Now there are things that have an impact on that. So for example, what factors affect stopping distance? So we've got to look at this from a thinking perspective and a braking perspective. So the speed of the vehicle, that would be something that affects the braking distance. The mass of the vehicle affects the braking distance. The road conditions affect the braking distance. The tire conditions affect the braking distance, the brake conditions, but whereas reaction time will affect the thinking distance. So the speed of the vehicle, the faster the vehicle is moving, the greater the stopping distance. Because it takes longer to deaccelerate. Mass of the vehicle. Heavier vehicles require more force to stop. Road conditions. So if it's wet, icy, it's got poor roads, it makes it harder to stop quickly. Tire condition. Worn out tires have less tread, meaning they don't have as much grip. So when you put your foot down on the brake takes longer to stop the tire from moving. Also, the brake condition. If the brakes are worn or not working very well, then they're going to take longer to cause they're going to take longer to produce enough friction to stop the tire. Now, reaction time. We can react to things because of tiredness, alcohol, distractions. All of these things increase the thinking distance and delay the braking. So all of these affect thinking distance. In some respects, age, younger people usually have faster reaction times. That's not always the case, but it should be the case. Fatigue, so if a person's tired, if they're distracted, alcohol and drugs lead to the brain processing things slower. So that means they put their foot down on the brake slower. Caffeine and stimulants can actually improve reaction time by increasing alertness. Stress and anxiety. High stress levels either speed up or slow down reaction time. Now, you guys are doing exams at the moment, so as you get closer and closer to the exams, for yourselves, your stress levels will get to a point where you'll actually find it optimal. So you'll find a certain level of stress will actually help you work better. And when you go past that level of stress, then you need to find something, and at that point you'll know that your stress has gone higher, because the more tired we become, our stress naturally increases. So at that point, take a break, rest, run, do whatever's necessary to just bring those stress levels down. There's lots of different things that bring stress levels down, but sometimes things work differently on different individuals. So question here, can we say dependent variable is the reaction time? Yeah, whatever you're calculating, whatever you're working out, whatever you're finding, that's a dependent variable. So in this case, when you're finding out someone's reaction time, that would be the dependent variable. Independent variable is the thing that you change. So independent variable is the thing that you change. Dependent variable is the thing that you measure. Okay, this is let me just quickly check. Yeah, 3.1 is the last part of combined. Then we move on to triple. So after this section here, if you're doing combined, you can log off. If you want to listen to triple, you're more than welcome to. Otherwise, it'll be the end of the session for combined. So dangers, so this is the last part. So dangers of deacceleration, what happens when brakes are used? So kinetic energy decreases. So we need to think about this logically. The wheel is moving, so that's kinetic energy. We've now applied the brakes. Now, as I said to you guys yesterday, if a particle touches a particle, what do we get? Friction. In other words, it's a release of heat energy. So what starts to happen is, as soon as the brake touches the wheel, we get friction. That means the kinetic energy converts into thermal energy. Now, kinetic energy equals half times the mass times the velocity squared. Now, the car's mass isn't going to change. So if you decrease the kinetic energy, what have you also automatically decreased? The velocity. What's happened to the kinetic energy is being converted into thermal. So braking converts kinetic energy to heat energy through the friction in the brakes. The greater the speed, the longer the braking distance needed to stop safely. Meaning, if you're going faster, if a person is driving faster, you've got to convert more kinetic energy into thermal, and that takes longer. How do safety features such as crash helmets actually work? So crash helmets are designed to reduce the impact force on the head during a collision, protecting the brain from serious injuries. So a helmet is actually very, very important. So it's got a hard outer shell. It's made from tough materials like polycarbonate or fiberglass. It spreads the force of the impact over a larger area. So remember, pressure equals, well, at the moment this is not something we've covered, but pressure equals force divided by area. So if you increase the area, it means you've decreased the pressure that acts on it. That prevents penetration and reduces the force on the skull. Now there's an inner foam layer. It's usually made from expanded polystyrene. This layer compresses on impact and that absorbs some of the kinetic energy. Now if it absorbs some of the kinetic energy, that reduces the acceleration and the force on the brain. It means whatever's hitting you hits you slightly slower. That lowers the risk of concussion. Then there's padding and comfort lining. The soft padding inside provides comfort and ensures a snug fit. And that improves safety by keeping the helmet in place during an accident. Now these are really important safety features. Seatbelts. Like we said before, when someone pushes the brake, you are going at the same speed as the car. But the brakes don't slow you down, they only slow the car down. So your body continues moving forward. So what does the brake do? It prevents you from being thrown forward. It provides a force, like Newton's third law, a force equal in size but opposite in direction. So it stops you from going through a windscreen. It reduces the risk of injury. Airbags. They inflate instantly during collision to cushion impact. Now before we said momentum equals, not momentum, force equals, I'll do it here, force equals the change in momentum divided by time. Now what the airbag does, because the airbag's there, the person's head has to go through the airbag before it hits, for example, the steering wheel or it hits the seat in front. That increases the time it takes before impact occurs. Now if the bottom number, the thing you're dividing by, increases, what happens to the overall answer? It decreases. So that decreases the force. So if you increase the bottom number, the thing you're dividing by, the time, you decrease the force acting on something. So this helps absorb some of the energy and it reduces the force. That's the big one on the head, chest and limbs. Crumple zones. Now in some cars what happens is this front part of the car, when it collides with something, it crumples. Meaning, before it actually collides with something, it takes longer for the collision to occur. So let's say there's a wall here. Because it crumples first, before the wall comes in contact with the people in the car, it ends up increasing the time it takes because of the crumple zone. Again, you've increased the time, so that reduces the force. So it's designed to deform and absorb crash energy. It reduces force transferred to the passengers. Okay, brilliant. Thank you very much. Everyone that was doing combined, I really appreciate the 3 hours that you've put into the session today. And like I said before, thank you so much for those people that have been sharing. It's amazing how we've gone from 400 to over 11,000 subscribers. Yesterday's video, the fact that it was watched by 1,000 people, outside 1,000 views, we really appreciate all the love and support. So thank you everyone. Have a lovely evening and hopefully see you guys tomorrow. If you enjoyed AMALing, then there's a link below and you can register yourself for AMALathon 2. Now some of you do triple award, so if you want to stay logged on, that's absolutely fine. So, work and braking. 2.32. How stopping distance changes with speed. So the total stopping distance of a vehicle in an emergency depends on two main components: thinking distance and the braking distance. So as speed increases, both thinking distance and braking distance automatically increase. Just because if you're going faster, if it takes you 5 seconds to do something, if you do it at 50 meters per second, then you're going to move 250 meters. So if you're doing something at it takes you 5 seconds to do something. If you are doing something at 50 meters per second, after 5 seconds, you've already traveled 250 meters. But if you do something in 5 seconds and you were only doing it at 2 meters per second, then you've only traveled 10 meters. So the speed has a massive impact. Braking distance increases exponentially. So if you double the speed, it quadruples the braking distance. If anything, it squares it because it's v squared, kinetic energy equals half mv squared. The overall stopping distance is much greater at higher speeds, making high speed driving more dangerous in emergencies. And this is just an example. So the whole point of 2.32 is just understanding that the faster you go, the longer it takes to stop. And it's more of an application topic. So there's nothing, in terms of the learning, there's nothing actually too complex there. It's just that if you're going faster, you're going to take longer to stop. So like I was saying to you guys, if you're going at 20 miles per hour, in that time you take to think, you've traveled 6 meters. But if you're going at 70 miles per hour, because you're going so fast, in that same thinking distance, in that same time it takes you to think about it, you've now traveled 21 meters. So that's a massive jump up. And braking distance wise, if you're going at 20 miles per hour, it's 6 meters. You can brake within 6 meters, so total stopping distance of 12. But if you're going at 70 miles per hour, it takes 75 meters before the car stops. So a total of 96 meters. Now, if the car's here and the object is 20 meters away, if someone's driving at 70 miles per hour, they're not going to stop till 76 meters after that person. Whereas if the person was driving at 12 miles per hour, they'll stop here. 30 miles per hour, they'll be on the verge of stopping. So they might hit the person lightly, but they'll still end up hitting the person. That's why in residential areas, around schools and things like that, the speed limit is 20 miles per hour, if not less. So higher speed equals longer stopping distance, which means more of a crash risk. So it's important to keep a safe gap to stop in time. What happens when the brakes are used. So this is similar to what we covered before. When a driver presses the brakes, the car's kinetic energy converts to thermal heat energy due to friction between the brake pads and the wheels. So what I explained before, I already explained it from a triple perspective, but I'll just go through it one more time. And that's because the friction between the brake pads and the wheels. So you've got your wheel, you've got your brake pads, and as they come in contact with each other, more of the kinetic energy converts into heat energy. And as more kinetic energy converts into heat energy, because kinetic energy equals half mass times velocity squared, the mass isn't going to change, so instead the velocity decreases as kinetic energy decreases. So let's look at this step by step. The driver applies the brakes, creating friction between the brake pads and the wheel discs. The car's kinetic energy is converted into thermal energy as the brakes slow it down. The car travels a certain distance before stopping completely. So what happens when the brakes are actually used? Let's link this to work done because that's important. Work done equals force times distance. So the more braking force, the shorter the distance. Higher speed, there's more work done needed. Because if you have a greater braking force, you need a shorter distance. So at higher speeds, more work needs to be done. So that means more heat in the brakes. Now, what are the key factors affecting braking efficiency? If you have more speed, more energy to remove. If you've got brake conditions are poor, then less friction. If the road conditions are wet or icy, that means less grip. And if the tire conditions are poor, there's less traction. So conclusion, brakes convert kinetic energy to heat via friction. The higher the speed, the longer the stopping distance. Okay, that's the end of the physics section. Now, if you guys want to let me know, I can move straight on to the biology triple content that I was going to cover today. Or I can give you all a 5-minute break first. It's entirely up to you. I think it'll take me about half an hour to go through the content. So if you guys want to let me know what you prefer, I'll work with that. So you guys can make that decision while I change the PowerPoint. Okay, that's fine. Let's continue. We've got 2 sections to do from topic 1, and there's a few sections. Thank you, Saif, for your feedback, for commenting in the live chat. So we're going to go through 1.12. Sorry, 1.11 and 1.12, 1.13 and 1.14 from this PowerPoint, from this presentation, and then we've got a few specification points to go through link to the brain and the eyes from topic 2. Like I said, if anyone needs to log off, it's absolutely fine. This will all be recorded. So 1.12, explain the importance of enzymes as biological catalysts in the synthesis of carbohydrates, proteins and lipids, and their breakdown into sugars, amino acids, and fatty acids and glycerol. So why are enzymes important? What enzymes do, they're biological catalysts, as we mentioned yesterday, and they break down lipids into fatty acids and glycerol. They break down starch into glucose and sugar, and they break down protein into amino acids. Now what they do is they lower the activation energy. So enzymes reduce the energy needed for reactions to occur. They speed up reactions. They are catalysts, they're biological catalysts, so they speed up the speed at which the reaction occurs. And each enzyme works on a specific substrate, ensuring the correct reaction takes place. So that means lipase only breaks down lipids into fatty acids and glycerol. Carbohydrates only breaks down starch into glucose. And protease only breaks down proteins into amino acids. And enzymes help control and coordinate metabolic pathways. The main thing that we need to look at the moment is this one, and this one. And they help with essential processes. They enable key functions like digestion, DNA replication, and energy production. That's important for us to know. So, enzymes can be used to make proteins from amino acids. So enzymes, not only do they break down proteins, enzymes are also used to combine proteins, so to make proteins. They work in both directions. And which enzyme works on proteins? It is protease. So this is important to understand, especially from a digestive system perspective. Starch and sugars. Carbohydrates, again, they work in both directions. They make the large macromolecules, and the macromolecules are also broken down to make the monomers. So this is our polymer, starch, and these are our monomers, glucose. And carbohydrates will combine glucose, the sugars, together to make starch, but it will also help break down the starch into individual sugars. And it's the same thing with lipids and lipase. The lipase enzyme is used to break down lipids and fats into fatty acids and glycerol, but lipase is also used to combine them to make lipids. So these are some common enzymes that you will need to know a little bit about. Amylase, we find it in the saliva, in the small intestine, and that breaks down starch into small sugars such as maltose. So it's a carbohydrate. Starch synthase is something found in plants, and that breaks down starch into glucose. Sorry, it makes starch from glucose. It's a synthesis of starch, so it makes starch. And DNA polymerase is found in the nucleus. RNA from the DNA template, and that is essential for protein synthesis. The RNA helps combine amino acids together to make proteins. So that's something we'll cover in, I think, tomorrow in the triple session, where we'll cover protein synthesis. A lot of the stuff we're covering today is quite straightforward. What we'll be covering tomorrow will need a little bit more time. So protein synthesis is one of those topics that, if you understand it, it's a really straightforward topic, but you've just got to understand it in detail. So now, describe and explain the names of chemical reagents and processes through which they help to identify starch, reducing sugars, proteins, and fats within food. So this is one of the core practicals. So in the core practical, you need to know how we can test for different macromolecules, different so we can test for different for proteins, for macromolecules, for proteins, carbohydrates, fats, etc. Now, things like milk and juice, they're already a solution, so we can test them as they are. You can add chemicals straight to it. The chemicals mix, and you get a colour change and you know what the substance is. But if you've got a piece of food like a piece of bread, then you need to grind it into a paste with a pestle and mortar first, add water to it and stir it so it becomes a solution. Now, testing for starch. If you add iodine to a food sample, if there's no starch, the colour doesn't change. If there is starch, then it turns blue-black. So iodine is an indicator that turns blue-black in the presence of starch. And it's harmful. So the safety precaution is avoid contact with skin. Or you can wear, and of course you should wear safety goggles. By the way, everything we're going through from topic 1, 2, 3 and 4, we have on AMALearning on our YouTube channel, which you're already on, we have animations that cover every single specification point. So now you've got access to this recorded AMALathon video, but you've also got the animations as well. Testing for glucose. You add Benedict's reagent to the food sample, heat it. If there's no glucose, the colour doesn't change. But if there is glucose, it turns red, brown, orange, yellow or green. Now, red being lots and lots of glucose, and the lighter the colour, that means less glucose. Protein. You add biuret solution or burette solution reagent to the food sample. If there's no protein, no colour change. If it turns this kind of mauve, purplish kind of colour, it means it does contain protein. Again, burette solution, it is harmful. And for that reason, safety goggles are needed and avoid contact with skin. Testing for lipids. You add ethanol and distilled water to the food sample and shake. Now, if there's no lipid, the colour doesn't change. If it turns to a cloudy white colour, then it shows that there's fat there, lipid there. Or you can just rub the food on a piece of paper. If it leaves a grease mark, that means it contains fat. 1.14b. Now, this is linked to the calorimeter. Again, this is one of those topics, it does come up and not everyone has covered it in enough detail to answer the questions. I've seen this come up a few times and it's one of the most poorly answered questions, particularly from a triple perspective. Well, it only comes in triple, so it can only be from a triple perspective. How can the energy contained within food be measured? So, a calorimeter is a machine used in the process of calorimetry. It's where you get a small sample of material, it's placed in the machine until it burns. So when you burn that material, it releases heat energy. That heat energy then, that is given off, is measured. Now how can we measure it? You can measure it just by heating up water. So calorimeters are often used in food industries to measure the energy in food. So the energy given off when the material burns is measured. So look, what we're doing is we're getting a piece of food, we're burning that piece of food, we're heating something up, so then we can calculate the change in energy, how much energy that food has given off. And the way we can calculate it is energy equals mass times specific heat capacity times the change in temperature. But you don't actually need to use that within biology. So how can we do that for this 10 gram of food? So we've got a thermometer, 100 centimeter cubes of water, and a combustion chamber. So calorimeters usually have a combustion chamber. So you put the food inside the combustion chamber, you burn that piece of food. Step 1, you measure the start temperature of the water. Afterwards, you're going to measure the end temperature as well. You burn the food to heat the water. Step 3, you use the increase in temperature of water to calculate the energy change. So you will need to use this formula, but there's no Q at the end. So now, if you look at the information that we've got, Q is the energy absorbed by the water, M is the mass of water, C is the specific heat capacity of water, and change in temperature is the temperature change. This is a bomb calorimeter now. So what we've got this time is slightly different. So if you look at it, you've got a thermometer to check the change in temperature. You've got ignition wires that are attached to the food, so the food can be set alight. You've got the water there that's going to heat up, a lid to stop the heat from being lost, and you have a stirrer, because when that stirrer starts moving, it helps the heat spread faster, so you get better results. So how can the energy contained within food be measured? The simplest calorimeter can be made by arranging a beaker of water above a burning sample. So you take your food here and burn it, and then you measure the temperature change. So the change in temperature of the water is a direct measure of the energy held within the sample. How can the energy be contained can be measured within food? So you burn the food to heat the water. The temperature change is measured. You measure the start and end temperature of the water. The overall increase in temperature of the water can be used to calculate the energy content of the food. Okay, excellent. So what we've just finished there is, so whoever, I'm not sure who this is, but Elite Dream, this is all Edexcel. So is this AQA or Edexcel? The answer to the question is AQA starts from 10 a.m. till 1 p.m. every day, and Edexcel is 1:30 till 4:30 p.m. But there's this additional session that we're doing right now, which is just for triple award, because Edexcel triple award is really extensive in paper 1. So this just gives the additional time to go through it. Whoops, I've opened the wrong one. Apologies. So now what we're going to do is we're going to look at the brain, and we're going to look at the eyes, and then we're going to stop. Now again with the brain and what we're about to cover, as long as you memorize the key facts, and what I would advise you do after this session, or if as soon as you can, is go through all questions linked to the brain from past paper practice. And what you're going to find, is it's the same kind of question that come up again and again. Because if you look at the brain, if you were to speak to a neurologist, a doctor that's linked to the brain, even they will say they don't know much about the brain. I mean, they know way more than us, but it's such a complicated topic. So what we're going to go through now is 2.10 to 2.12, and then we're going to go 2.15 to 2.17. Okay, as soon as this loads up, apologies for the delay. And it's the same with the topic of the eye. It's similar questions every single time. So my advice is go away, and my advice, sorry, not to go away. My advice is go through as many past papers as possible, and just have a look at the questions linked to the brain, have a look at the questions linked to the eyes. And what you're going to realize by tomorrow is it's the same thing over and over again. So 2.10b describes the structures and functions of the brain, including the cerebellum, the cerebral hemispheres, and the medulla oblongata. Now, what you need to be able to do is label and describe the function. That's all 2.10b is. So the brain is an organ that controls complex behavior. It's made of billions of interconnected neurons and has different regions that carry out different functions. This is the bit that you need to know. The cerebral hemisphere. What does it do? It is the part of the brain that controls intelligence, personality, and conscious thought. It's responsible for decision making. Then we've got the cerebellum. It's the part of the brain that controls balance. It coordinates movement and muscular activity. And then medulla oblongata It is the part of the brain which controls unconscious activities such as breathing and heart rate. So let's quickly repeat that. Cerebral hemisphere is the part of the brain that controls intelligence, personality, and conscious thought. The cerebellum controls balance, coordination, movement and muscular activity. And the medulla oblongata controls unconscious activities such as breathing and heart rate. So let's have a quick look at this practice paper question. This is an actual question from a past paper. Or it's been slightly reworded, but it's essentially the same. Which region of the brain is labelled structure X? So which parts label structure X? And then the second part of the question is describe the function of X. So it's the cerebellum. So if we go back, there, cerebellum. So the cerebellum is this part here. The medulla oblongata is that part there. And what does the cerebellum do? It's the part of the brain that controls balance, coordination, movement and muscular activity. That's the kind of question that comes up for 2.10b. Okay, 2.11b. Explain how the difficulties of accessing the brain tissue inside the skull can be overcome by using CT scans and PET scans to investigate brain function. So how do we know what different regions of the brain actually do? One way to know about the different parts of the brain is through studying patients with brain damage or injury. So, for example, if someone has an injury to a certain part of the brain, they would discuss with them how their behavior has changed. They would look at how they respond to different activities to see how the brain has been impacted. Damage to different parts of the brain leads to different behavioral and psychological effects. So for example, if someone damages the front part of the brain, it impacts the ability to make decisions. If it's the back part of the brain, then it affects the person's vision. How do we know what different regions of the brain do? So some of you may have come across this story. For example, in 1800s, Phineas Gage was working and an iron rod went straight through his skull, similar to what you can see in that image there. He survived, but his personality changed. So at that time, doctors would have done experiments. They were much harsher in the 1800s when they did experiments. They wouldn't have been nice experiments. But when people have had accidents like this, it does allow them to see what the issue is or how it's affected the brain. Now, how do we know what different regions of the brain do? So another way of mapping the brain involves electrically stimulating different parts of the brain by using weak electrical currents. The patient's awake, so they're able to describe what they experience when the brain's stimulated. This may be that they see something, they might remember something, or they might smell something. If a motor area of the brain is stimulated, the person might even move involuntarily, without knowing, involuntarily. So how do we know what different parts of the brain do? We've also got, so neuroscientists are scientists that study the structure and function of the brain. As the brain is protected by a skull, accessing the brain tissue is difficult. So getting to the brain tissue in the first place is difficult because the skull is such a strong protective layer. This is why neuroscientists use CT or PET scans to understand the brain function better. So these are examples of CET and PET scans. Now, a CT scan uses X-rays to produce detailed two-dimensional images of the brain. So that's what we've got here. Joint CT scan hundreds of images. So not joint, sorry, that should say during. During CT scans, hundreds of images are taken which show the doctors or neuroscientists the main structure of the brain. So they're almost like cross-sections. Cross-section after cross-section after cross-section. So you've got the whole brain like that. is taking images here as cross sections. The CT scan does not directly show the function of the brain, but it does highlight parts of the brain which are diseased or damaged. Now the PET scan uses radioactive tracer chemicals to show which parts of the brain are functioning whilst in the scanner. So more active parts of the brain showing red or orange, and the least active parts of the brain can be seen as green. So depending on what activity the person is doing, you can start to see what parts of the brain are stimulated. So PET scans are useful for studying disorders like Alzheimer's disease. So with Alzheimer's, this is a neurological illness which impacts how quickly messages are sent through the sensory neurons or through nerve cells. So by looking at which parts of the brain are red and orange, it helps them to start understanding what parts of the brain are starting to break down or aren't working as well. 2.12b explains some of the limitations in treating damage and disease in the brain and other parts of the nervous system, including spinal injuries and brain tumours. So why is it difficult to treat the brain? When neurons or nerve cells are lost, they don't regrow. So nerve cells, neurons, they don't regrow. What that means is neurons do not automatically repair themselves, unlike some other cells in the body. So in future, stem cells may be used to treat spinal cord injuries. However, right now, injuries or conditions linked to the damage of neurons are permanent conditions. So the issue here is that neurons and nerve cells, when they're damaged, so for example the spine, it's a permanent damage. But through stem cells, it's possible in the future there'll be cures. Now the brain and spinal cord have a protective membrane around it. You can see it as a colour, well it's just under, on the screen you can probably see like an outer layer which is like a light blue. Just under it you can see like this greyishness just before you start seeing purple and orange. That's your protective membrane. Both the brain and the spinal cord are protected by our skeleton, so our skull, and our spinal cord, our spine, sorry. The brain is protected by the skull and the spinal cord is protected by our vertebrae, our spine. This makes accessing the brain and the spinal cord difficult because if any damage is done on accessing it, it's permanent damage. So state one way the brain tumours can be detected. CT scans or PET scans, but you can accept MRI and X-ray. So you can see with 2.10, 11 and 12, all the questions that you get asked are, or the majority of the questions are recall. They are memorize and answer type questions. Describe why brain tumours are difficult to treat. And these last two questions, you memorize them. You've memorized so much. By memorizing this mark scheme, you've understood so much of 2.12. The brain is protected by a skull. It's difficult to access. Nerves do not repair and regenerate. And there's a risk of damage to the brain. It also has the thin membrane around it, which if damaged, that can cause more problems. Okay, now we're going to cover 2.13 and 14, the nervous system, tomorrow. And so 2.13 is the nervous system, 2.14 is the reflex arc. We'll cover both of these in detail tomorrow. This is going to be our biology lesson from 1:30 till 2:30. But what we're going to go through now is 2.15, 16 and 17. And then that will be the end of our AMALathon session day 2. So guys, really appreciate everyone that's still logged on. And those that are listening to this afterwards, really appreciate the support of everyone. Now explain the structure and function of the eye as a sensory receptor, including the role of the cornea and lens, the iris, the rod and cone cells in the retina. So what are the different parts of the eye? So first part is recall. It's understanding that the lens is a transparent biconvex structure in the eye that refracts light into the retina. So the whole point is that when the light hits the lens, it's refracted. The lens in the eye is transparent, flexible, curved structure that helps focus light onto the retina. So it's just memorization. The lens is transparent biconvex structure. It refracts light onto the retina. That's the main thing there. Now we've got ciliary muscles. These muscles are connected to the lens by the suspensory ligaments. They control the shape of the lens to help the eye focus on near and distant objects. So the ciliary muscles help change the shape of the lens, and that's something that's about to come up. As it is, it's called accommodation. It's coming in the next few slides. The suspensory ligaments are connected to the lens. They connect the lens to the ciliary muscles. Their job is to hold the lens in place and change its shape during focusing. Then we've got the pupil. That's this hole just here in the middle of the iris that lets light in. The diameter is controlled by the iris. So in darkness, the iris retracts, relaxes so that the pupil becomes bigger and more light can go in. In brightness, the iris will make the size of the pupil smaller, so less light goes in. So here we've got the iris. The iris is the coloured part of the eye. It controls how much light enters the pupil. Then we've got cornea. It's a transparent layer at the front of the eye. It refracts light. So we have the cornea that refracts light, and we have the lens that refracts light. So both of these refract light. Now, when we get to 2.17, we'll be looking at how the body, what we have to do if the cornea gets damaged. So when we say cornea refracts light, what do we mean? We mean that the cornea bends the light. It changes the light's direction to ensure the light enters the eye. It is something that we'll cover in physics in a lot more detail. What are the different parts of the eye? So we've got the sclera. Sclera is the white outer layer of the eye that protects it from injury. Now, if anyone's ever done an eye dissection, they'll know that when they're dissecting the eye initially, there's a tough outer layer. That's the sclera. That's what protects us, protects our eye. So if a finger goes into your eye or something pokes your eye, it's the sclera, because it's tough and strong. It helps protect the eye and hold its shape. The retina. The retina is made up of a group of cells called the rods and cones that are sensitive to light. The retina is made up of rods and cones. Rods detect light intensity and work in dim light, whereas cones detect colour and require bright light. So cones start with a C and they detect colour. Rods detect light intensity. They work better in dim light. Cones work better in bright light. So at night time, so for example, you wake up in the middle of the night and you can't see anything because you just opened your eyes. Or when you enter, for example, a dark room, initially you can't see anything, but afterwards you start to see things. That's because your body breaks down the cones, so it removes the cone cells and it increases the rod cells. Now, as soon as you open your eyes in the morning and you're struggling to see something, some people pick up the phone and they can't really see what's there because their eyes can't, it's too bright all of a sudden. At that point, the body starts making more cone cells to help you deal with the bright light. And while your eyes are adjusting to that bright light, they're adjusting while there's enough that adjustment phase is because the cone cells are being produced. Now we've got the optic nerve. So the optic nerve is a sensory neuron, which is something we're going to cover tomorrow as part of the nervous system. Now, rod and cone cells in the retina, they detect a stimulus and then detect the light. They then send the message to the central nervous system through the sensory neuron, which in this case is the optic nerve. What are rods and cones? So these are your cones and these are your rods. So rods enable us to see in dim light. Rods are receptor cells. So if you're not sure what receptor cells are, we are going to be covering it tomorrow as part of the nervous system. But they detect the light and they detect differences in light intensity. They do not detect color. We have the cone cells for that, but they are very sensitive to light. Now here, these three, we've got cone cells. They enable us to see objects clearly in bright light and in color. There are three types: red, green and blue. They do not function well in dim light, and they're less sensitive to light. They are more sensitive to color. Now, how do our eyes help us to see objects that are near or far? So this is what we call accommodation. So it's the process of changing the shape of the lens to focus on something which is near or distant. Now, if something's near, the ciliary muscles contract. Now again, if you look through past papers, it's the same questions again and again. Everything I've just covered with you from a triple perspective is all recall. You'll notice as well, you need to understand it, which I'm sure if you're all doing triple, so I'm sure you understand to a really, really high standard. You've got to understand, but there's a lot of memorization here. It's a lot of recall at the end of it. So ciliary muscle contracts, the suspensory ligaments loosen. So this is we're looking at something from far away. The lens becomes thicker, meaning it becomes wider. And that means it refracts the light rays more strongly. So it bends the light more. Now if we're looking at, so that was when we were looking at something close by. So if this is our lens and we're looking at something close by, it needs to refract it more. So it can focus on the back of the eye. Now if we're looking at something from far away, then the opposite happens. So before we said ciliary muscles contract, for a far away object, ciliary muscles relax. And the opposite, the suspensory ligaments are pulled tight, they don't loosen this time. And that means the lens is pulled thin. So I've crossed that out rather than. So now the suspensory, the lens is pulled thin. So when the light comes from a distance, it doesn't need to be refracted too much. When something's close by, the lens becomes fatter because it needs to refract more. When you're looking at something far away, it doesn't need to refract as much. So the lens becomes thinner. Now, how does the eye respond to dim and bright light? In bright light, the pupil reduces in size. So that means again, this is memorization, the radial muscle of the iris relax. Now everything we're saying here to make the pupil reduce in size, when we say we want the pupil to increase in size, it's going to be the opposite of everything that we're learning now. So the radial muscle, instead of relaxing, will contract. So when the pupil reduces in size, that reduces the amount of light entering and that protects the retina. In dim light, the pupil increases in size, so the radial muscle of the iris contracts. The circular muscle of the iris relaxes. Now that's missing from here for some reason, but the circular muscle, M-U-S-C-L-E, muscle, so the circular muscle contracts. So that's been accidentally missed out in the notes. Now, so in dim light, the pupil increases in size, the radial muscles of the iris contract, The circular muscles relax. That lets more light into the eye to help you see better. Explain the function of the rod and cone cells. So rod cells enable us to see images in dim light. The rods are receptor cells that detect differences in light intensity. So this is what we've already covered, so I'm just going to skip past this now. And that was only there as a question. So I'll pause so you can see the answer. So afterwards, when you watch the video, you can have a look through the question. But what you'll see is it's exactly what we've covered. This is just another multiple choice question. So again, if you're watching the video, you can pause and then have a go answering it. So the answer is B. When the eye changes from focusing on a distant object to focusing on a near object, the lens gets thicker to bend the light rays more. Okay, I'm just going to skip past these questions because when you go through them in your own time, what you're going to notice is that it's all recall. 2.16b. What are some common defects of the eye? The following are common defects that affect vision quality. So one is myopia or short-sightedness, and hyperopia or long-sightedness, cataracts, and color blindness. So let's go through each one at a time. So two common defects of the eyes are myopia and hyperopia. So a lot of people have at least a little bit of one, or a lot of one. Most people have some link to myopia or hyperopia. So myopia is when a person is short-sighted, so they can see things close. When something's far away, the image is focused in front of the retina, meaning it doesn't end up focusing on the retina where the cone and rod cells are. Now, hyperopia, long-sightedness, is where the image is focused behind the retina. Now, if someone's short-sighted, a short-sighted person is someone that can see things that are close or short distance from themselves. So they are short-sighted, they can see things that are close by. But people who are short-sighted cannot focus on objects that are far away. So it usually occurs when the eyeball is too long. And that means the light is refracted too much by the lens. And if the light is refracted too much by the lens, it means the light image focuses in front of the retina. Light rays from a distant object focus on the eyeball in front of the retina. Long-sightedness is the complete opposite. So it's someone that can see things far away, but they can't see things that are close or nearby. And that's because the eyeball is too short. And that means the light is refracted too little by the lens. Now, if the light is refracted too little by the lens, it means the light doesn't bend as much, so the image forms behind the retina. So everything looks blurry. Now, what are cataracts? 2.17 will cover how to cure these things. So 2.16 is just the defects. So a cataract, it occurs when protein builds up inside the lens and makes it cloudy. So you can see the cataract here. That protein is built up in the lens and made it cloudy. Due to the cloudy patch on the lens, light is unable to enter the eye normally. Cataracts are when the lens of the eye becomes cloudy. This makes it harder for light to pass through, causing blurry vision. Finally, color blindness. Color blindness means a person has trouble telling certain colors apart, usually red and green. So it's an inherited condition, means a person's born with it. More men are colorblind because the gene is actually carried on the X chromosome. So this is something that you need to understand from a genetics perspective. So when we start covering genetics, which will be on Tuesday, for those who are doing triple, well, everyone that's listening right now, we'll be covering that as a topic. So sex-linked inheritance. So the most common condition is red-green color blindness. That means the red and green cones in the retina are faulty, making it difficult to tell the difference between red, green and brown. Unfortunately, there's no cure for this condition. Okay, now these are past paper style questions, which I'll show really slowly, so afterwards it comes up on the recording, so you can compare the questions to the answers. Because the main point of our sessions at the moment is just the understanding. Next time, when we have our sessions in May for AMALathon 2, then we'll do a lot more past paper practice as well. Now describe why a person with cataracts could not see the letter E clearly. Because cataracts occur when protein builds up inside the lens and makes it cloudy. So due to this cloudy patch on the lens, light is unable to enter the eye normally, and so they see it as blurry. And there's the mark scheme. Okay, this is the final unit that we're going to do, specification point that we're going to do. So I really appreciate everyone that's stayed logged on. Explain how cataracts, long-sightedness and short-sightedness can be corrected. Now the reason why colour blindness is not here is because colour blindness doesn't have a cure. So how are these defects corrected? They can be treated with spectacle lenses which refract light rays so they stay focused, so the light focuses on the retina. So both myopia and hyperopia can be treated with the glasses. So myopia, you use a concave lens. So a short-sighted person is someone that can see things that are close by or a short distance from themselves. So that means the light's coming from quite far and it ends up refracting the light too much. So what the concave lens does, it refracts it outwards just slightly. So the concave lens bends the light rays outwards so the light rays focus on the retina. So this is something that needs to be memorized. What a concave lens is and how it actually helps myopia. Now hyperopia uses a convex lens. Now here, the light is not being refracted enough. So this convex lens does some additional refracting inwards so that it focuses on the retina. So this is a person that can see far away but can't see something close by. The convex lens bends light inwards so that the light rays focus on the retina itself. How are these defects corrected? So people other than glasses, they can use laser eye surgery. They can use hard and soft contact lenses. They can, if someone has an issue with a lens, for example cataracts, they can even replace defected lenses with new ones. So laser eye surgery, the outer layer of the cornea is peeled back and lasers are used to change the shape of the cornea so the light rays will refract and form an image on the retina. So if you thin down the cornea slightly, it won't refract as much. One of the main advantages of this is a permanent solution. However, it's a surgical procedure which could cause infections. So contact lenses, they can be hard or soft. They're inserted directly into the eye and sit on the cornea. Contact lenses are small, clear lenses you put directly onto your eye to help you see better. So the advantages of contact lenses are they're lightweight, they're almost invisible, and they're good for people playing sports as glasses can fall off and break. But contact lenses are not a permanent solution to the eye defect, and they can cause irritation and infections. So lens replacement surgery. In other words, the defective lens is removed and replaced with a new artificial one. So if someone's got cataracts, you can actually take out the old one and replace it with a new one. That's a permanent solution, so that's a positive. However, there's disadvantages because it is a surgical procedure. It can lead to infections. So how are cataracts corrected? Cataracts occur when proteins build up inside the lens and make it cloudy. So cataracts can be cured by removing the clouded lens and replacing with an artificial plastic one. So what we covered in the previous slide, just a bit more detail. So this is an exam question. Myopia or short-sightedness can be corrected with concave lenses. The concave lens bends the light rays outwards so that the light rays focus on the retina. That would get you 2 marks. Cataracts can be cured by removing the clouded lens and replacing with an artificial plastic one. So this is explain how cataracts can be treated. So if I just go back one question, explain how a concave lens corrects short-sightedness. This is what we've just covered a few slides before. And so essentially what you're doing to answer this question is you're memorizing it. So look, a diverging lens or a concave lens refracts light outwards, so the light rays focus on the retina. And in this question, how are cataracts treated? You remove the clouded lens and replace with the artificial plastic one. Let's see the mark scheme. Remove the lens, replace with a plastic lens. Okay, brilliant. Thank you everyone. I really appreciate all the hard work you put in today. I hope you all get amazing GCSE results. I'll see you all tomorrow at 1:30. Thank you guys. And if there's any topics you want to go in through more detail, then if you go to AMALearning, it goes to the YouTube channel. There's lots of different animations there that cover biology topic 1 onwards. Thank you everyone. Keep AMALing. Take care. Have a nice evening.